Thank you.
Oh,
I am not hearing anything online from the speaker.
yep.
I didn't.
I'm muted, yeah.
Yeah. And then you can turn the camera on.
We can hear you now, Berton. I don't know that this camera will matter.
Yeah.
Oh, perfect. You can see it. Yep. Can anybody hear me online? Yes. Can you hear us? We
should have.
Yeah, I don't know.
Yeah.
That's one positive response.
Someone say something online.
Okay, they can hear us. Okay, so
Okay.
simply unmuting seemed to have worked. Surprise, surprise.
Yeah.
Cassiano, we're not sure if we can hear you. Do you want to try? Can you hear me now? I think it's connected to your computer, Burton. That's the problem. We would need to connect to the CVPR computer, but it still looks like muted for some reason. Well, we can hear you on my laptop. It's a bit janky, but it could work. You could like. Can I scream really loud and then yeah, make this sound?
We need to change in the Zoom. Oh, actually, you can open sound again. It says the Epson.
That's a projector.
Yeah, I don't think we want that.
I see.
We're just gonna put the microphone to the speakers and we're gonna get started.
Yeah.
I'm thinking about
Yeah. Can you hear me okay on your computer, Burton? I can hear you, and I'm pointing my mic at the speakers on my laptop. And keep talking. I'm getting some positive
Can you
feedback.
guys hear me on the screen there? Do we have like some thumbs up?
Yeah, there's thumbs up.
Okay, perfect. So we're gonna, without much delay, I'm gonna introduce. Just a small note, like you guys are muting the first part of the workshop, so you kind of miss the people that's online. I think miss the initial part. But without much delay, hello everyone. My name is Cassiano Carmel. I am one of the directors here of Recursion of In Vitro Technologies. And today, one of the co-organizers of this year's CVMI workshop, and it's my pleasure to introduce Dr. Berton Earnshaw to the talk today. Berton is a founding fellow at Recursion and the scientific director at Valence Labs. He earned his PhD in mathematics from the University of Utah and has an impressive career working in many scientific and leadership roles in the industry. At Recursion, Berton has led the development and deployment of many of the machine learning capabilities that are the core of Recursion's drug discovery workflows, some of which he'll begin talking about today. Currently, he is directs multiple research programs across Recursion and Valence Labs, pioneering the driving innovation in the field of tech bio and the AI-assisted drug discovery. A few fun facts about Burton. He is the father of 5 lovely children and recently climbed the Mount Kilimanjaro with his oldest son. He also won a cooking competition in Salt Lake City. Some of you in the audience may know about what I'm talking about. And finally, he's as friendly as he's smart. So I'm sure that the guy's going to have a lot of fun interacting with him. For the ones that, if there's anyone that speaks Portuguese, he's a fluent speaker of Portuguese as well. With that, I'm going to pass the mic to Berton to talk about the exciting technology that the group's developed at Recursion. Thank you.
Thanks, Cassiano. Slightly embarrassing, but thank you nonetheless. Also, what's funny is that cooking competition actually just barely happened in Salt Lake City with our some of our collaborators at Genentech who are in the audience. And you'll be glad to know I didn't wear my medal today. Okay, thank you to the organizers, many others for the invitation to speak today. I'll be speaking on some of our research into how best to represent microscopy images, cellular microscopy images, how we're building foundation models, how we're testing the scaling hypothesis for this kind of data. Happy to take questions throughout. I don't think I'm going to be able to monitor what's in chat, so well, someone else will have to help me figure that out. Just to set the stage, the thing that we're primarily trying to do here is to take images of cells like these. This is a kind of a characteristic set of cell paint images, sort of faux colored, because there's actually 6 different channels being combined here together into a RGB image. But to be able to say something about the underlying biology happening in the cells in these images from the morphology of the cells themselves. So a kind of inverse problem of taking the sort of functional response of cells read out by their cellular morphology and somehow inferring what is going on with the cell state in response to perturbation or other changes in that state. First, though, we're going to go back in time to 2021. I was sitting here regretting not having put masks on their faces, but go back to 2021 when I gave another keynote talk here in this workshop about the progress that Recursion had made up to this point in this problem of representing the biology in these cellular images. And we put at that time, we had recently released a data set publicly that we called RxRx1. It's the first in a series of public data sets that we have been releasing and will continue to release. And it was primarily a data set for studying how one would do batch correction in data sets generated like these, right? So this particular data set contains about 125,000 of these images. But what it is, it consists of 51 sort of runs or batches of the same experimental design, run 51 different times in different cell types, but always the same design in the sense of the same perturbations, the same treatments, the same control strategy, and so forth. And we actually had created and ran a Kaggle competition at that time, actually started in 2019, to also sort of crowdsource what people would do with these images and working on this problem. And the simple problem was could you classify the perturbations in these images, basically the perturbation, in this case, an siRNA, so a kind of genetic knockout or knockdown that had been applied to the cells in each of these images. And that competition went really well. We learned a lot both from that competition and internal research. And around 2021, we were representing the content of these images by training classifiers in this weakly supervised way to predict the perturbation that had been applied to the image. We were using convolutional networks to deal with batch effects. We were using an adaptive batch norm, which is very much like the batch norm you know and love, but it at test time does not use the learned statistics from the training set, but actually continues to use the statistics from the mini batch even at test time. And so it's important to sample from the correct domain to get the right statistics, so on and so forth. We were training these models on RxRx1 because well, an internal version of it, because it performed so well. And I'll talk about like what, as we try to move away from this, what kinds of things we were seeing. And we were doing kind of the typical things you would do with convolutional neural networks, like for regularization, augmentation, channel dropout, cut mix, things like that. So this is kind of state of the art 3 years ago for us. And it drove a lot of progress. We wrote up a paper on this that we put up on an archive last year and was presented in some workshops about all this work, about our kind of studies on ways to do end-to-end batch correction while learning this weakly supervised task, showing what we thought was best and just sort of the recipe behind how to do this all. Good. So we have used
these sorts of techniques to do all kinds of at recursion drug discovery tasks. For example, hit identification. So hit identification is I've got a cellular disease model. Think of something like COVID-19. We can infect the cells with the SARS-CoV-2 virus, and that's going to give us a disease phenotype. Sometimes it's very obvious. By the way, these pictures right here actually don't correspond to that particular case. I'm just giving you an example. But sometimes those phenotypes are visually, you can see them visually with your own eye. Oftentimes they're far more subtle than that, and the networks do the job of sort of featureizing so that we can have a more sensitive readout of that and be able to kind of to look at them. But the idea is, in hit identification, is if we can represent the disease and we can start applying drugs to that disease model, the cellular model, then we can start to see, do any of them seem to return us to what the healthy population looks like? And in these pictures here, you're seeing kind of just the way we think about this. We embed all of these images, or actually the aggregate, usually the mean of a sample of these perturbations, these images corresponding to the same perturbation or the healthy treatment, into a vector space, and we start to reason about kind of the differences between them in the geometry of that space. So we start to think about where the vector lies, how if we were to, if we had something that represented the disease, like if we take now the disease plus the compound images, we can project onto that particular vector. We can kind of see how aligned it is, if there's sort of like other stuff going on that we term an off disease score. And we can start to do these kinds of plots you see on the far right, where you have sort of a distribution of the disease, a distribution of the healthy, and you're looking for compounds that, as you increase the concentration, you sort of trace out this trajectory that takes us from the disease cloud to this healthy cloud, and we're looking for anything that gets us back into that healthy cloud in a reasonable way with a reasonable dose. And so you see kind of two examples here, remdesivir and hydrochloroquine. One, remdesivir returning you to the healthy state, and hydrochloroquine not. And actually, we at this time, we created a data set, two data sets called RX-19A and B. One where the model was early stage COVID, the other being late stage. And using these and this kind of technique for thinking about what drugs seem to be a hit, right? That hit meaning they return us to the healthy state. We could make predictions about what the outcomes of randomized clinical trials would be for these different drugs. And so far, of the 10 drugs that have gone into randomized clinical trials and have read out, we have predicted 9 of those correctly. And the 10th one, we think we know why, that the mechanism is just something we wouldn't actually pick up in our assay. And we don't expect that in this cellular functional assay, that we would pick up everything, but it's really gratifying to see that, like, we made these predictions really early on. This was all done in a matter of 4 weeks, produce the data, do the analysis with these kinds of models, and make these predictions. Something that we still feel very proud of, having so accurately predicted these outcomes for such an important treatment, especially at that time. There's other ways that we think about using these kinds of representations as well. So you take these features from your, from your classifier, and again, you're going to embed these all into a joint vector space or representation space. And again, you're going to start to do reasoning about their functions depending on how similar different perturbations might be together to each other, or possibly how opposite they might be to each other. So here's a simple example we talk about often, just when we give examples at recursion. mTOR pathway modulates many important aspects of cell growth, metabolism, also involved in tumor formation and growth. And what you see here is you see the representations of two of these knockouts, TSC1 and TSC2, which form a complex in cells, and when activated, actually inhibits mTOR, which is the activator of this pathway, or an important constituent of this pathway. And what we see here when we think about these representations in the space, and we start to think about the similarities between them, we see high similarity between TSC1 and TSC2, which makes a lot of sense because these are two different units of the same complex. And if we knock out one or we knock out the other, we should see something similar functionally, which we get this holistic readout of the cell morphology. And since TSC1 and TSC2 inhibit mTOR, then we would expect that sort of functionally we ought to see some sort of opposite action. And indeed, we see that the vector corresponding to the mTOR knockout lies more or less opposite. I mean, there's, it isn't perfectly pointing in the other direction, but if it's pointing enough, something happened with the mic. I think I kicked this. No, not that one. I feel like I stubbed this podium and it stopped.
We can hear you okay, Burton.
Well, I can do that, but I don't think people online are going to hear.
Oh, we're hearing okay.
What does the chat look like? Yeah, I tried turning it off and on.
Yeah, even online, I hear you fine.
Work out all the kinks. Oh, wait, that's back on. Okay, don't move, Steve. Stay right there.
This isn't a good look.
Okay, so now just let's stand perfectly still. All right. Okay, so in this way, we can just reason about the functions. Let's see, how do I get that to go back up? I don't know. We can reason about the functions of these different perturbations. And if we now take the scale of the kind of data, of this kind of data that we can produce at recursion, then we can actually map out how all these perturbations relate to each other across the entire genome and across large libraries of small molecules. And so you get these, you can produce these kinds of heat maps that are showing us what this readout believes to be the functional similarities and sort of oppositeness of these different genes, the different molecules. And here we're showing an example of the JAK-STAT pathway, and you see the kinds of relationships that you expect to see, those that act similarly to each other, those that act opposite to each other, do actually show that in this readout. And again, what this is that classifier that's been trained on that particular data set. We're taking the features from one of the final layers of that network, and we're aggregating a bunch of those together where we're sort of normalizing those vectors as well according to the controls that we have in these assays. And then we're putting them in this vector space and computing all these cosine similarities between them. And this is one of the primary tools we use for just exploring biology and the effect of small molecules on that biology at recursion. We can do these kinds of maps in various cellular contexts, so different kinds of cells, cell types, like different ways of perturbing that cell state. We can apply different things to say put the cells into an inflamed state, and so forth. And so there's many discoveries to be made in heat maps like this, at least hypotheses to generate. And so this has become one of the primary things that we want to get right at recursion is how do we best represent these images to get correct high dimensional representations that we can then relate in these kinds of heat maps. With our colleagues at Genentech, we actually kind of wrote up our ideas on this and put up this paper that talks about just like how do you think about building these kinds of maps of biology and chemistry, and in particular, like what's the right ways to try to evaluate them. And there's sort of two levels of metrics that I'll be talking about today, but primarily the second. But there's like what we called in the paper univariate metrics. These have to do with like the consistency of a perturbation, how similar different replicates of the same perturbation are in these assays. How do they distinguish themselves from other perturbations? So a whole set of univariate metrics in that way. But importantly, for that particular task of inferring biological relationships, we want to be able to take the databases of known relationships, things like, I didn't kick it this time. Steve, we need your expertise. This feels like a pretty janky
Maybe
room.
as good as Steve.
I don't know. Feeling somewhat silly, right? It's like a rejection sound that happens.
Maybe summing on Steve's energy field just makes it work.
Someone should call the IT aides just so they can come make it more permanent.
Making it even worse.
I don't know. Can people online still hear? Okay, so it looks like people online can still hear. So I'm just going to speak loudly and just trying to talk through this. I don't know. Okay. Okay. Good. So this is the setup. So good. All right. So weekly supervised learning was good for us for a while, right? Like we would keep iterating on ideas and things kept getting better. And it felt great for a while, but then we did hit a wall pretty hard. We couldn't push up on these metrics any further using these weekly supervised techniques. And what was the reason for that? Well, first of all, hitting the wall, a couple things were characteristic of this. As we would increase the size of the data sets, right? 125,000 images, well, that might be large in biology kind of generally, but that's tiny for recursion. We have well over 100 million of these kinds of images. So, but when we would go to larger data sets using weekly supervised learning, okay, we would often see that yes, we were increasing the consistency. Yeah, I'm gonna try. Just teasing me now. We would be able to increase the sensitivity and the consistency of these representations, but we were doing that for both expressed genes and non-expressed genes, which is good and bad and kind of a hard thing to reason about at times. But importantly, we could not improve on this ability to sort of recover known biological relationships. And that often they would go down as we would increase the size of the data sets. And the reasons for that, we think, are that perturbations, so perturbations themselves, so genetic knockdowns and knockouts and these small molecules and other ways that we were perturbing them, they are very noisy labels to be using for classification. Many different perturbations actually have similar effects, right? They're affecting some part of the function of the cell similarly, and many of them have no effects at all. So already this was a real problem in terms of like of just confusing the classifiers as they're trying to learn this. Also, this is also another kind of noisy label problem, but there's a lot of within class heterogeneity that also is difficult for the models to learn or to not learn, as would be the case. And actually some of the work that came out of, some of the things that we realized kind of coming out of this work, we've recently published in this Nature Genetics article that describe that actually, you know, CRISPR-Cas9 systems have a tendency to truncate whole chromosome arms at times. And so you know, as you're perturbing these cells with CRISPR reagents, you know, sometimes with those cells in the population are going to look very different from the single knockout, because you're actually losing a bunch of genes rather than just the one that you tried to knock out. And there's other, you know, other related issues. Also, this end-to-end batch correction like seems like the right thing to do. Like it feels right. But the trouble is that as you're learning to like get rid of some of the signal, the correlated signal from the batch, if the model learns to do that, it doesn't know when it shouldn't do that, or it's hard to get it to know when it shouldn't do that, because it's not part of the batch, but it's actually part of the signal of a perturbation. And I often like to refer to this as one perturbation's trash as another perturbation's treasure. You want to do batch correction, but deal with the batch level correlations sort of at a different level depending on how much batch correction one actually needs. So with these things in mind, we started on a new research program to get ourselves away from weakly supervised learning, and you know, to move on to something else that would allow us to kind of move on those metrics. So what were the desiderata of this research program? Well, we really wanted to test the scaling hypothesis. We have the data. You know, there was plenty of evidence that this was working in other vision and obviously language contexts. We really wanted to get away from weak labels. We wanted to learn all the factors generating our images, and deal with batch correction later. And we wanted to continue learning on large crops rather than single cell crops, mostly for efficiency reasons, because we generate so much data at recursion. So, oh yeah, the scaling hypothesis, I mean, sure, most everybody's heard of this, but just very simply, more compute, more data, more parameters in your model. If that's all kind of done right in some kind of general training task, you're going to get improved downstream performance. And this is sometimes referred to as a bitter lesson, something that Richard Sutton coined. So we put together a team. Some of them are in the audience today, and we went after this idea. So for more compute, luckily for us at Recursion, there's been a real investment in our compute infrastructure. We recently announced an addition to the BioHive 1 compute cluster called BioHive 2. This is a large NVIDIA DGX H100 cluster. It just was recently benchmarked and is number 35 on the top 500 most powerful compute clusters in the world. So you know, it's not huge compared to Meta or something like that, but it's huge for biopharma, and is certainly one of the largest, if not the largest, compute clusters wholly owned by a biopharma company. More data. So, you know, I've been talking about RxRx1 and the work we had done kind of starting from that data set. You know, that was a pretty simple data set, just cell paint images, just siRNA perturbations, only 125,000 images and roughly 1,100 perturbations represented. We started kind of playing around with that in the early days. We created a kind of a similar set that had 2 million images in it, so, or nearly 2 million images, 1.6 images. We then put together a CRISPR data set that we made public called RxRx3. This also had small molecules in it, 2.2 images. This is a great data set that we did a lot of exploration on, and we actually made public. We haven't unblinded all of the labels there, but it is public and can be used for different kinds of learning techniques. And then, of course, we have internal data sets. And so there's a 52 million data set that consists of both cell paint and bright field, as we're doing more and more bright field images. So jointly learning those, and far more perturbation types, both knockdown, knockout, overexpression, small molecules, soluble factors. So nearly 52 million images and well over 2 million perturbations represented there. And of course, our largest that we trained on is this 93 million data set, which, you know, kind of all the same characteristics, but just much, much larger, and nearly 4 million perturbations. So none of this stuff would have worked for weakly supervised learning, again, because most of those perturbations as labels are just very, very, very noisy. But we needed, so this is our more data, and we need more parameters. In fact, we just needed to go to an entirely different task itself. And so we tried a number of different sort of self-supervised learning tasks. And the one that we landed on that was working really well, especially when we got to much larger data sets, and this was the key for making these masked autoencoders work. We landed on using masked autoencoders with a vision transformer backbone. And, you know, the task there is fairly simple. Take your input image, break it up into certain number of patches, either 8x8 or 16x16, mask a certain set of those randomly. We would typically use 75%, we would mask 75% of those patches. And then from the remaining 25%, reconstruct those masked patches. And then once we're done with this, we can aggregate the tokens from some layer in the VIT encoder and use that as a representation for the image to do all of that kind of geometric reasoning. Again, there's no labels here. This is all self-supervised. And because we're using transformers and this task is fairly simple, it's pretty easy to scale up. So we could scale up the size of the model, the size of the data sets pretty easily. And of course, we had to compute to scale the compute as well. We found it necessary to not just do the plain vanilla masked autoencoder training. In particular, we added this Fourier domain reconstruction loss that not only stabilized the training, but it also helped. It was critical in learning the high frequency features of our images. Yeah, absolutely, this was critical, especially as the models are getting larger and we're moving to larger and larger data sets. Okay, so what do reconstructions look like here? So here's a bunch of examples from our best model, which we call Phenom-1. And you're seeing kind of triplets of columns here. On the left is on the right is the original image. On the left is the image masked. And in the middle is the reconstructed image. And I realize that like this isn't the greatest resolution for everybody in this large audience, but the fidelity of the reconstructions are really incredible here. The models really learn to do this super well. But that's not the task that we care about. The task that we care about is being able to do better on these kinds of relationships. So one way that we think about measuring this is we look at the we pick some sort of threshold of the top. So we compute all the cosine similarities between all of the perturbations in one of these large scale data sets like ARCS, ARCS3 across the whole genome. And we pick some sort of threshold like the top 1% and the bottom 1%, or the top 5% and the bottom 5%. And we ask, what fraction of the known relationships from databases like Quorum, HUMAP, Reactome, StringDB, so forth, what fraction of those are actually found in that top and bottom, those top and bottom percentiles? And what you're seeing here is a lot of plots across these different databases. The x-axis here are just like different choices of the threshold. So they go from like the strictest on the left to so like 1% on either side to 10% on either side. So that's why the curves are going up. This isn't scaling yet, but what you are seeing is relationships between the light blue and the dark blue, which is the weakly supervised ability to recall, and then the others being different forms of the masked autoencoders, different backbones, and then different different model sizes, and then different data set sizes. And as we made all of those larger, you get to the dark red, which is on the top, which is recalling the most. And this is potentially easiest to see in a different format. One here is just to show this trend across a lot of different models. So the 4 databases that we were just looking at, you can see that as we use masked autoencoders with transformer backbones and moving to larger and larger architectures, and then the plus there being adding the Fourier loss, we get better and better and better reconstructions. And those better and better reconstructions lead to better and better recall of known biological relationships in these databases. This last line is what we call phenom 1, by the way. Another way to see this is we take one of those databases and we plot on the x-axis the log of the total number of flops used during training. So that's a combination, again, of model size, data set size, and just number of iterations during training. And then recall again at the top and bottom 5%. And you can see in the bottom we have the smaller models trained on smaller data sets, and giving us somewhere to like 40 to 43% recall, almost 44. But then as we move to the bigger ones, we're getting up above 48% recall. And that like that 0.05 percent, or that 0.05, that difference may not seem like a lot, but when we look at databases filled with hundreds of thousands of relationships, this corresponds to thousands and thousands of relationships that are now amongst the most significant. And we're, as a drug discovery company, we're of course not simply interested in known biology. We actually want to explore novel biology, but being able to use this as an evaluation criteria helps us to believe that that any novel biology that's there, there's a higher chance of that being actual biology. So this ability to finally crack the ceiling and move well beyond this with recall was a really exciting development. And we continue to do work on this today, pushing out the number of flops and the recall as we go. So this is that scaling hypothesis being tested and looking like it proves true here. That's not the only thing, the only way that we evaluate the biological content of these embeddings on. We do other things. This is a nice study that's like trying to predict the venerable cell profiler features that one would compute from these images. And you see on these are scatter plots. X-axis is one of these weakly supervised models. The y-axis is this phenom 1 model. And you can see that in almost all cases that these are R-squareds for the regression, and that they're much higher for the phenom 1, the masked autoencoder approach. So just improvements everywhere you look. This has been, the architectures here have been extended to be able to allow at inference time, be able to use channel configurations for similar images, cell paint and bright field of any kind, and be able to get a well-defined and vector with good biological content. These are what we call channel agnostic vision transformers, VITs. And they perform super well. They increase the compute because you basically have an encoder for every channel, but they're super powerful, and they're also the kinds of things that we're scaling up. And in fact, we actually made available on NVIDIA's BioNeMo platform something we call Phenom Beta, so a much smaller version of the Phenom 1 model, but still very performant. It's available on NVIDIA's BioNeMo. You can play with it. It does come under a non-commercial license, but can definitely power a lot of research nonetheless. Okay, so the conclusions there are that these days we're now working a lot with masked autoencoders. They are the kinds of models that we're working on expanding as we try to continue to test the scaling hypothesis. They are our state of the art today and how we're approaching this problem of representing the biological content of these types of images, these cellular microscopy images, so doing image-based profiling. I was asked, and I realize I'm running out of time, I was asked, though, to speak about LOW, and I'll just sort of really quickly say that while I'm a longtime member of the Recursion team, I have also, with the acquisition of Valence Discovery and the formation of Valence Labs or within Recursion, I am now the scientific director of that group. And I'll just briefly say that we have a mission to industrialize scientific discovery to radically improve lives. We believe that at some point in the future, highly autonomous systems will be capable of noble-worthy insights that will usher in a new era of scientific discovery. Let's see what happened. Oh, sort of skip through. We sort of have 3 pillars of how we're going after this in terms of research programs. Building world models of biology and chemistry, so foundation models, but beyond that, they need to have some notion of causality, some way of being able to work out counterfactual reasoning and such in these models. To have inference engines that can do that kind of that can draw those kinds of inferences from such world models, that can also produce them through active learning, and then of course systems to sort of orchestrate all of this and reason over all the things happening within the system. So we have research programs going on in all of these areas. LOW sort of falls under the orchestration and reasoning systems. There's a lot of systems there's a lot of tools one could use to test a hypothesis, for example. And LOW is the yeah, we're not going to listen to music. LOW is the initial sort of step forward along these lines. This is by no means an autonomous system, but it is an LLM that has been instruction fine-tuned to be able to work with the concepts of biology and chemistry and drug discovery and control the different tools available in Recursion's labs, or what we call Recursion's OS. So this is both wet lab capabilities and dry lab through models, predictions, things like that. So one can simply sit at the console of LOW and just start asking questions about diseases, genes, compounds, how they relate to each other. LOW is going and getting the answers from tools like these big maps of biology that I've been discussing today, and then returning them with nice visualizations and so forth. So that was my 1-minute description of LOW. And oops, restart it. So we'll be moving to these highly autonomous systems sometime in the future. I say soon. I mean, this is certainly a 10+ year vision, but systems that can generate hypotheses, design and execute experiments, learn from results, rinse and repeat 24/7. This will be enabling not only drug discovery, but scientific discovery more generally. If any of this was interesting to you, join us at portal.valencelabs.com, where we have a lot of discussions, reading groups organized, and so forth. And of course, there's lots of job openings always at Recursion and Valence Labs, and you can just check out what those openings are on our career pages. I'd encourage you to do that, or come up and talk to me if you have any interest in learning more about that. And thank you for your time. Almost on time.
You're so gracious.
There
with all these
were a couple
questions.
questions.
Yeah, sure.
Let's see, I saw it first, right? Sorry, right behind you. No. Yeah. We need to do this better, yeah.
Thank you very much. Great talk, thank you. You landed on mascot encoder. Have you tried Dino?
Yep.
And why did you choose?
Yeah. So we have tried Dino. In fact, you're going to see a great talk later from our colleagues at Genentech, who have a great paper on using this. I will just, so they have an expert with Dino and like really understands kind of the finicky nature of how to train that. And perhaps that's why they got Dino to work so well. And you'll see that later. In our hands, we really had a hard time making Dino work well. And really, when we got to the size, the data set sizes we have, it was much easier to train masked autoencoders and get them to train, to learn, to converge And ultimately, they gave us much, much better results. So what I am leaving open is the possibility that if someone really spent a ton of time with Dino, they might be able to, to make that work, and maybe that's going to scale as well. I'm not saying that couldn't happen, but in our hands, and it didn't. Mass autoencoders completely outperformed it, in fact, really quickly. Yeah, let's see. I had so the far back on the right here, and then we can come forward. Yeah. So it's you. Yeah. Oh yeah, I should repeat the question.
Thank you so much. Which one about batch effect? You still see it's challenging.
Yeah. Yeah. So the question is about batch effect. Is it still challenging? Actually, in kind of rushing a little bit at the end here, I kind of failed to mention that there's when preparing the embeddings to be for those kind of heat maps, there's still batch correction that goes on. We have tried so many different things, so many different things. But I will tell you that the thing that works most generally and works the best, and it's incredible that it still does, is TVN, total variation normalization. We can thank Mike Ando and colleagues for doing that years and years and years ago. It still works super well, and mostly because it's based on negative controls, and it's a decent way of just being able to scale everything in the right way, given whatever controls you have. And one of the things that Recursion has done really, really well over the years is have a really good control strategy. We use a lot of the wells of every experimental plate that we have dedicated to controls, and the same kinds of controls kind of for all many different kinds of these assays.
Did you try to generalize the previous model that challenge? See how it generalize we have the challenge. Challenge agnostic that as you mentioned, competition is expensive. And what happens?
Yeah, I mean, we did certain things like, take jump CP data and just like repeat one of the channels or something like that. I don't have anything conclusive to say there. I mean, you can do it. I think these approaches are they're much better. They're much more performant, and they're just much more flexible. So it's a good kind of thing to put on like BioNeMo, for instance, for because who knows what your channel configuration will be when you bring it, and we can't guarantee, but we expect that to be a good representation. Yeah. Okay. There was one.
Hi. So fantastic talk. Pretty impressive what you did about getting the data. So I see that you're increasing the end of your samples, but I'm wondering if you also think about increasing the depth of the data. So if I understand it correctly, of fluorescence assays currently is like 10-plex or 100-plex, but it would be for 1% maybe helps of the proteomes. So I wonder about your perspective on that.
Yeah, we definitely do. So what's nice is that we're always generating more data at Recursion. Hey, that seems to be working. We're always generating more data at Recursion, and we can add that to, if it's of high quality, we can add it to the data sets. We do find that for example, for cell types that are very, very different from what was in the training sets, that we can get much better performance if we take a small sample of that data and we attempt to fine tune it, right? They will do much better in that new cell context. So it certainly hasn't, unfortunately, learned every single thing it can possibly learn from the training sets. We've seen this for example, in our work with Genentech. So definitely depth or variety is definitely beneficial. So it's not just always adding one more image, but adding something that's like different enough is of more value than just adding more of the same. But more of the same also can be very helpful because actually there's a lot of factors going on, and we don't have control over all of them. And the more of like the distribution that you see, the more that you actually learn that faithfully. So I don't know if that answers your question, but if there's more, you can ask me afterwards. I think there's one more question right here.
Known biology, especially the class 1, class 2 chemical is very well known by anyone who looks into biology. Say yes. my question is, there is known biology, unknown, and then there is the surprising biology,
Right.
which is what we do either in drug discovery or when we encounter drug resistance after a certain time, is how do we deal with surprising biology? So I want to know what kind of methods or what do you do when you encounter this surprising
Right.
biology?
Right. So of course, for us, this is the most important biology that we can be exploring, right? What looks novel to us in our assays, because we think of this as a massive sort of advantage that we have, just being able to investigate the whole genome and the effects of small molecules in this way. So we do have, so this is only one of the many data layers that we have at Recursion in the Recursion OS. There are transcriptomic layers, there are proteomic layers, there are other layers of data that one can look at, and we can start to test hypotheses generated in this way in other ways and look for you know, any kind of signal that like is corroborating this hypothesis. Sometimes though, you know, one doesn't, there can still be a reason to believe that the biology is real and novel, even if it isn't like super well recapitulated in like another one of these layers, because you know there's a lot of different things happening in these biological systems, and so you know sometimes I don't know, it's hard to go through like what's all the decision-making process, but there are a number of ways that we think about this. We actually try to automate as much of it as possible through the use of LLMs and so forth. But ultimately, you know, we are looking for corroborating evidence for these kinds of things. And of course, getting these in vitro as early as possible for a good hypothesis is one of the critical steps for us so that we can have that kind of early evidence as well. So I don't have one like definitive answer for you, but you know, we are building an industrialized process to go quickly from these kinds of insights into like in vitro readouts. Yeah. I don't know, I'm really out over time. Okay.
Just wondering for the reconstruction, loss and accuracy, to look at the kind of the subcategory of the cell lines you have, what would you like to recount? For example, if you have like the cancerous cell lines, you're going to have a lot of abnormal morphology, and it's going to be more challenging for the
Right.
to reconstruct those abnormal cell lines. Yeah, we're just wondering if you look at like subcategory of the model, and just related another question. Did you look at where your model is failing after when we have really high recall, but then for that, like
yeah, we definitely do. We definitely do these things. You know, as we move into new disease areas and new cell types, like really quickly we see that the model isn't performing like we would hope it would, and it usually has to do with the fact that it's not, you know, explored all the morphology yet, and it's in these huge training sets. So that's an interesting observation that, you know, even in 100 million images, like there's still some morphology to learn and properly organize in the space. We're learning too how to best do this and how to best make use of all of the data we have to be able to generalize out of distribution as best we can. There's lots of research going on in those directions, but you know, I'm not going to say we have it figured out, but it's an important problem that we're definitely working on. That was one of your questions. The other one was, I forgot now.
I was just wondering if you look at some of the failure of the reconstruction.
Oh yeah, failure of the reconstruction. Yeah, I mean, we do. Yeah, that's a tricky task. Just because it's really difficult to look at a cell and go, oh, I see what's wrong with it. You know, like this is one of the reasons why we're even on this program at all, right? Is that no one really believed, you know, 7, 8, 10 years ago that like you could get so much information out of just the morphology. Mostly because like when we look at these images, we're like, of course I'm preaching to the choir here. Everybody at CVMI believes in that strongly, right? But yeah, it's just the case. Like even for people who sit and look at a lot of images, it's really hard, you know, hard to say. So like to be able to kind of reason about it in that way is hard. But yeah, like there are like again, just repeating myself, I apologize, but there are definitely cell types that we don't do as well on, and the solution at this point is more data. And fortunately at Recursion, that is one of the things we're really good at. We can produce a lot of data really fast. Okay.
I was just curious, I mean, you guys have relied a lot on cell paint, and
Yeah.
And so, what's your view on that? Do you feel like you've captured? I mean, to what extent have you captured the universe of labels? Do you ever find things you can't resolve, and then you choose a different set of labels? Now you can resolve them.
There's definitely.
Yeah. How do you measure like what where to go and what to do?
Yeah, we definitely do not expect to capture all relevant biology in cell paint. And in fact, these days we do a lot more in bright field. There's some similarities there. They're not perfect overlap of each other. But there's enough signal there in our hands to really continue to drive at a very low cost, and also to keep the cells alive so we can do other things like multiple time point and then do another assay afterwards, like a transcriptomic assay on the same sample, same biological sample. There definitely are areas that we're not going to capture. We kind of know some of that. I wouldn't say we know. We don't have a very exhaustive view of that. But it does dictate the kinds of like disease programs we can go after. But our objective was never to go after necessarily like every single possible disease. Although one day, I should say that cautiously. Of course, like in our heart of hearts, we want a treatment for every single disease. But like with this platform, the idea is to go after those things that we can see well and that give us an advantage in terms of like how to develop the drug. So yeah. Okay, thank you.
Always.
Thank you.
And you will be.
Yes, absolutely.
Burton, can you hear me?
Professor Birdie, are you online? Are you ready?
I am online and I am ready.
Yes, he is. All right, he is perfect. He's sharing his screen. Let me pop the questions. No. How do I do that? Let me first close this. Does this work? And let me.
Hi, Stefan.
Yeah, let me share the.
Hey, Demetrius. Good to hear your voice.
So people can see the. Can people see the room on Zoom?
I can see the room now.
Oh, he's muted.
Are you muted, Professor Birdie? We can't hear you. Can you hear me? Can people hear us?
I can hear you, and I thought I was online.
Okay. You can hear, right?
I can hear you. Can you hear me?
No, we cannot hear.
Can somebody guide me?
Let's see. Oh, is it because of this? Professor Birdie, can you try to speak again?
I'm speaking now.
Oh, good. Well, now we can hear you. Perfect.
Very good. Thank you. Thank you
Let
very much.
Let me kill this for now. And let's get started.
Yes.
Okay, we can just use the mic again.
Can you use the mic?
Yeah.
Okay. So, hi, Stefan. So we're very happy to, I'll announce now your talk. So we're very happy to have Professor Stephen Burley from Rutgers University, who's going to talk to us about protein data bank, from 2 epidemics to the global pandemics to mRNA vaccines and Paxlovid. So I'm very honored to be a colleague with Professor Burley, who is an expert in data science and bioinformatics, structural biology, and structure-guided drug discovery for oncology. He's director of the RCSB Protein Data Bank at Rutgers, and he serves as university professor and Henry Rutgers Chair and founder director of the Institute for Quantitative Biomedicine and Cancer Pharmacology, and research program leader within the Rutgers Cancer Institute of New Jersey. He has had very distinguished positions previously. For example, he was a distinguished Lilly research scholar from Eli Lilly and Co. So Chief Scientific Officer and Senior Vice President for Research at SGX Pharmaceuticals, and the Richard M. and Isabel P. Furlow Professor at Rockefeller University. And he has degrees. He's one of these unusual cases where they have multiple degrees, important degrees, like a medical degree from Harvard Medical School, and a PhD from Oxford University, and a BSc in physics as well, and Doctor of Science at Western University. And Burley has published extensively in data science, bioinformatics, AI, and machine learning, structural biology, and clinical oncology. And I'm personally very happy to collaborate with him. So, Professor Burley, you can now start.
Thank you very much. I'm very grateful for your introduction, and more important, for the honor of presenting at today's IEEE CVF Computer Vision and Pattern Recognition Conference. Let me tell you that my plan today is to explain the critical role that structural biologists and the Protein Data Bank, the PDB, together played in fighting the COVID-19 pandemic. The punchline of my talk was put succinctly by Dr. Anthony Fauci, former head of the National Institute of Allergy and Infectious Disease. In the New York Times Magazine in 2023, he cited the importance of 3D biostructure information and said, "Show me a person who's vaccinated, got infected, took Paxlovid, and died. I can't find anybody." I'll now explain why Fauci made these statements and put the global efforts of structural biologists and the PDB into context. My talk today will cover the history of the Protein Data Bank and the Worldwide Protein Data Bank Partnership, the RCSB Protein Data Bank, which serves as the US data center for the Worldwide Protein Data Bank, or WWPDB, rcsb.org, our one-stop shop for public 3D biostructure data. And then I'll spend most of my time talking about how structural biologists and the PDB played decisive roles in the fight against SARS-CoV-2 and taming of the COVID-19 pandemic. I will close with a sobering postscript. The PDB, the Protein Data Bank, was established as the first open access digital data resource in all of biology. It was founded in 1971 with just 7 X-ray crystal structures of proteins and has been continuously funded by the United States government ever since. 4 of the early structures motivating protein crystallographers to establish the PDB are illustrated in the right panel. In biology, function follows form. This variation on the famous phrase coined by the Boston-born architect Louis B. Sullivan explains succinctly that the function of a biological macromolecule is determined by its form, its shape, its 3D atomic level structure. Since its inception, the PDB archive has grown more than 30,000-fold to become the singular global resource for experimentally determined atomic level 3D biostructure information. At present, the PDB houses more than 220,000 structures of proteins, nucleic acids, viruses, and macromolecular machines. Recognizing the global importance of the PDB, it has been managed jointly since 2003 by the Worldwide Protein Data Bank Partnership, including data centers in the US, Europe, Japan, and the People's Republic of China, and 2 specialist data repositories for 3D electron microscopy and nuclear magnetic resonance spectroscopy. Reflecting our long-standing commitment to the FAIR, findability, accessibility, interoperability, reusability, and FACT, fairness, accuracy, confidentiality, transparency principles that are emblematic of responsible data stewardship in the modern era, PDB data are distributed under the most permissive Creative Commons license. CC0 licensing means that the information can be downloaded anytime by anyone, anywhere, at no charge, and with no limitations on data usage. Previously accredited by Core Trust Seal, the Protein Data Bank was recognized in 2022 by the Global Biodata Coalition as a global core biodata resource, and I quote, of fundamental importance to the wider biological and life sciences community and the long-term preservation of biological data. The RCSB Protein Data Bank, which I lead, serves as the US WWPDB Data Center. It is supported jointly by the National Science Foundation, the National Institutes of Health, and the US Department of Energy. The RCSB Protein Data Bank is headquartered at Rutgers University in New Jersey, with additional performance sites at UC San Diego and UC San Francisco. We operate 5 interlocking services that together convert global data into global knowledge for the public good. The RCSB PDB supports research and education across fundamental biology, biomedicine, and the energy sciences, serving many millions of PDB data depositors and data consumers in nearly every sovereign country and territory recognized by the United Nations. Every subdiscipline of biology is supported by open access to PDB data, literally running the gamut from agriculture to zoology. Among non-biological researchers and educators, PDB data are used by chemists, computer scientists, engineers, linguists, mathematicians, physicists, and statisticians. Finally, PDB data are used extensively by all major biopharmaceutical companies and many smaller biotechnology companies. Once 3D biostructure data are archived in the PDB, our rcsb.org research-focused web portal serves as a one-stop shop for open access to more than 220,000 experimentally determined PDB structures, alongside more than 1 million computed structure models coming from the AlphaFold database and the Model Archive. We deliver public domain information to many millions of basic and applied researchers, educators, and students around the world at no cost and with no limitations on data usage. For the avoidance of doubt, the provenance and reliability of both types of 3D biostructure data, experimentally determined and computationally predicted, are clearly identified on our website. This slide shows the rcsb.org homepage. Highlighted within the red ellipse is the toggle that allows you to opt in to access more than 1 million computed structure models, or CSMs. Moving the toggle from left to right enables combined PDB and CSM searches from the top bar present on every web page, and when using our powerful advanced search option, which supports full Boolean logic. PDB data are essential for responding to emerging viruses. Since the early 2000s, the world has faced down 3 major outbreaks of coronaviral infections that jumped the species barrier to humans. First detected in 2002, the SARS coronavirus infected about 8,500 individuals worldwide, killing just over 800 people between 2002 and 2004. The second coronavirus epidemic, caused by the Middle East Respiratory Syndrome, or MERS coronavirus, struck in 2012. To date, it has killed more than 900 individuals and remains a public health threat in the Middle East and parts of Asia, where it is endemic in camel populations. Despite these 2 warning signs, very few countries prepared for the possibility of a much more serious coronavirus epidemic. Fortunately for the global community, structural biologists and the PDB laid the groundwork for a successful response to the inevitable 3rd coronavirus wave. Effective mRNA vaccines were designed and antiviral agents were discovered and developed with the benefit of open access to PDB structures of SARS-CoV, MERS-CoV, and SARS-CoV-2 proteins. SARS-CoV-2, the unseen enemy, is shown on the right within a respiratory droplet in a haunting painting by our artist-in-residence, David Goodsell, who has authored the RCSB PDB Molecule of the Month series for more than 20 years. The organization of the SARS-CoV-2 genome is depicted schematically in this slide. All coronavirus genomes are very long, single-stranded, positive sense, 5' capped, 3' polyadenylated messenger RNAs ready for translation by host cell ribosomes. Most of the non-structural proteins are expressed within a pair of related viral polyproteins. Individual non-structural proteins are excised from the polyproteins by 2 SARS-CoV-2 proteases. Both are non-structural proteins themselves and are part of the polyproteins. The papain-like proteinase cuts at 3 sites denoted with dark blue down arrows. The even more important main protease, or MPRO, cuts at 10 sites denoted with light blue inverted triangles. Quite rightly, the first PDB structure of a SARS-CoV protein was that of non-structural protein 5, the main protease, or MPRO, PDB ID 1 Quebec 2 Whiskey using the NATO alphabet. I say rightly because without the main protease, the viral polyprotein cannot be cleaved to release most of the non-structural proteins, stopping an infection in its tracks. This structure was determined in 2003 by my company, SGX Pharmaceuticals Incorporated, where I led research and development as the chief scientific officer. It has the distinction of being the only PDB structure with its primary publication appearing in the New York Times. Immediately upon deposition to the protein data bank, my company released the structure to the public so that work on viral countermeasures could begin without delay. To date, more than 90 apo and co-crystal structures of this SARS-CoV protease have been contributed to the archive. The second Achilles heel of coronaviruses is the papain-like proteinase. Without the action of PL-Pro, the viral polyprotein cannot be cleaved to release 3 of the non-structural proteins, again, stopping an infection in its tracks. The first SARS-CoV PL-Pro structure was contributed to the PDB and released in 2006, 2 years after the epidemic ended. To date, more than 10 apo and co-crystal structures of this second SARS-CoV protease have been contributed to the archive. The first PDB structure of the entire extracellular portion of the homotrimeric SARS-CoV spike protein, ID 5 Whiskey Romeo Golf, was determined using single particle cryo-electron microscopy and only released to the public in 2017 in the wake of the cryo-EM resolution revolution. To date, more than 70 SARS-CoV spike protein-related structures have been contributed to the archive, including pre- and post-fusion conformations and various complexes with its cellular receptor angiotensin-converting enzyme 2 or ACE2 and various antibody FAB fragments. The first PDB structure of a MERS-CoV protein, released in 2013, was that of the ACE2 binding domain of the spike protein, shown here in the right panel in a ribbon representation, ID 4 Kilo Quebec Zulu. To date, for MERS-CoV, nearly 70 spike protein-related structures have been contributed to the archive, alongside nearly 40 MPRO structures and more than 10 PL-Pro structures. I will return to the SARS-CoV and MERS-CoV spike proteins a little later in my talk because they played critical roles in guiding SARS-CoV-2 vaccine design. This is an example of what is called prototype pathogen preparedness. Since the very first SARS-CoV-2 protein structures were deposited to the PDB in early 2020, my organization, the RCSB Protein Data Bank, has been hard at work responding to the ongoing pandemic. Together with our partners in the Worldwide Protein Data Bank, we prioritize complete deposition, rigorous validation, and expert biocuration of COVID-19 related protein structures, and strongly encouraged immediate pre-publication release of these data. I'm pleased to report that most structural biologists did in fact release their COVID-19 PDB structures to the public before they were reported in scientific journal articles. We created a COVID-19 coronavirus resources page on our rcsb.org web portal, which is updated weekly with each release cycle, adding approximately 300 new structures of any stripe to the archive every Wednesday. We also developed education and outreach resources, such as this animated hand-washing video viewed more than 500,000 times, and a series of RCSB PDB Molecule of the Month articles, authored by David Goodsell, featuring SARS-CoV-2 proteases, the RNA-dependent RNA polymerase, and spike proteins. The right panel of this slide summarizes our now nearly complete atomic-level 3D knowledge of SARS-CoV-2 protein structures, embodied in more than 4,200 PDB depositions released since early 2020. That number represents approximately 2% of the entire PDB archive devoted to a single pathogen. The left panel shows an artist's rendition of the lipid bilayer of SARS-CoV-2 fusing with the plasma membrane of a soon-to-be infected cell in another haunting painting from David Goodsell. This viral entry process is blocked by the immune response to mRNA vaccines against SARS-CoV-2 that I'll come to next. In 2012, I concluded a 10-year stint in industry and attempted retirement. I had no plan and, not surprisingly, failed miserably. Fortunately for me, Rutgers was there to pick up the pieces. In January 2013, I joined the university and the RCSB Protein Data Bank and became a colleague of Dimitris Metaxas. In addition to succeeding Helen Berman as director, I decided to reinvent myself as a data scientist and focus my research on the information stored in the PDB. Over the past 4 years, members of the RCSB PDB team and I have been analyzing many of the more than 4,200 SARS-CoV-2 related structures represented in the archive. First, we examined how the SARS-CoV-2 proteome evolved during the first 6 months of the pandemic. During the summer of 2020, 11 undergraduates and 1 very talented high school student, now studying engineering at Columbia University, worked with 6 faculty mentors to analyze how 29 viral proteins changed in amino acid sequence and 3D structure using genome sequence data downloaded from the GISAID web resource. For each of 29 viral proteins, we analyzed nearly 50,000 translated viral genome sequences that were available to us in early June of that year. The key findings reported in our Lubin et al. 2020 proteins paper revealed that the 29 viral proteins underwent very few amino acid changes during the first 6 months of the pandemic, reflecting the fact that all known coronavirus genomes encode a proofreading exonuclease known as non-structural protein 14. Coronaviruses depend critically on this exonuclease because they have the longest genomes of any known RNA virus and are most prone to the introduction of mutations. In nearly 90% of the cases, we found that the sequences of the 29 viral proteins did not change during the first 6 months of the pandemic. When changes did occur, they were largely restricted to 1 amino acid at a time. We found that most of these point substitutions occurred on protein surfaces where they were predicted using Rosetta to have little or no effect on structural stability. But for spike proteins, these changes allow the virus to evade the human immune system. Importantly, we also found that the hydrophobic cores of the viral proteins and key enzyme active sites were highly conserved throughout the first 6 months of the pandemic. It was clear from these data that essential coronavirus enzymes, such as the proteases I described earlier, represent attractive targets for drug discovery. Now, you may be asking yourselves why it was only SARS-CoV-2 that went on to cause a global pandemic. The reasons are twofold. First, SARS-CoV-2 is capable of pre-symptomatic spread. Victims of SARS-CoV and MERS-CoV could only pass on the virus to others when they became acutely ill and required hospitalization, making it much easier to contain the outbreaks. The most successful viruses are those that can be transmitted to others during the incubation period, when the asymptomatic infected individual is out and about and even flying in commercial airliners to distant parts of the globe. This explains the very rapid international spread of SARS-CoV-2 from Wuhan in China, where it originated. Second, both SARS-CoV and MERS-CoV were deadlier than SARS-CoV-2. Viruses tend to be much more successful if they do not kill their infected hosts, thereby maximizing the likelihood of transmission throughout the population. The first PDB structure of a SARS-CoV-2 spike protein, ID 6 Victor Sierra Bravo, was released to the public in February 2020. Today, there are more than 1,700 SARS-CoV-2 spike protein-related structures in the archive. Like earlier coronavirus spike protein structures in PDB, they provide important insights into receptor binding, fusion of the viral lipid bilayer with the plasma membrane, and vaccine and antibody design. The very first mRNA vaccine designs were actually generated in January 2020 based on PDB structures of the SARS-CoV and MERS-CoV spike proteins, which are very similar in amino acid sequence and 3D structure to one another and to their SARS-CoV-2 counterpart. When designing Moderna's inaugural mRNA-1273 vaccine encoding the SARS-CoV-2 spike protein, researchers at NIAID and the company relied on PDB structures of double proline mutant forms of the SARS-CoV and MERS-CoV spike proteins. This engineered double proline modification stabilizes the spike protein in a highly immunogenic pre-fusion conformation. I believe that the vaccine designers at BioNTech used the same information because their inaugural mRNA vaccine design also encoded a SARS-CoV-2 spike protein bearing the identical double proline modification. The rest of the story is well known to you. Two essentially identical, highly effective mRNA vaccines against COVID-19 were brought to the market in record time by Operation Warp Speed. To date, these vaccines and their successors have been administered to more than 5.5 billion individuals worldwide with impressive results. The vaccines designed with the benefit of open access to PDB structures, accredited with saving tens of millions of lives globally, and preventing severe forms of infection in hundreds of millions, if not more than 1 billion individuals worldwide. You've got to love the protein data bank. I'll now turn from the spike protein and vaccine design to discovery of small molecule anti-SARS-CoV-2 drugs. Early in the global pandemic, drug repurposing proved to be a successful strategy for targeting the SARS-CoV-2 RNA-dependent RNA polymerase, shown in the right panel, ID 6 Yankee Yankee Tango. The viral holoenzyme consists of 4 non-structural proteins, including NSP7 in light purple, the larger NSP12, the RNA polymerase subunit itself, in cyan, and 2 copies of NSP8 in darker purple, the molecular chopsticks that hold the yellow-orange RNA duplex, which is the enzyme substrate product combination. Gilead's remdesivir was originally developed for treatment of hepatitis C virus infections. It received emergency use authorization for SARS-CoV-2 from the US FDA in 2020. The right panel shows PDB ID 7 Bravo Victor 2 with remdesivir incorporated into the RNA product of the holoenzyme. This structure explains the mechanism of action of the repurposed drug at the atomic level in 3D. Merck's molnupiravir was originally discovered at Emory University during work targeting Venezuelan equine encephalitis virus. It received emergency use authorization for SARS-CoV-2 from the US FDA in 2021. The right panel shows PDB ID 7 Oscar Zulu uniform with molnupiravir incorporated into the RNA product of the holoenzyme. This structure explains the mechanism of action of the repurposed drug at the atomic level. Moving on, I'll now describe structure-guided drug discovery for SARS-CoV-2 main protease, or MPRO, the Achilles heel of the virus that I introduced earlier. It is the target of Pfizer's highly effective, orally bioavailable small molecule drug known as Paxlovid, a fixed dose combination of nirmatrelvir, the active ingredient, and ritonavir. The PDB currently houses more than 1,400 crystal structures of SARS-CoV-2 MPRO, the first of which was ID 6 Lima Uniform 7, deposited in January 2020 by colleagues based in Shanghai. I should point out that they completed their structure determination in less than 2 weeks following the public release of the viral genome sequence. That's synthesize the gene, express the protein, purify it, crystallize it, do the X-ray crystallography experiment, complete the analysis, and deposit the information to the protein data bank, all within 2 weeks. An incredible accomplishment. The nirmatrelvir origin story actually begins in the early 2000s with discovery and development of Pfizer's PF-00835231, shown here in the right panel inhibiting SARS-CoV MPRO in PDB-ID 6 X-ray Hotel Mike. This would-be Pfizer drug was intended for use as an injectable, in-hospital antiviral agent for acutely ill SARS-CoV infected patients. It was the product of a structure-guided drug discovery campaign initiated in 2003 that was facilitated by open access to the SGX structure of SARS-CoV MPRO PDB-ID 1 Quebec 2 whiskey that I introduced earlier, the structure that my company determined and put into the public domain with the goal of making it possible for somebody to use it to develop viral countermeasures. Regrettably, Pfizer halted their project in the mid-2000s because SARS-CoV had disappeared, and the company correctly judged that there would be no commercial market for such a drug. In early 2020, as the pandemic hit, Pfizer quickly reactivated the SARS-CoV project with the goal of discovering and developing an orally bioavailable antiviral agent for outpatient use, facilitated by open access to PDB-ID 6 Lima Uniform 7, the first SARS-CoV-2 MPRO structure I described moments ago. The result was nirmatrelvir, shown here in the right panel, inhibiting SARS-CoV-2 MPRO, ID 7 Romeo Foxtrot whiskey. You've got to love the PDB. Both nirmatrelvir and PF-00835231, shown on the previous slide, are covalently acting inhibitors of the protease, binding to the invariant active site cysteine residue visible within the red circle. Upon encountering the drug, these enzymes catalyze a single turnover reaction, forming the inactive covalent adduct visible in the right panel, thereby preventing processing of the viral polyprotein and stopping the infection in its tracks. Nirmatrelvir is structurally similar to Pfizer's first-generation SARS-CoV compound, with the chemical differences that allow it to be administered as a pill on an outpatient basis to newly infected individuals. Nirmatrelvir does, however, have one major drawback. It is rapidly metabolized by the cytochrome P450 isozyme CYP3A4. Co-administration with ritonavir, a potent CYP3A4 inhibitor, prolongs the half-life of nirmatrelvir by preventing degradation as it passes through the liver. Paxlovid, a fixed-dose combination of the two drugs, received emergency use authorization from US FDA in December 2021, less than 2 years after public release of the viral genome sequence. The speed with which Pfizer was able to move was unprecedented. It often takes a decade or more to go from identifying a target to approval of a new drug. At the end of my talk, I'm going to come back to the fact that Pfizer reported that nirmatrelvir is also active against the main proteases of both SARS-CoV and MERS-CoV. It's this important finding that will lead me to my sobering postscript. The PDB archives houses approximately 100 apo and co-crystal structures of the human cytochrome P450 isozyme CYP3A4. The right panel shows ID 5 Victor Charles 0, a co-crystal structure of human CYP3A4 inhibited by ritonavir. This drug was originally discovered and developed by Abbott, now AbbVie, as an HIV protease inhibitor. The structure explains in 3D at the atomic level why ritonavir has also proven useful for prolongation of the half-lives of other viral protease inhibitors that are susceptible to CYP3A4 degradation. The fact that nirmatrelvir inhibits the main proteases of SARS-CoV-2, SARS-CoV, and MERS-CoV is not actually surprising. In 2021, 4 summer undergraduates working remotely at Rutgers with me and Professor Sagar Kare in the Institute for Quantitative Biomedicine. Our goal was to understand MPRO, active amino acid sequence, 3D structure, active site, and polyprotein cut site diversity across all known coronaviruses. Because this work has not been published, I'll summarize our results in some detail. All coronavirus main proteases fall into one of 4 genera, as shown in this cladogram, based on clustering of full-length MPRO amino acid sequences. SARS-CoV, MERS-CoV, and SARS-CoV-2 all fall within the beta coronavirus genus, denoted in light blue. Please take a moment to note how close SARS-CoV and SARS-CoV-2 are to one another within the cladogram. The image showed in the right panel explains how the MPRO active site revealed by PDB-ID 6 Lima uniform 7 can be partitioned into 6 pockets or subsites designated S1' to S5. The invariant active site cysteine visible within the red circle is located between S1' and S1. Like full-length MPRO amino acid sequences, all coronavirus main protease active sites fall into one of 4 distinct genera, as shown in this cladogram, based on clustering of the amino acid residues that line the enzyme active site. Again, SARS-CoV, MERS-CoV, and SARS-CoV-2 all fall within the beta coronavirus genus, denoted in light blue. You can see from the detail of this cladogram that the amino acids lining their MPRO active site sequences are more similar to one another than are their full-length amino acid sequences. Conservation of the amino acid residues lining the main protease active sites of all known coronaviruses is nothing short of remarkable. Each column in the right panel shows the frequency of each of the 20 possible amino acids occurring at each residue lining each enzyme active site. Dark blue identifies amino acids that do not occur at that position. There's a lot of dark blue in this plot, reflecting the fact that MPRO active sites are highly conserved across all known coronaviruses. For example, the SARS-CoV and MERS-CoV-2 MPRO active sites are made up of exactly the same amino acids occur in exactly the same positions in 3D. Across all known coronavirus, you can see that 12 of the amino acid residues lining the active site of the enzyme are essentially invariant. At position number 27, for example, leucine occurs more than 99% of the time. At position number 140, phenylalanine occurs 100% of the time. At position number 145, the invariant active site cysteine, shown in previous slides, occurs 100% of the time, and so on. Based on computed structure modeling using all of the known coronavirus MPRO structures housed in the PDB, you can see in the right panel the MPRO polyprotein cut site substrate modeled into PDB-ID 6 Lima uniform 7 consists of 6 amino acid residues designated P1 to P1', occupying sites S1' to S5. Main protease catalyzed cleavage of the polyprotein occurs between P1 and P1', within each of the 10 cut sites distributed along its length. The invariant active site cysteine, number 145, visible within the red circle, is positioned to break the peptide bond between P1 and P1 prime in each of these 10 cut sites distributed along the length of the polyprotein. Main protease polyprotein cut site amino acid sequences are also highly conserved across all known coronaviruses. Each column in the right panel shows the frequency of each of the 20 possible amino acids occurring at P5 through P1 prime across all known coronavirus polyprotein cut sites. Again, dark blue identifies amino acids that do not occur at that position, and there's a lot of dark blue. At P2, leucine and valine together account for more than 94% of all amino acid occurrences across all known coronaviruses. At P1, glutamine occurs more than 99% of the time. At P1 prime, alanine, asparagine, and serine together account for more than 96% of all amino acid occurrences. In effect, there is double evolutionary selection pressure at work on both the 3D structures of coronavirus MPRO active sites and the amino acid sequences of the 10 MPRO cut sites found in each coronavirus polyprotein. During viral evolution, the catalytic activity of the enzyme must be preserved, and each of the 10 polyprotein cut site amino acid sequences cannot change significantly. Without an effective enzyme, the virion will not be able to replicate itself inside an infected cell and spread throughout the host and beyond. Similarly, without all 10 functional cut sites within the polyprotein, the virion will not be able to replicate itself inside an infected cell and spread throughout the host and beyond. We therefore predict that nirmatrelvir will be effective against many of the known coronaviruses, not just SARS-CoV, MERS-CoV, and SARS-CoV-2, as shown by Pfizer. Going beyond their work, we're currently testing these in silico predictions with representative main proteases from a diverse array of coronaviruses from across all 4 genera before proceeding to publication of the results that I've just described. Understanding constraints on the evolution of coronavirus main protease active site structures in 3D and polyprotein cut site amino acid sequences also tells us that the free market for emergency pharmaceuticals failed us. As my late father, an economics professor, told me on more than one occasion, the failure of the free market is defined as an inefficient distribution of goods and services within the economy. He believed that repetition was the key to learning. Such phenomena occur when individual incentives for rational economic behavior do not lead to a rational outcome for society. Simply put, we cannot always rely on capitalism to get the right outcome when it comes to the public good. For better pandemic preparedness benefiting the entire world, Pfizer could have been incentivized with public money in the 2000s to continue working on structure-guided drug discovery of a broad-spectrum coronavirus MPRO inhibitor. With the benefit of hindsight, we now know that investment of a few $100 million in a SARS-CoV main protease inhibitor could have saved millions of lives worldwide before the Moderna and BioNTech mRNA vaccines became broadly available, and also prevented economic losses to the tune of trillions of US dollars. When the MERS-CoV epidemic struck, there could, and I think should have been, a scientifically informed discussion regarding the very real opportunity and desirability of public investment in pharmacologic countermeasures targeting new coronaviruses that would cross the species barrier to humans. Experts knew after MERS-CoV that it was no longer a matter of if, but simply a matter of when the next coronavirus epidemic would strike, with the attendant risk of becoming a global pandemic. Experts also knew that the active sites of SARS-CoV and MERS-CoV were nearly identical in 3D structure, and that their polyprotein cut site amino acid sequences were highly similar. But the free market for emergency pharmaceuticals failed us. Fortunately for the world, structural biologists studied key SARS-CoV and MERS-CoV proteins and deposited their results into the open access protein data bank, which laid the groundwork for engineering of life-saving vaccines and discovery and development of a highly effective antiviral agent against SARS-CoV-2. Pfizer made a sizable investment in structure-guided drug discovery and development of a SARS-CoV Mpro inhibitor, which set the stage for very rapid discovery, development, and emergency use authorization of Paxlovid. Similarly, the NIH, Moderna, and Pfizer invested in developing mRNA vaccine technologies. One of the few silver linings in the COVID-19 global pandemic cloud is the fact that our experience with SARS-CoV-2 has revolutionized how vaccines are designed, manufactured, tested in the clinic, and undergo regulatory approval. Over the next few years, we can expect to see new mRNA vaccines come onto the market, offering fresh hope for millions of patients and their families. As a research oncologist, I'm very hopeful that mRNA vaccines targeting mutated oncoproteins commonly found in patient tumors will provide new weapons in the fight against cancer. I am also hopeful that we will be better prepared for the next global pandemic. To recap, I've explained how open access to research data generated with public and private funds, particularly thousands of atomic level 3D structures of coronavirus proteins archived in the PDB, enabled basic and applied researchers to combat the COVID-19 pandemic and save tens of millions of lives worldwide. Please join me and Dr. Fauci in loving the protein data bank. In closing, I thank you, the audience, for listening and for the honor of giving this talk.
Yeah.
Next, I must thank some 60,000 or so structural biologists worldwide, including some of you in the audience perhaps, who freely contributed your data to the PDB, EMDB, and BMRB, making these three worldwide protein data bank core archives what they are today. I also thank the many millions of basic and applied researchers and their trainees around the world who utilize open access 3D biostructure data from the PDB to advance our knowledge of fundamental biology, biomedicine, and the energy sciences. I am grateful to our NSF, NIH, and DOE federal funders, our host institutions, Rutgers, UCSD, and UCSF, and our WWPDB partners. I acknowledge the large team of dedicated RCSP PDB structural biologists, data scientists, software developers, and IT professionals shown here. Together, we deliver RCSP PDB data and services to many millions of users worldwide at no charge and with no limitations on usage. Thank you and be safe.
Thank you. Thank you very much. In the interest of times, we can have questions at the end of the session after the papers now are going to be presented. And you can also email to Dr. Burley and if you have any other questions. So we'll move on now with the, thank you very much.
Yeah.
Thank you.
Thank you.
Dr. Burley,
Thank you.
yeah. You can introduce the next speaker
Yes.
while they
Where
fix
is-
the audio.
Yes, so I would like to introduce the first speaker, which is, we'll talk about discovering interpretable models of scientific image data with deep learning, and the speaker will be Christopher Solisto from the Alan Turing Institute.
Is the speaker on Zoom or in the room?
Doesn't.
I guess it's
Zoom.
Okay, great. Could you try start sharing your screen while we fix the audio in the room?
Yes.
Thank
Will
you.
do. Thanks.
On the volume.
Just give me a second. I'll rejoin the meeting. I think it's a permissions thing. Go back in a second.
Audio settings.
Switch it on the pod. We have a
Your audio settings.
Yeah.
Oh, here. Which one should we choose?
Headphones.
Yeah.
Headphone.
Yeah. Let's get the podium mic out of there.
There you
Check 1, 2, podium.
go. Okay, you try something.
Yeah, you want to try this just to
Is it
video.
making any noise or?
What video?
Whichever video, just to test this.
Oh, you mean whether they can hear us or
Okay.
whether?
The video to play on this. We need audio from the laptop. Yeah.
Oh, I see. Christopher, can you speak up? We're just
Yes,
testing the audio.
Oh, hi. Can you hear me?
Yes.
Great. Am I good to start?
Yes, please.
Yep.
Okay.
Yeah,
And
please.
it's, you have 10 minutes.
Okay, great. Yeah, let me know if I'm going over. But anyway, I'll begin. So yeah, I'm Christopher. Until recently, a postdoc at the Alan Turing Institute, and my talk will be focused on how we can use deep learning potentially to extract new scientific knowledge from image data, basically. And the idea here is basically that when we
You can turn on your video to make people can see you in the room so that.
Oh, I think it should be on. Can you not see my my video?
We don't see, we see your slides. We don't see yourself.
Oh, that's odd. It should be on. Okay. I don't know what's going on. I can see my video, but maybe because of a permission.
Now we see you.
Oh, great. Okay. Anyway, so I'll just continue. Yeah. So basically, the core idea of my presentation is that when we train deep learning models or any machine learning model to basically internalize or model some kind of biological or scientific phenomenon, the model is essentially learning something about it, whether it's to do with the dynamics of the system or whatever. And the question is, can we design interpretable models that can not only learn about an underlying system, but actually tell us what it is that they've learned about it? And the background to this whole idea is the idea that we don't always have an accuracy complexity trade-off when we design models. In other words, it is possible to design models that can successfully model biological phenomenon or any other phenomena. So in other words, be accurate, but also be interpretable. And this might be by virtue of their simplicity or whatever. And the core idea here is Occam's razor, the fact that we can actually design many models that perform equally well in terms of accuracy, but some will be better than others in terms of simplicity or interpretability or so on. And so the ideal models are models that are not only performant in the sense of having high accuracy, but are also interpretable and hopefully domain appropriate. And I'll explain a bit about what this means in a second. So in order to kind of investigate this broad idea, what I tried to do was design a few models that could do a very simple image classification task. So look at some cells and say whether or not they're an interphase or metaphase. And these are just fluorescence images taken where we basically stain the histones. So basically what you see here is the cell sorry, is the cell nucleus. So interphase images are characterized by cells which are quite widely diffuse and round, and this is because the chromatin is quite diffuse. And metaphase images are classified by the cell profile is generally quite small and a lot more elongated or eccentric. So these descriptions are descriptions that we kind of human scientists use to say what the difference is. But the question is, can we train neural networks that could learn to tell the difference between interphase and metaphase and tell us what the difference is in terms that we can understand? And just to talk a little bit about the domain constraints here, of course, there are some things that we want our models to, some characteristics that we want it to have. So for example, ideally, we'd want it to be invariant to spatial transformation. And this is because the orientation of the cells doesn't actually tell us anything about the biology. It's just to do with, how the microscope is oriented and so on. and we'd ideally want it to be insensitive to background noise, which can also be an artifact of the microscopy process. And hopefully, as an additional constraint, we'd want it to be ignorant of neighboring cells, because for this specific task, we just want it to classify the cell state of the object in the center. So can we get our models to do that is the question. So basically, in order to investigate our hypothesis of can we train kind of accurate but interpretable models, I kind of designed 4 different model schemes, and they go in kind of increasing interpretability from left to right. So on the left here, we have a dense convolutional neural network that takes in the image, and a dense kind of feed forward network, which then gives us the classification. So this is a very kind of traditional CNN design, and it's totally uninterpretable because there's lots of connection weights that do lots of things, and it's difficult to tell exactly what it is they're doing. From model 2 onwards, we use representation learning. So in other words, we try to run a disentangled, interpretable, variational autoencoder model on our images to essentially distill our raw pixels into some kind of interpretable semantic representation. And in scheme 2, we have a dense kind of classification head. Then in scheme 3, we have a sparse classification head. And in scheme 4, we have a symbolic expression. So this is the most interpretable, because it actually gives us a mathematical expression that tells us whether or not the image is an interphase cell or a metaphase cell. So then the question is it necessarily the case that we suffer a big decrease in model accuracy as we make our models more interpretable? So just to talk very briefly about some of the methods that we used. So for disentangled representation learning, we used a total correlation VAE, and I'm happy to give some more details if anyone's interested. For the sparse neural network training, we use a dynamic pruning algorithm called RigL. Again, happy to talk about that. And for symbolic regression, we use a genetic algorithm called PySR, and I can talk a bit more about that as well. So basically, in terms of disentanglement, what we did was we trained a VAE on about 2 million of these cell images to see if we can identify certain latent features that correspond to semantic concepts. And this was quite successful. So in the top left, Z3 there, which is sort of one latent variable, has a very strong correspondence with cell size. So increasing values of Z3 mean that the cell gets bigger and so on. Z17 and Z21 are both characteristic of eccentricity. So there, the greater the absolute value of the variable, the more eccentric the cell is. And Z29 here is also a representation of cell size, but it behaves slightly differently in the sense that it's a lot less sensitive to cell crowding. So there's a bit of a subtle difference between Z3 and Z29, but nevertheless, these are both size variables. Okay, so we've got 2 size variables and 2 eccentricity variables. And just as an interesting aside, I've also noticed that there were some variables for position along the X and Y axes, which I thought was pretty cool. Yeah, so aside from that, the rest of the latent variables are ones that describe the neighborhood and so on. So you might already be able to see where I'm going with this. Basically, we want our models ideally to only look at these 4 latent variables, because these are the ones that, using our prior knowledge, we would say are informative of cell state. So the question is, does our model learn to ignore sort of the rest of these? And this is something that we'll see. So just to talk a bit about some of the other stuff some of the other techniques that we were using. So sparsity training was a very important component of this. And the goal was basically following Occam's razor to train a neural network with to be as simple as possible, or to have as few active connection weights as possible, while still retaining a big deal of model accuracy. So this is kind of an illustration of how the dynamic pruning algorithm works. So here, blue connections are positive weights, red connections are negative weights. So what it basically does is it finds the topology of the neural network that increases performance given a certain sparsity parameter. And this is a parameter that we found just using kind of hyperparameter tuning. And when we do this, we find out that indeed we can train a feed-forward network so the input layer here is basically the latent representation, the VAE latent representation of a single image. And this output neuron here at the bottom gives us a score. And if it's positive, the network is saying that the cell is in metaphase. And if it's negative, it's saying that the cell is in interphase. So basically, this is our classification network. And what we found was, interestingly, we trained several of these, and pretty much every single time it identified 4 input variables that were the relevant ones, and these matched exactly with the variables that characterize the central cell. So this was quite a pleasing outcome of our research. Given time constraints, I won't go over this a big deal, but basically, if you take this network, you can actually break it down to a few smaller subnetworks, and you can actually analyze what these subnetworks are computing. So for example, we can tell that this subnetwork, the output of which is in this intermediate layer here, is basically sensitive to cell size. And this is because it takes the 2 size variables, Z3 and Z29, and in a radial fashion, it basically tells you how big the cell is. And there's another network for eccentricity and so on. So basically, what these subnetworks are doing are distilling or forming concepts that will later be used downstream to make the final classification. And so the idea being that if we can identify these concepts, we can actually map out how the network is responding to things like size and roundness. So for example, here, we can tell that the response of the network is highest when the cell is small. And also there's some kind of dependence on the roundness as well, but it's not as drastic. So in other words, we can actually look at a sparse network and tell how it's forming its decisions. So I'll park that for now and then talk a bit about the symbolic regression. So symbolic regression is another alternative strategy that one can use for kind of creating very simple models using our latent VAE feature space as an input. And basically what we found is that you can actually find some mathematical expressions that take our latent variables and give you a score for each image. And if that score is positive, then the cell is in metaphase, and if the score is negative, then this cell is in interphase. And this is quite interesting because you can actually get some pretty high accuracy expressions that are quite simple to some degree. For example, this expression here was our highest performing one with a nearly 98% test accuracy. And it is quite simple and interpretable in some ways. So you can see that we have our 2 kind of eccentricity terms in here that acts as a weighting term that balances out our 2 size terms, Z3 and Z29. And this basically allows you to find a decision boundary in the feature space on one side of which the cell is in metaphase and on one side of which the cell is in interphase. And this is perfectly interpretable because it is literally symbolic. So I'll just mention that with our symbolic expressions and sparse networks, we can actually get quite high accuracy with only a small amount of the complexity. So comparing our dense classification heads with our symbolic expressions, we can actually retain like pretty much all of the performance with a very small slice of the complexity. And if anyone's interested, I can explain how we calculated complexity. But for now, I'll move on. So really, we can train our models to perform well, but what we really want is for them to somehow inform us about the underlying system. So for example, if we have this expression here, and it says, well, if this symbolic expression is greater than 0, it's an interphase. Or actually, sorry, I've got that the opposite way around. If it's greater than 0, it's a metaphase. If it's less than 0, it's an interphase. what does this actually tell us? Well, it tells us a few things. The first is that this model is arriving at a classification of cell state primarily using the cell size. And this is because essentially what you've got in this expression is you've got two terms that both feature cell size features, so Z29 and Z3, and they're essentially being weighted against each other using this eccentricity term here, Z17 squared plus Z21 squared. But whether or not it's a negative value or a positive value is determined primarily by our size variables. The other thing we know is that metaphase cells tend to be smaller than interphase cells. Why? This is because if you increase Z29 or decrease Z3, which means making the cells smaller, then this increases the score and it pushes us towards the positive region, which is metaphase. Again, I apologize for getting it the opposite way around here. And the other thing it tells us is that metaphase cells tend to be eccentric. And this is because if you increase the value of our eccentricity term here, Z17 squared plus Z21 squared, that basically weights it more in the direction of the first term here, which then pushes our overall score over 0. And finally, we can tell from our model that cell state is independent of spatial orientation. And this is because our eccentricity term here, one, it only includes the squares of the terms, which means that it doesn't really matter which orientation it is. And it also treats Z21 and Z17 equally, which means it doesn't really matter. Again, Z17 and Z21 have slightly different sensitivity to orientation, and this doesn't matter. So basically, we know that our eventual model has learned to be invariant to spatial orientation, which is quite cool. So in other words, we basically know that we can create a set of models that all have a very similar accuracy while having very drastically different levels of interpretability. And the models that are more interpretable can tell us a lot more about what they're doing without sacrificing a lot of accuracy to get there. Do I have, how much time do I have?
I think we are right over. There was some, yeah, I think we have
So,
to move along.
Yeah,
okay.
if you can wrap up because we're running late. Yeah.
Okay, sure. Okay, I'll skip the adversarial attacks, but anyway, basically the conclusion is that it's possible to obtain high accuracy interpretable domain appropriate models, and it's possible to use them to gain scientific knowledge of an underlying system, but it can be tricky and requires the use of some techniques. And the next steps forward, so the problem I've showed you is very simple, literally just essentially boils down to, is this cell round or not? But the goal of this, the intent is to use it for systems in which we lack the prior knowledge and actually want to use deep learning to gain new scientific knowledge. So that's it. And thanks to everyone involved. Thank you.
Okay, thank you very much. Again, in the interest of time, maybe we'll have one question. Okay, if not, we move to the next presentation, which is Vim for Path, self-supervised state space modeling for histopathology images. And the speaker will be Ali and Siri Sarvi from Concordia University. And that paper, the other co-authors are all from Concordia.
Is the speaker online or in person?
Yeah.
Also online.
Okay, you're online. Okay.
Yeah. Thank you, Christopher.
Can I share your screen with your
Ali, if you could share your screen.
Yeah. And also, please make sure it's only 10 minutes because we're very late in the program. Yeah. Thank you.
Yeah, sure. Can I see my screen?
No.
Allow recording? Or no? Okay.
That's okay.
Sorry.
Yeah, please get started, Ali.
Okay, yeah. Hi, I'm Ali Nasir Sarvi, master's student from Concordia University and Atlas Analytics Lab. Unfortunately, I couldn't be at Seattle with you, but I will present our work through the virtual platform. Our work is focused on Vision Mamba trained on a self-supervised manner for histopathology images. So the problem that we're trying to tackle is when you're learning representation from gigapixel whole site images, you usually have, because of the image size, you have some aggregator models, which could be multi-instance learning or MIL. And because of scarcity of detailed labels, you want to use self-supervised learning as well to achieve the highest performance you can from your data. The main thing is the feature encoder that you choose can heavily influence the overall performance of your system. And in this work, we basically propose using Vision Mamba as an alternative encoder to vision transformers and CNNs, which are very common in computer vision and pathology databases. So as I mentioned, with gigapixel images, we have an MIL, we have our whole site image that is gigapixel. We would extract patches from non-background regions, and we would have bag of patches. We would pass them through a pre-trained feature extractor and get patch embeddings. And we would train an MIL network solely on these patch embeddings, which are low dimensional. So you don't require as much resources as you would have required if you trained on the whole the gigapixel image. And this feature extractor is usually always pre-trained, and our focus is on basically improving the performance of this feature extractor. So Vision Mamba, this image is from their paper. Vision Mamba is based on the Mamba architecture, which was introduced last December, and it was introduced for language modeling as an alternative to transformers, and it solves a lot of issues that transformers have with the scaling. So Vision Mamba and Mamba is basically a sequential model for language, audio, DNA sequences. And what authors in Vision Mamba paper do is they add two things to Mamba. One is a 2D position encoding because of the 2D structure of images, and also a bidirectional. They use a bidirectional Mamba instead of one Mamba because of images are not causal like language, and you need your tokens and patches would depend on previous and next tokens. So they use bidirectional Mamba and add positional encoding. So if you want to basically compare the architectures of Vision Mamba, Vision Transformer, and CNNs at the abstract level, what we would have is with convolution, if you have these patches A, B, Z, convolutions because of the inductive bias of kernels, what you would have is you would be able to capture these short range dependencies between A and B very good because of the inductive bias, but your model would fail to capture any long range dependencies because of the limited kernel size and perceptive field. With Vision Transformer, you get rid of that inductive bias and you introduce attention mechanism and you treat all the tokens and patches the same. Because of that, because of the lack of inductive bias, you usually need more data to reach the CNN level of performance and usually outperform them with enough data and enough training. But one issue with Vision Transformers is, first of all, if you want to increase the image size, because of quadratic scaling of attention mechanism, you would need a quadratic memory, which is not really good. And also because you have no inductive bias, the only thing that would separate between A and B and the pairs of A and B and A and Z is positional encoding. And you need to train a lot for the positional encoding to basically be able to distinguish between short-range dependencies and long-range dependencies. With Vision Mamba, you have an inductive bias, which is not as restrictive as the CNN one. It can capture long-range dependencies. Vision Mamba is bidirectional. We are just showing one direction here. You have, because of the sequential processing, the short-range dependencies of A and B are captured much better. And because of Mamba architecture, its performance on longer sequences, you can also capture these long-range dependencies between A and Z. And our basically architecture here is we are training using Dino framework. And with Dino, one important thing with Dino is you have this student model. And this student model is given different sets of views or image sizes. And your model, any model that need to work on Dino, it needs to be able to process images at different scales and different sizes, it should maybe use global average pooling or any methods to handle it. With Vision Mamba, it cannot do that inherently. So what we did was we added a positional encoding interpolation module. So if you have your default image size, you would just pass on the default positional encoding. But if the image size is lower or higher, you could just interpolate the positional encoding and then add it to your patches. With that modification, Vision Mamba is now capable of processing images at any image size. And the dataset that we trained on is Camilla 16, and this is for metastatic cancer in breast cancer patients' lymph node, and it has ROI annotations as well. And these are the statistics of the dataset, and we train our model at 10x zooming level, and that's mainly because of the computational limitations. And we get around 1 million patches in total, and these are not labeled patches. Like these are just like 1 million images. We also have we extract labels from the ROI regions. I didn't show it in this presentation, but you can also look at it in our paper. So for a side level classification performance, we provided here. We mainly compared against vision transformers because they performed the best when we train with Dino. Vision transformers are much better than CNNs and their overall performance. And we also provide some baselines from other methods, but we don't compare with them because even though our method is performing much better, they are trained at 20x, and with they have pre-training weights from other datasets. And what we find out is with Vision Mamba at a smaller scale, it's and the metric that we care about is AUC because of imbalance in dataset, and we didn't tune for the thresholds. And with AUC, with Vision Mamba tiny and Vision Transformer tiny of its equal size, the performance of Vision Mamba is significantly better, around 8% AUC, which is great. But when you scale Vision Transformers, the gap is reduced, but still Vision Transformers are outperformed by Vision Mamba of its equal size, and even Vision Mamba of much smaller size is outperforming the Vision Transformer for Camilla 16. We also look at the heat maps of our these Vision Mamba is small and Vision Transformer is small. And what we find, and we pass these heat maps that are extracted using DirectCam to our board certified pathologists. And what we find out is that Vision Transformers are mainly focused on just trying to distinguish between cancer and non-cancer. And all of these models are also they haven't seen labels, so they don't know what is cancer or not. These are all pre-trained models only. And what we find out is Vision Transformers behavior is similar to pathologists in cases that require minimal examination, and they are very obvious for a pathologist to say whether it's cancer or not. With Vision Mamba, it's mainly focused on pathology-specific features that pathologists would use to identify whether an image is cancer or not. So on Camilla 16 and this tissue and this tumor type, it seems that Vision Mamba is mainly trying to reproduce what pathologists would follow for their difficult cases. And that's our work. Thank you to the audience and the organizers. I also wanted to thank my co-authors and supervisors. You can find our work through this barcode. And if you have any questions, you can ask me here. And a little bit of self-advertisement. I'm also looking for PhD positions for fall of 2025. If you have a position or know anyone who has an interesting position, I would appreciate it if you contact me on any of these platforms and we can chat about it. Thank you.
Yeah, thank you, Ali. Does anybody have one question? Possibly to us. Okay.
How does scale to 3 dimensions?
How does your method scales in 3 dimensions?
On 3 dimensions,
yes.
I'm not familiar with processing at 3 dimensions, but what you could do is if you have any sort of tokens here and you can have some linear projection from your 3D or any sort of data set that you have, if you can extract them and similar to transformers, you can just make them a sequence of tokens. And if there's a sequential structure, you can just pass them to, you can have a 3D Mamba architecture basically for that.
Okay. All right, thank you very much. So again, now in the interest of time, we move to the next and final paper. Thanks again, Ali, for the last paper, which is on low resolution only microscopy super resolution models, generalizing to non-periodicities at atomic scale. And the speaker is Björn Müller from Technical University Braunschweig, right? Okay.
PC here. Yes, use USB-C connector to your protector. You need it. Jack. Also, you need this. You need this. Also, you need this. You need this. Yeah, I need it.
Okay.
I see. Use my computer, just mute everything.
It's muted here.
Okay.
But
it should work, right?
it's muted here too.
Yeah, muted.
Okay.
Now you can try to talk. See if you can.
All right. So I was just explaining the low resolution only aspect of super resolution microscopy models. And we basically have a low resolution input that is processed by a super resolution model producing some output. And that output is compared to a high resolution image, and an error signal is computed, which is then used to train a super resolution model to reconstruct this high resolution image.
Now, for low resolution only trainings, the high resolution image is not available, and we have to derive the training completely from the low resolution, available low resolution image only. Our work is in the field of material science, and we train those models to super resolve scanning transmission electron microscopy images of atomic structures. A problem that occurs is that the amount of training data is low and also very low in variability, which causes our models to overfit to the highly repetitive patterns, as atomic lattices, especially of crystalline materials, are very periodic. For our use case, we captured images of gallium nitride and indium gallium nitride at multiple magnifications, and we focus on the pixel sizes of 5 picometer and 20 picometer here, which differ by a factor of 4, which is also the super resolution factor for our models. The challenge here is model generalization, especially to non-periodicities occurring within those highly periodic structures. In the right image, in some atomic layers, the gallium atoms are basically exchanged for indium atoms, introducing this higher intensity area or area of higher brightness, which we call a non-periodicity. As described, we follow the low resolution only training paradigm, meaning we use the 20 picometer low resolution images for training and the 5 picometer high resolution images for validation and testing only. Our approach is a training pipeline that includes the strong augmentation technique of PixMix, which was originally designed to learn robust representations for higher level vision tasks. This is the diagram of our pipeline, which creates low resolution, high resolution training pairs from the available low resolution images. It utilizes data augmentation and additional out-of-domain mixing images to introduce variability into the microscopy data. The pipeline starts by scaling the training image in the range of 25% and 400% of the original image size, which is based on our earlier work. Then you have to define a mixing data set, and then you have to take images from the mixing data set and the training data set, drop those to the same size, and those are input to the PixMix augmentation. PixMix is an iterative, highly randomized process in which the training image is mixed with a transformed version of itself, or the mixing image, which stems from an additional data set. It has two main components, basically the random addition or multiplication, which defines the mixing, and the random image transformation, which includes a set of basic transformations. Now, after k iterations, we yield a pseudo high-resolution target image, which we then use to downscale the image by a random degradation function to obtain our low-resolution, high-resolution training pair. And with those pseudo training pair, we can do model inference and calculate the loss. Basically, every component in this pipeline involves some form of randomness, which can be configured. And for the most important ones impacting the augmentation range, mixing frequency and mixing intensity, we do a parameter study in our work. Here I brought some example outputs for pseudo high resolution images from our pipeline. Starting on the far left, there are two example training images, and right next to them there are example images from our mixing dataset. The mixing dataset shows interesting fractal-like structures with lots of image detail. And in the green box, you can see the output examples for the PixMix augmentation at the iterations 0, 4, 20, and 30. And the top row has a weaker mixing intensity, meaning the mixing image has less impact during the mixing, while for the bottom row, there's a stronger impact. we can clearly observe that the method creates quite some interesting training images, introducing a lot of variability to the atomic patterns. For our experiments and results, we tackled a 4 times super resolution task using a Squint transformer model. And as we adopt this augmentation technique from a higher level vision task to the low level super resolution task, we did a hyperparameter study and found that a relatively high augmentation strength involving up to 30 iterations of mixing the images performed best in our setting. Here we compare with 2 baselines. One is by cubic interpolation upscaling, and one is basically random rotation and flipping, which I call basic augmentation here. We test on non-periodic high-resolution images and report PSNR, so peak signal-to-noise ratio, and a structured similarity index measure, SSIM. Those 2 are image similarity metrics, and we show that we can improve PSNR by almost 1 dB when generalizing to non-periodic structures. But also, we captured the data in multiple data capturing sessions and reserved the data from one of those sessions for testing only. This allows us to also evaluate the generalization capability to unseen noise characteristics stemming from the configuration of the microscope during this session. And here we can see that the image similarity can be improved even more. For qualitative results, you can see on the left side the high resolution target image. Then there's bicubic interpolation for reference. And in the middle, there is a model trained with basic augmentation, where you can see the problem of missing generalization or overfitting. As you can see, the image lacks, or the reconstructed image lacks a lot of its structure. And using our pipeline, which involves the PixMix augmentation, a lot of that structure can be reconstructed. And so our pipeline even enables the reconstruction for those unseen noise characteristics. Now, to conclude, we improve super resolution model generalization for non-periodic high resolution STEM data of atomic structures using only periodic low resolution STEM data for training. We propose a training pipeline for training those models using low resolution data only based on PixMix augmentation. And we also show improved generalization to recording specific noise characteristics. And lastly, for future works, I would recommend to try out different mixing data sets. Right, thanks for your attention.
We have time for maybe one question before we move to the live talks. Okay. Right. Thank you very much.
Yes, I will be glad to host it.
We will also be at the session for people to ask more questions. Yeah.
So I guess you.
Yeah, I will. Thank you so much.
Thanks.
Paula, are you still there? Paula, is it getting pretty late for you?
Yeah,
Yes.
I thought there would be a break in between, but I'm here.
Yeah, let me try to fix this. What did I do? Okay, yeah. So we ran way over, but the talks are all really interesting. So just so that people can have lunch, since we are already after 11, Paula, are you okay to just, kind of talk now? So we won't have enough break. People should feel free to take your own break. And so we can more or less finish. And so people have time for lunch and move on with the afternoon session. So Paula, let me introduce you while you try to share your screen.
Sorry, May, are you talking?
I'm looking for a file. Yeah, Paula is a senior scientist at the machine learning research group from Bayer AG in Berlin, and is a board member of the Cytodata Society. She obtained a PhD in biology from the German Cancer Research Center, DKFC, and a master's in applied mathematics from, I'm going to butcher the pronunciation, Eindhoven University of Technology, and also a Bachelor's of Science from Biological Engineering from the National University of Columbia in Medellin. Her research focuses on computer vision applications in drug discovery with emphasis on phenotypic profiling and cell painting. I hope I didn't butcher too many pronunciations. Yeah, please take it away.
Thank you. A small disclaimer, I have a very bad cold, so I hope I don't start coughing in the middle of the talk. So thank you for the introduction. And today I will be telling you a bit of what we're doing regarding self-supervised learning at Bayer on phenotypic small molecule screens. Let me first put the laser pointer. Yes. So here I'm showing the typical small molecule discovery pipeline, which consists of a funnel-like process where we progressively reduce the number of candidates before going to the clinics or the fields. And this type of discovery processes usually start with the so-called compound screening, where we perform high-throughput experiments and test hundreds of thousands to millions of compounds. Of course, assisted by robotic systems and miniaturized assays. And within those experiments, there are the so-called phenotypic assays, which are those assays that use images as a readout. And we use this type of assays, miniaturized assays, in both Bayer divisions, both in crop science and in pharmaceutical research. And in crop science, we use it within the context of herbicide, insecticide, and fungicide discovery, like in this example where we're growing small Arabidopsis plants in 96 well plates, searching for new herbicides. And in pharmaceuticals, we use it in the context of drug discovery, where we usually test compounds on cells and use fluorescence microscopy to image the results. And these image data sets are very large and very powerful because one can infer many things from the data sets, such as toxicity, off-target effects, or so on. However, there is always a question on how to extract meaningful features for each application, especially because these assays are these large screens are exploratory in nature, and of course, we don't have any labels. And therefore, that's how self-supervised learning comes into place, where we design surrogate tasks that can help us learn representations in this type of screens. And in this talk, I want to show you some examples of how we have applied self-supervised learning in crop science and in pharmaceuticals. So starting with crop science, here the example is from herbicide screens where we have image time series. So here is the type of data that we generate in our herbicide screens. We treat these Arabidopsis plants, we either leave them untreated, so the neutral control, and monitor the growth of the plant for several days or almost a week. And the plant can grow normally as shown in the first row, or we can treat them with potential herbicides, so with new compounds, which will kill the plants with different symptoms and also at different time points. And to find
Paula, just a sec, we can't hear you in the room. In just a sec.
You didn't hear anything?
No, just the last sentence. Now we can hear you.
Okay.
Some connection error.
Okay.
You were just starting to
Right.
talk about the slides, so now we can hear you. Please continue. I think some connection is not good, but now we can hear you. So just continue.
Okay. Yes, so maybe the last part. So we treat the plants with compounds, which could be potential herbicides, and kill the plants by different symptoms, and the symptoms can appear at different time points. And to design a self-supervised task for this type of data, we thought that we could exploit the time point information, relying on the fact that images from consecutive time points should be more similar than images from non-consecutive time points, since we know that the plants die progressively and usually the phenotype is not reversed. So based on this fact, we formulated a triplet ranking task with a triplet network where we always feed a reference, positive, so more similar, and negative, less similar images. And we use a ranking loss to ensure that the distance to positive is always smaller than the distance to negative. And to handle the complete time course information, so once we get a time course, each image is processed through Siamese networks, which provide embeddings for each individual time point. And from this, we define all the triplet combinations, possible triplet combinations with consecutive and non-consecutive time points to train the ranking loss. And at the end, at difference time, to have a descriptor of the complete time course, we simply concatenate the embeddings from the individual time points. And this strategy turned out to be very efficient to find representation of this type of data. So, for example, we could see that our embeddings show very meaningful biological correlations. Here on the left, I'm showing a UMAP projection of our embeddings, colored by the health of the photosystem at the end of the experiment. And here we can see that the embeddings clearly separates between damaged photosynthesis here in red or healthy photosynthesis on the right. Also, if we color by mode of action, so two common modes of actions, which is a amino acid synthesis inhibitor or a photosynthesis inhibitor, we also see that the embeddings clearly separate the modes of action and also the healthy plants. Besides that, we could also use our embeddings to find phenotypic clusters, and we found that these clusters followed a very nice organization, were very nicely organized in the embedding space. We see this by constructing a minimum spanning tree from the cluster centroids, which basically shows a 2D visualization of the hierarchical organization of the cluster centroids in the high dimensional space. And what we could see is that, for example, if we follow one of the branches in the minimum spanning tree, for example, the red one, we see yellow in phenotypes with increasing strength, while if we follow the yellow line here, we see brown in phenotypes also with increasing strength as we move along the branch. And finally, we could also use the embeddings for similarity retrieval. So here I'm showing on the top a query image that was not included in the training set, and the first and second closest neighbors in the training set. And you can see that the embeddings match very powerfully not only the symptom and the strength of the symptom, but also the time course of appearance of the symptom. A disclaimer, so I didn't include it here, but for this really to work, we also applied the typical variance normalization that Berton talked about, where we do PCF whitening on the DMSO controls. All right, and this brings me to the second part of the talk, where I will show you an example from pharma. And here we have been mainly used self-supervised learning for learning feature representations in cell painting images. And instead of, like in the example of crop science, instead of coming up with a self-supervised task by ourselves, we performed a benchmark study of SSL methods from natural images on cell painting images, which I will show you in the next slides. So in the benchmark study, we included 3 SSL methods. So the masked autoencoder, which is trained on the task of reconstructing a partially masked input image. We include Sinclair, which is based on contrastive learning, which tries to pull together augmented views from the same images and push apart views from a different image. And finally, Dino, which is inspired in model distillation, which tries to maximize the agreement between a student and teacher networks, which receive different augmented views of the images, and where the teacher weights are computed as an exponential moving average of the student weights, among other details. And we also included 2 baselines, so the vanilla transfer learning, where we feed each channel independently through a pre-trained architecture, to an architecture pre-trained on ImageNet, followed by a concatenation of the single channel embeddings. And finally, we also include Cell Profiler, which basically performs first a single cell segmentation and then computes handcrafted features on single cells. And in contrast to Cell Profiler, I want to highlight that all the deep learning models included do not require segmentation, because they are based on random crops, or on crops of the images. All right, and the data set that we used for this study was a subset of the JOMP-CP consortium, which provided images for 100,000 chemical and genetic perturbations imaged with repetitions across different labs. We didn't use a complete data set, but a subset of around 10,000 compounds, which came from a commercial library. So we know that they are bioactive from the Select Chem and MedChem Express libraries. And we included repetitions for each compound had repetitions from 4 different laboratories. And for validation, we used the target 2 plates from the same laboratories as the training set. And these plates are sentinel plates that are imaged in every experimental batch, and therefore are very nice, provide a nice alternative to study batch effects and to study biological signals, since they also have available target annotations. All right, and last about the last slide about the study design, to compare the models, we used several metrics of reproducibility and biological relevance, all of them based on k-nearest neighbor classifiers. So for reproducibility, we performed a k-nearest neighbor classification of the perturbations, so basically predict which compound was applied to the image. And here we report the mean average precision and the not same batch nearest neighbor accuracy, which is a nearest neighbor classifier, which restrict matches to compounds that don't come from the same batch. And for evaluating biological relevance, we use K-NN classification of annotated targets in the validation sets, and again report the mean average precision or the not same batch or perturbation accuracy, which restricts the matches to data points that don't come from the same batch or the same compound. So as you will see in the presentation, we always show these plots for each of the metrics. All right, and before starting to compare the models, we first wanted to understand what is important to take into account when we use self-supervised learning methods that were used for this that were designed for natural images. What is important when we use them and apply them to cell painting images. So for example, do we need to do the same augmentations? Do we still need normalization and feature selection as is usually done for cell profiler profiles, and so on? So first, to study the relevant, to identify the relevant augmentations, we did a composition study similar to the study presented in the Sinclair model, which I show here on the right, in which we report the performance. Here, in this case, we're looking at the reproducibility metrics of pairs of perturbations. And what we see here is that similar to natural images, the color transformation, the color augmentation is very important for cell painting images, and that this augmentation is even more prevalent in cell painting images than in natural images. Okay, here I forgot to say that, of course, for cell painting, we don't color doesn't make sense in the RGB color space, but we generalize this augmentation by including random brightness change and intensity shift applied independently to each channel. All right, and another interesting finding from here is that the size and crop and resize operation, which is also quite important for natural images, is actually detrimental for cell painting images. And if we think about it, this actually makes sense because the JOMP-CP data set was all imaged at the same resolution. And if we do this resize operation, we're pushing the model to be invariant to changes in cell size, which is actually a desired feature or an important feature to define cellular phenotypes. So we therefore excluded this resize operation from our augmentations. The next question that we asked was about plate normalization. Do we need plate normalization, and or what is the best normalization strategy for SSL models? And we tested robot different normalizations and combinations of these normalizations, including Z-scoring to a complete plate, robot Z-scoring, and sphering, which is the typical variance normalization proposed by Ando and et al., which, as I said before, performs a PC8 whitening based on the controls of the full data set, not per plate, and basically puts the negative controls of the data set to the unit sphere. And the interesting findings that we found, that we got from here, is that plate normalization is also essential for SSL models. So there is a very large performance gap between the on-process profiles and the normalized profiles. And second, we found that sphering, so the typical variance normalization transform should be applied plate normalization, since as you see here, the two scenarios where we applied sphering after C-scoring produce the worst normalization, the worst results, and sometimes it's even detrimental. And finally, that the sphering followed by robot C-scoring is the best normalization for the SSL, for all the self-supervised models, as you see here on the top. And the last question regarding processing of profiles, we also asked whether feature selection is needed for these models, and what we included two feature selection methods that are available in PyCytominer. So removing features with low variance or features with low variance and highly correlated features. And we saw that although for Cell Profiler, you get an improvement, performance improvement by performing feature selection, for the deep learning models or the SSL models, this was not the case. So then, based on these findings, we proceeded to compare the SSL models with our baselines using the best processing post-processing method which we found for each model, which we found before. And here I'm showing then the three SSL models, our two baselines, and some randomized baselines as well. And we also included the small vision transformer. So all the deep learning models are based on vision transformer architectures, and we included the small as well as the base vision transformer. And here are the results for the reproducibility and biological relevance metrics, where we can see that both mask autoencoder and Dino either match or exceed the performance of Cell Profiler, here in blue, in both metrics, with Dino being the best model showing the best performance among all the models. And another advantage of Dino, but actually that can also be, I mean, not specifically from Dino, but in general from the SSL models, is that even with Dino, that is quite computationally intensive, we are able to be 50 times cheaper and faster than Cell Profiler. But one result that puzzled us was the lack of scalability between the small and the base vision transformers. So if you compare the circles and the triangles, they are always very close to each other. And since we didn't use the full consortium data set, we think that the data set size was a limiting factor to see scalability in this case. And indeed, as I said before, our data set are 10,000 compounds imaged in 4 different laboratories. And we did a small experiment where we train our models on the data set of a single lab compared to including the repetition from the 4 labs. And here we see that indeed the performance scales quite well by adding data from more labs. So yeah, we hypothesize that including more data will help us to see the scaling of the vision transformers. And finally, we can also see, we can confirm the results from our quantitative metrics visually. So here I'm showing a UMAP projection of well profiles color-coded by target annotation for the most prominent 20 targets in the validation set. And you can see that for MAE, Dino, and Cell Profiler, we see very nice clusters of targets, while for Sinclair, the results are not as good. And besides that, we also wanted to test the performance of our best model, so Dino, in a different data set. And for this, we took the gene overexpression data set from the JumpCP consortium. So this data set was particularly, will tell us about the generalization of the models because it includes a different perturbation type, so genetic versus compounds. And it was also imaged in a different lab from the training set and the validation sets. And here I'm showing the results for the hierarchical clustering of Dino profiles compared to cell painting profiles, where we see that Dino recapitulates better clusters of gene families. And this is, we can see this qualitatively, but also if we compute the adjusted mutual information between the cluster assignments and the gene family labels, we see that Dino has a higher mutual information compared to Cell Profiler. However, not everything is perfect. And one of the drawbacks that we saw from the SSL models is that they are more susceptible to batch effects and laboratory effects compared to cell profiler. So here again, I'm plotting UMAP projections of well profiles colored by batch or by the laboratory in which they were imaged. And as you can see, cell profiler shows a more homogeneous distribution among batches and laboratories, while the two SSL models show very obvious distinctions between the sources. If we look at the cell count, we see that these differences are related to differences in the cell count. And this kind of makes sense since cell profiler performs feature extraction in single cells, while the SSL models take image crops and therefore are including several cells with background, which also gives information about the cell density to the embeddings. All right. And in the last slides, I want to tell you some of the examples, or tell you a bit how we're leveraging cell painting image features in small molecule discovery. So the first example is, in the first example, we try to improve the prediction of mitochondrial toxicity models. So we try to complement QSAR models, which predict mitochondrial toxicity based on chemical structure. And on the left, I'm showing images of healthy of a DMSO control with cell painting, where the red channel is the mitochondrial channel, and on the right of papaverine, which is a complex 1 inhibitor, so a mitochondrial inhibitor. And here we could see very clear changes in the red channels, not only in the intensity, but also in the distribution of the channel inside the cells, which motivated us to think that if we include cell painting profiles to complement as an input to our models, then we could improve predictivity. And here we show the results of the performance of the models, including chemical structure, cell painting, or both, for two different holdout strategies between train and validation sets. So a random holdout, so these are very common in chemoinformatics. So a random holdout where the train and validation sets might be structurally similar because they are split randomly, and a cluster holdout strategy where the training set, where compounds are first clustered, and then based on this cluster, the training and validation sets are defined. And therefore, in this case, it's the most difficult case because the training set is structurally different. The compounds in the training set are structurally different from the compounds in the validation set. And what we see here is that combining cell painting with chemical descriptors improves a bit the predictions of the models. However, these differences here were not significant. And therefore, to further improve the model, we proposed a strategy where we expanded the data set for the chemical models using cell painting, using the cell painting models. So the motivation here is that in toxicity, it's always very difficult to find large data sets, and data is always a limiting factor. However, we have hundreds of thousands of compounds for which we have cell painting data. So the idea was then that we could use the models predicting mitochondrial toxicity from cell painting profiles in order to label all the available cell painting data, and then pick the most certain predictions as let's say pseudo labels of mitochondrial toxicity to extend the data set for the chemical models.
Paula, it happened again. So it just let us.
Oh no,
I don't know. Last time what fixed it? So it's working again. Paula, can you try to speak? Okay, now we hear you.
So where did it stop?
No idea. Some connection issue. Now it's working.
All right. So where do okay. So to summarize this slide, so the idea will be to extend the data set for chemical compounds, because toxicity labels are always very scarce, but cell painting data is very abundant. So what we did is to use the models predicting toxicity from cell painting features to predict toxicity in the remaining of the cell painting data, so in the very large cell painting data set, and then use the most confident predictions as pseudo labels to enhance the data set for the chemical models. And here we see the results of the baseline and the extended training set, both for the random and cluster splits, where we could see that for the random, the difference was not so much, but especially for the cluster split, which is the difficult case where the model needs to generalize to new chemistry, enhancing the data set with cell painting predictions significantly improved the performance of the model. All right, and another example comes from our colleagues in agrochemical safety assessment. And there, before they releasing a product to the market and spread it in the environment and in the fields, it is important to determine the acute oral toxicity of the product. And this is done by feeding rats with the product, and then finding the single dose at which mortality is induced in at least 50% of the animals. And since this is an in vivo assay, then it's very expensive, and it also raises ethical concerns about unnecessary experiments. And there are already some QSAR models predicting acute oral toxicity based from chemical structures, but we saw that these models didn't generalize well to the biochemistry. Therefore, what our colleagues in safety assessment did was to obtain cell painting profiles or cell painting images for 216 compounds from our library at 3 different concentrations, and train classifiers on chemical descriptors, in this case, Morgan fingerprints, or cell painting features. And here, then we classified the compounds as being very toxic or non-very toxic, taking different thresholds on the LD50 value. And as similar to the previous project, we also use the 2 different holdout strategies, the random, which is the easy case, and the cluster splits, which is the difficult case. And here are the results for the chemical descriptors and the different cell painting concentrations. Not cell painting concentrations, but like concentrations, image, and cell painting, where we can see that in the case where we have random splits, the chemical models are much better than the cell painting models. However, when we try to, the model needs to generalize to new chemistry. So in the cluster split, the cell painting models at the middle concentration show a much better performance than the chemical models. And therefore, based on this, our colleagues in crop science now implemented a decision support model, which decides on which prediction to use depending when the chemical or the cell painting model don't agree with each other. And this is very interesting because it shows again, or it confirms again that cell painting profiles are mainly beneficial to generalize to new chemistry, since the profiles just reflect the biological effect and don't depend on the similarity between the applied compounds to the cell. And the last slide shows the last application, how we're using cell painting images. This is a more out-of-the-box idea, where an exploratory project where we inspired in the latest, now not so latest, generative models. So, for example, the models where we provide text, and then it synthesizes images for us, such as in these examples from the transformer autoregressive model DALI or the diffusion models, where if you give the text an armchair in the shape of an avocado, you get these images synthesized, for example. We thought that we could try a similar approach, but instead of feeding text, we could feed image features to a generative model, and this generative model will propose compounds which will have a high probability of inducing the desired phenotype that we see in the images. And this is then addressing the task of de novo molecular design, proposing chemical structures that satisfy a desired phenotype. But for the sake of time, I'm not going to talk about this here. This was my last slide. But if you're interested, you can go to the paper, or there is also a recording in the M22 talk series from Valence Labs. And yeah, I want to thank all the people that contributed to this work, and I will be very happy to take any questions.
Yeah, thank you so much, Paula, for a fascinating talk and really good time management. We have time for yeah. Any questions in the room? Yeah, go ahead. Loud, or come to the front. Loud, please.
Thank you so much for the nice work. I have a quick question about actually, again, about the value.
Can you hear the question, Paula?
Yes, but I'm sorry. It's like I don't see the it's fine.
Come here. The camera is here. Yeah, be loud.
Oh, right
yeah,
here.
Be loud. Yeah,
it's good. Oh yeah, I'm asking about the batch effect when you try, to analyze data across multi-sources, right? Different pharma companies. So I understand the total variation normalization and the scoring when you work within the same data from same company. So I'm asking, did you find the challenges to harmonize this data when you try, to look at the same perturbation or effect across different sources? Did you do a special? Yes. Thank you.
Okay, maybe I can share my screen again. Just a second. Yes, we found it very challenging. And for example, here in this plot, I will zoom in. I'm reporting the performance of the models when we match, but restricting across sources. So we only match perturbations from one source, so from one laboratory to the perturbations of another laboratory. And there we see a very large performance drop compared to the performance that I showed you before. And yes, I think this is still an open question. And yeah, we're also doing some research in trying to include batch normalization or train networks which are aware of the batches so that they can, during the learning process, take into account these biases, but it's still very inconclusive in this direction.
Yeah, thank you for the question and answer. Any other questions in the room? Any questions in the Zoom room? Hello, are you talking from Berlin? It's pretty late on your side, right?
No, I'm in Colombia now. I'm from Colombia. I'm here, so it's not. It's just 1:40 p.m. It's fine.
That's good. Yeah, we don't feel too guilty now. Yeah, thank you so much for bringing us back on schedule, and I hope you can stay, for some other talks. So we'll move on with the program. Thank you so much again.
Thank you.
With a couple more accepted papers. I think the upcoming one is from Oxford and Yale collaboration. Is that Alexander? Yes. Okay, yeah. Let's set things up. You need
oh yeah, we have an adapter.
Yeah, we have USB-C, yeah. And mute yourself on Zoom. Share over Zoom, but mute yourself. And because you can use it. But share over Zoom, okay. Over Zoom.
Now I can do it.
Are you in Zoom?
Yes.
Should I share?
Sure.
Yeah, we can see it. You probably need to go to slideshow. Yeah, now it's working.
Okay. Yeah, thank you for having me and allow me to present our work on refining biologically inconsistent segmentation masks. So to get started, this work comes from a project working with extension microscopy. And extension microscopy is a way to achieve super resolution with normal confocal microscopes by basically expanding the cell physically.
And more specifically, we are interested in mitochondria. So let me quickly recap about mitochondria. So mitochondria, they have like a double membrane structure. So you have like an outer membrane, and then between the outer and the intermembrane, you have the so-called intermembrane space. And then the space within the inner membrane is called matrix. And now this inner membrane, in order to maximize its surface area, forms these invaginations, which are called cristae. And the important thing to point out is that this kind of biological structure imposes some constraints on what like a valid segmentation means. So for example, you would not expect to see the inner membrane directly touching the background, or because the cristae are these invaginations by the matrix, you would not expect to see any cristae appearing on its own without any surrounding matrix. And as long as your images are that nice as the example shown here, that's not a problem. But then sometimes with microscopy, you know, we have like certain artifacts, or like the staining is a bit off, the sample, the imaging, all kind of things that could lead to potentially an uneven signal quality. So for example, the two images shown here, like you have the matrix shown very clearly, but then with that area, it's not quite clear what's going on here. So when we applied our segmentation model to that, you could kind of see that this also shows in the segmentation now. So you get segmentation masks that violate some of the constraints that I mentioned earlier. And on top of that, you also get like fragmented boundaries, something that you just like would not expect to see. So the aim of the work is basically, can we correct for that? And can we impose a prior that basically allows us to have only segmentation masks that are biologically valid? So the first step in doing that is, of course, to basically first we need to find the areas where we have these potentially violations. And the way I framed this was that, well, we kind of saw these segmentation errors or inconsistencies, mostly in areas where they had that low signal quality. So you can basically reframe that question in asking, where in your staining do we have a lot of uncertainty about the resulting segmentation mask that we want to see? And then you can basically go back to the literature, and there's a lot of methods that investigate that problem of finding prediction uncertainty. So what we employed here in the end was basically just building an ensemble of models, and then looking at the disagreement between the models, and that interpreting that disagreement or the entropy between the model predictions as an indication for poor signal quality. So we ran a bunch of experiments, for example, like this one, where we took like a nicely imaged mitochondria and then artificially deteriorated the staining quality. And as we hope to see, we actually could observe that like this led to an increase in the entropy in that specific area. So coming a bit back to our example, so what we do now is basically we start with our global staining, have our model applied to that, and get like an initial segmentation just by the majority vote. And we also get a mask based on that ensemble entropy that allows us to see where we potentially make mistakes. And now the idea is to, okay, these mistakes, can we reconstruct them or fill them with like a prior that's like strong enough to only give us valid segmentations. And as you've seen a couple of times today, already like mask encoders work very nicely for that. So what's the idea here really? The idea here really is that within mitochondria, and especially in the ultrastructure of mitochondria, we just find a lot of self-similarity and local repetitiveness. So if we have, or let's say in our example, we have 10,000 mitochondria, and we only look at the mitochondria that look like imaged very nicely, and then are able to learn what these segmentations look like, we're able to basically fill in small areas like this easily from the surrounding areas. And to formalize that a bit more, so basically going back to the very, very basis of what we're actually doing here, like if we're predicting our segmentation mask, conditional our image, the standard thing that we do normally is just like we do like the conditional independence assumption and say we can basically look at every pixel individually conditioned on the input image. But what we do now in that work is basically say, well, okay, let's keep that for the reliable pixels, but then for the unreliable pixel, let's change that assumption. So let's not throw away
the remaining segmentation mask, so all pixels except our pixel I that we consider, but it actually throw away the underlying image because we just said, well, for these areas, there's not a lot of information for the segmentation mask. So for these pixels, let's actually just reconstruct them based on the prior of the surrounding areas that is not masked out here. So to learn this prior now, we go back basically to the basic idea of how to train a mask autoencoder. So we take our mask, we randomly sample the mask, and then reconstruct the original image. But the only important thing is that when we reconstruct that image now, we don't reconstruct on everything, but we only reconstruct on the images that we previously identified with the ensemble model as being of like high quality. So what does that look like? So for example, if we have an image like that, and then the initial segmentation mask is an example where we like violate our constraints. So as a reminder, that greenish here, that's supposed to be Christie area, and we said that Christie only like by definition has to be surrounded by matrix area. And now looking at the corresponding entropy mask, we actually really observe that this also lies in an area where there's a lot of disagreement between the ensemble models, and then applying our mask autoencoder trained on the reliable segmentations. This works very nicely that the model actually says like, based on that prior knowledge that I learned from like well-behaved mitochondria, we should not see that, and then it in-paints that or like reconstructs that in a sensible way, where like based on this surrounding just with intermembrane space. So we compare that to a bunch of different models like conditional random fields or an autoencoder that's like global. And we see that like this mask encoder, autoencoder strategy works very nicely. So for example, with that case, we see like very fragmented boundaries, but then again, they do appear in areas where there's like high uncertainty, and then the reconstructed case is a lot smoother. Yeah, a bunch more examples. But then remember that like the segmentation is only the first bit. Like in the end, we actually want to analyze these mitochondria in terms of like how large are they, what kind of morphology they have. So we try to quantify that a bit more by constructing various like auxiliary metrics here to see how well behaved our segmentations are. So we picked 100 mitochondria that had like segmentation masks that biologically were valid, and computed these metrics to like serve as a baseline, and then we saw that the method we proposed actually helps us to get these metrics the closest to the control group. So yeah, to sum up, we basically presented that like data-driven way to identify and correct biologically implausible segmentation masks. And I think the important bit really here is to emphasize that data-driven way, because all these constraints that I told you about, that like matrix should always appear around Christie, we didn't hard code that anywhere in the model. We just learned that purely from the data, and that made it very scalable. And then the final bit, we basically were able to show that this worked very nicely for our use case with mitochondria in expansion microscopy, but we would be really keen to like apply that to other methods. So if you know, or if you work on some data set that also has some similar structure with like very local repetitiveness with some forced staining, feel free to reach out, and I would be very happy to try to give it a go on that as well. Yeah, so a quick thanks to collaborators and funding, and thank you for your attention.
Can I ask you a quick question? You said the mitochondria have double, but can you see, or you see the thing?
So the way it shows up is normally that there's like that somewhat black halo around it, right. And that's kind of how you see it, but you can't really see the membrane.
Really the actual, as opposed to artifacts from imaging.
Yeah. So we applied some so the nice thing about that expansion microscopy is that you can, on top of that global staining, you can still apply immunolabels that specifically bind to that membrane. And you will see that, for example, TOM20 would basically bind all over that black space. So we would think that most of that actually is out of the intermembrane space.
Any other questions? Thank you so much.
Thank you.
Pretty much on point now. I think, and you are going to be at the poster session?
Yes.
Okay, yeah, so people can ask more questions and interact at the poster session. Our next talk of an accepted paper is from ETH Zurich. Is this Andre? Are you online or in person? You're online, right?
Yes, I am online. I started sharing the screen. Let me know if you can hear me
Yes.
and if you can see us.
We can hear you well. Let me connect my laptop.
Okay, so what about the screen? Can you also see it?
Not yet. I need to connect my laptop to the projector.
Yeah.
Just one sec. Yes, we can see. Not yet. Why we can't see it? We can see it on my laptop. We don't see it on the projector. Oh, we see it on projector. Okay, please take it away.
Okay, hello everyone. My name is Andrei Ignatov, and today I am presenting the paper Histopathological Image Classification with Cell Morphology via Deep Neural Networks. I guess I shouldn't give a long introduction on why do we need efficient computer vision methods for whole slide image analysis. Well, this kind of images, they contain lots of information, lots of visual information that is vital for understanding different cancer types, for predicting patient survival, for treatment selection, and right now even for gene expression prediction. So the majority of deep learning models that were developed in the past for this task can be divided into two different groups. The first group, the primary option, is that we are taking some common computer vision architecture, common CNN model, initializing it with ImageNet weights, and training on the target dataset. This method is very easy to implement, but as you know, the size of these datasets is quite small. And if you're taking a tiny CNN model, then it usually does not have enough predictive capacity, so the results won't be very good. If you're taking large CNN model, you will be running into overfitting. So in both cases, the results are not ideal. So right now, the majority of works moved into another option, selected another option. So it's about taking large vision transformer models, training them on millions of images in unsupervised way, usually with contrastive learning, and then using these models to generate features for images from your dataset and classifying these features with some external classifier. And this approach, it gives much better accuracy, and it can even be applied to new data sets without much of fine-tuning. But the problem here is that it's really hard to train these models. So you cannot do it like with a couple of GPUs. You need really sometimes like hundreds of GPUs, and the majority of issues come from unsupervised learning because it's hard to train the model fast and using limited resources. So what solution would we ideally want to tackle whole slide image analysis? Well, of course, we want to get even slightly higher accuracies than vision transformer models give. We want solution to be more interpretable. We want it to be easily fine-tunable on new data sets and trainable on just normal hardware that we all have in the lab. So if we have few GPUs, then the solution should be fully trained just on these GPUs. And the question is, can we actually come up with such solution? And the answer is yes. And here I move directly to the model that we developed. It's called DeepSeaMorph. And it proposes a different paradigm for working with whole slide image analysis. So the proposed model consists of two different modules, segmentation module and classification module. And segmentation, these are independent modules, and segmentation module is trained separately to learn cell semantics. So we are considering it like as a student model, and the goal of this model is to learn as much information about cell types, about cell shapes as possible, and let's say summarize this information. For this model is trained to perform nuclei segmentation and cell type annotations. So it should identify all cells, all nuclei, segment nucleus and provide the class of each cell on the input RGB image. And this is the output of this module. And this model is also trained on a couple of different data sets. Now the biggest question, why do we need this? Why should we use this segmental module, and why should we actually care about nuclei segmentation, cell type annotation? Well, when it comes to nuclei segmentation, this is nuclear morphology is extremely important for many diseases because it reflects cell type, cell cycle progression, and in particular, it allows us, for instance, to detect actively dividing cells. It's like one of the major hallmarks of cancer. Another thing is that deviations from a normal nucleus shape, these deviations are good indicators of external stress. So if we are able to detect some irregularities, this means that we can identify either mutated cells or diseased cells, so cells that are normal. And yeah, another just trivial reason is that it can be used for cell identification and counting. So if we identify cell nucleus, then we understand the position of the cell, where neighboring cells are located, and we can even see how many cells are on our whole slide image. And when it comes to cell type annotation, this is mainly about detecting immune cells, because this is probably the most important characteristics when dealing with the majority of diseases. Whether we have immune cells in our disease region, what type of immune cells, how good they are infiltrated into the diseased region, how they interacted with disease cells, and this provides lots of information that, in particular, pathologists are using for when trying to identify different diseases, in particular cancer. So this information on its own can provide in us background for coming up with a correct decision. And therefore, it would be very good if we have a standalone model that learns this information and is able to predict it for previously unseen images. And for this, the segmentation module, we trained it on a combination of almost all publicly available data sets that we found at that point. So these are 8 data sets that contain tissues of different types for different organs and cells of different types. And whenever available, we are also using cell type annotations. So this is how the training data looks like. So first, in the top row, we see original whole slide images. In the second row, this is segmentation maps, and the last row, these are cell type annotations. And once this segmentation module is trained, it is frozen, and we are then training the classification module. So usually when you're training classification network for this task, you're just taking RGB image, pass them to this model, and generating some predictions. Here we're taking this RGB image together with nucleus segmentation maps and with cell type annotation maps. So for each cell, our model already knows the location of the nucleus, its shape, the type of the cell, so quite a lot of extra information. And this classification module is pre-trained on TCGA dataset and then fine-tuned on the downstream task. And a few more technical details. So we're using Efficient MB7 model as a backbone for both segmentation and classification encoder modules. This is a very large and quite powerful model. It's fully convolutional, but it has a large number of parameters, and the entire solution is trained in an end-to-end fashion, no unsupervised losses, only cross entropy, and yeah, it's implemented in PyTorch and trained just on a few NVIDIA GPUs. Now the biggest question: can this model compete with vision transformers? And to understand this, we performed a couple of experiments. We benchmarked this model on pan-cancer TCGA dataset. This is a perfect dataset for understanding model capacity because it contains 32 different cancer types, and many of these cancer types are quite similar to each other, so it's quite difficult to distinguish between them. And also it contains over 270,000 image pages from over 7,000 patients, so it's a really diverse set, and it reveals the predictive performance of the models easily. Here are the results obtained on this data set. So you can see that like old-fashioned classical ResNet VDG-based models, well, they are showing quite bad results in general, but this is expectable. Then when we go into transformer models, in particular C-Transparse or Uni, they are showing an accuracy around 75%. These models are not untuned, so just taking the models as is, generating features and classifying them with SVM. But if we are taking C-transpose model that is publicly available and tuning it on the CGA dataset, we are still getting an accuracy of only 78.7%. So, the proposed solution, DeepSeaMorph, is able to achieve 82.7%, so almost 4% accuracy improvement. And this is despite the fact that both C-transpose and DeepSeaMorph, they were trained on this dataset and tuned on this dataset. And also on the slide, you can see pretty much an ablation study. If you're taking DeepSeaMorph and removing the segmentation module, you are getting Efficient-NB7 model. And Efficient-NB7 model here shows an accuracy of also 78%. So just the segmentation module that predicts cell types and segments nuclei, it just gives extra 4% on this task. Now moving to the next dataset, it's colorectal adenocarcinoma dataset. It's also relatively big, contains 9 tissue classes, and the beauty of this dataset is that it has an external validation set that was collected from different patients in a different institution, in a different geographical region, so it's perfect for benchmarking the generalization capacity of the model. Here we're getting pretty much the same results. Old-fashioned models, like 92–95% of accuracy. If I go into some standard transformer models, we're getting around 95–96%. C-transpose on this task shows 96.5%, and DeepSeaMorph still, it achieves the best accuracy, almost 97%, and 0.5% accuracy improvement compared to C-transpose. And here we also see the same pretty much difference between Efficient-NB7 without segmentation module and DeepSeaMorph that contains the segmentation module. So we see quite a dramatic improvement here. And the last dataset that we were interested in is a colorectal cancer dataset. The beauty of this dataset is that it is very small. So you can simulate how well the model functions on really a small number of image pages. Here we have only 5,000 images in image pages in total, corresponding to 8 tissue classes, so really a small dataset. And here the results are also not very different from the previous cases, like old-fashioned models shows accuracy up to 96%, C-transpose 98.2%, DeepSeaMorph shows slightly higher results, but yeah, it's still able to outperform more complicated, outperform previously proposed models. And here we also can see a 2% improvement compared to the model without segmentation module. So the segmentation module really matters. In the paper, we have some extra experiments. For instance, we show that this model can also be applied to images of larger size because it's fully convolutional architecture, so the size of the input image does not matter. It can be 1,000 times 1,000 pixels, 10,000 times 10,000, whatever GPU RAM you have, whatever your GPU RAM can handle. So in this case, compared to transformer models that are usually limited to some fixed size, quite often it's 224 times 224 pixels. Here, you can apply it to images of arbitrary resolution. And yeah, a quick summary. So the proposed architecture performs comparably or slightly better than transformer models. It is easy to train, it can handle images of arbitrary resolutions, and it is much more interpretable because if you want to visualize heat maps, usually they will show you the impact of the features generated by segmentation module, which means that the types of the cells, their relative position, and so on. And yeah, it can be trained just on a few GPUs. The only downside is that it requires fine-tuning, small fine-tuning on unseen data sets, but this isn't a big issue. And we have already released the codes and pre-trained models on GitHub. So it's quite easy it should be quite easy to integrate this model into your pipeline if you want to tune it on your own data set. So you just import this model from one file. It's PyTorch shown and no external dependencies. You define the model, define the number of classes, define the weights that should be loaded, and then you can get predictions, you can get features, you can get nuclear segmentation map and nuclear classification map. So everything is given. You can freeze and unfreeze some weights. So it provides quite a lot of flexibility. So we can hope that this model can be useful for you if you want to tune it on your own datasets. So that's it from my side. If you are interested in some additional details, please check our paper and GitHub repositories. And if you have any extra questions, you can always contact us by email. Thank you for your attention.
Yeah, thank you so much, Andre. Any questions in the room? People are hungry.
I think that
Any
people are hungry. They are already brave enough to stay here like 8 minutes past.
Any questions in the Zoom?
12.
Okay, so are you or any of your partners will be attending for the poster session? Will you be attending?
Me?
Okay, I think people can ask you questions via the email or your
yes,
QR
it's
code.
email. Yeah, unfortunately,
Yeah,
I'm not.
thank you so much. Yeah, thank you for the presentation. So now we break for lunch. We'll start punctual at 1 p.m. There'll be a talk on building foundation model for digital pathology using generative AI and on whole slide imaging. So hope to see you in 50 minutes. Yeah, thank you, everyone. We'll break for now.
There we go, Norris. Trying to go back here. Sorry, guys. All right. So eventually, we try to evaluate, how well this network to work. And the best solution was like we asked our pathologists to annotate a bunch across different tissues and try to evaluate how it works, right? So back then, our baselines were field models and relatively smaller than ours and regarding data and model size, and try to see how a bigger model, with bigger data trained, can work, right? So far, and I think so far with everything we benchmarked, including our in-house data set, Virchow is still kind of the best there. And we're waiting for basically newer models come out here to beat it, right? So that's kind of why we published the model. So most importantly, right, other than say, well, you're better than other model or something. So to have such a model within the company, it basically changed the pattern, how we make products at Page. Right? So previously, the traditional way you can see here is like we have a team, have this little data set curated, for one tissue and for one application. And then, at the time when I joined the company, there were like a per se team, there's a breast team, there's a colon team, and there's another one of the bladder team, right? So whenever you have another tissue of interest, you form a team, and within the team, basically people are doing pretty much the same thing. But the little bit of difference in how they curate the labels, how they curate the data splits, but pretty much the same, right? So, and when you want to work on, let's say, a biomarker, let's say we want to work on a breast biomarker, like we're showing here, right? HER2 complete. So your best bet is basically, you have a tissue-specific model that's already done, and you can use the embeddings from there and hope for the best. So, but with the foundation model in hand, basically that changed us how we think, right? So now we have kind of one team working on foundation model only and try to iterate bigger and better algorithms there. And there are a few smaller teams basically to use the foundation model for different downstream tasks. So that basically changed the mindset, right? And redefined basically what we can do. So this is also to show basically after you have a foundation model, how the product portfolio could change, right? So now we have a new blue box here. So that's basically everything pan cancer and pan tissue. That means like one model, one product for all the tissues. And we have like pan cancer detection, classification, even for the cell detection. We're gonna go into each one of them separately. So the most obvious one, right? So after you can do pan tissue foundation model is the pan cancer detection. So we made a initial product out of the foundation model to detect cancer like this everywhere. So basically the idea is like you take a whole slide in, and on select level, the model is gonna detect where the cancer is and point like what we're showing here, right? So where do we find the cancer? And it can give a very rough segmentation map to tell the pathologist, here are all the cancer foci that we found. And for that guy, basically, we try to benchmark it as well, across different tissues, across different cancer types. So, at the very beginning, before we have these numbers, right, and even some of our internal pathologists, they were pretty skeptical. Then, they say, well, the cancers are different, right? There are different tissues, they have different visual patterns, how do you merge them into same network? So basically, after we see this, and we go through a couple of examples, some of our
skeptical pathologists, they even become like the supporter of this, and they try to sell this idea to everywhere else. Basically, you can't. So you can detect all the cancers, right, altogether. So we try to also compare, the cancer types, let's say, between the common ones and the rare ones. So where this model compared to the other embeddings and so on. So another thing, we found interesting is like how do you compare it on the scale, right? So we have a few baselines that's relatively smaller models, and you see the difference, let's say, regarding detecting cancers, right? So basically there's difference, but that's like not like magically different. So it's basically we are almost 10 times larger than many models, but it is better, right? But when you see the model size on the x-axis and the data sets on the x-axis here, right? So you can see the change factor is like a log scale, right? So it's like after you have like 10 times difference in the scale, you can see some difference, right, in cancer detection accuracy. But I think, in such a critical task, right, a critical application, 1%, that still really matters for that 1% of the patients. So another thing we found interesting is that, we try to also use this universal model to compare with our clinical models, right? The expert cancer detectors, let's say page prostate, page breast detect, and page lymph node detect, right? So see, okay, so how does it compare to the specialists? So sorry about the small fonts here, right? But the idea is basically the conclusion is overall, so they're very much on par. But when you dig down into the rare variants, even in the tissue types who are very common, in the clinical workflow, there's a difference. Basically, the overall pan tissue detection model can work better, on the rare variants. Another interesting thing is like for, let's say, the cell detection, the cell segmentation, right? So now a patient we also can do it across different tissues easily. So you don't need to prioritize what tissue you work on, what tissue you don't. Right? When we annotate everything, let's say for cell level detection, you can annotate from all the tissues. That's our plan now. And given that, right, so you can right away work on something like, let's say, pan tissue tumor quantification for microdissection, and so on and so forth, right? So I'm trying to show some examples here. The last example is like basically biomarker prediction. So previously, how we do biomarker prediction is like, we need to prioritize, let's say, on a specific tissue, there's a specific genetic mutation, plus probably even perturbation, right? So that's going to be important. There might be a business case for that. So we try to prioritize that, form a team, train a model, and now it's kind of the mindset that we changed. So now what we're trying to do is like to use the same model for all the tissues and all the genetic mutations all together, and it actually works pretty well. So good news, basically the first version of our Virchow model has been open sourced for non-commercial use. So for the researcher community, we try to see also benchmarks for everywhere else. And we have to accompany this open source model. So we also have a small code base, right? So just to train aggregator on top of it, and try to develop this all together with the community. At the same time, so we have a even bigger model, as I said before. So try to also include HC slides, try to double the size of the training set, and also different magnifications and so on. So we're trying to also come up with the technical reports, to summarize all the differences here. One another stream of the work, that we started not very long ago is, if you think about, what we showed before, right, our foundation model for digital pathology should be, right? So if it's only tile encoders, the job is not done, right? So there's most of the tasks that we deal with here at slide level. So we try to also train the aggregator network, right? So there's like one network that can aggregate all the embeddings from the whole slide when you try to pre-train it with something. So the first step, like what we took here is to try to use COGA network framework to predict the diagnostic report that's associated to the whole slides, and at the same time also do the pairing job, right? So you try to pair also the slide with the right report. So the report here contains both the diagnostic information and also the molecular testing results whenever it's ready, right? Whenever that's available. So after training this network, basically you can use this network to reproduce the report, right? So in text. But at the same time, there are other two bonus products they can have. Number one is you're going to get like a slide level embedding that can be used right away, with linear probing and with nearest neighbor methods. And there's also a pre-trained network that you can use to fine-tune on the other slide level applications. So here's some more detailed plot like of how the network looks like, right? So basically we have still a slide encoder here, and I try to get like a slide level aggregated classification token, right? And that's here basically we reuse a pre-trained language model. It could be any large language model. So basically for this first work, we use BioGPT, consider many different factors, right? First of all, legal. So it is legal for us to use it. And it's already pre-trained with a pretty good vocabulary to cover the pathology terminologies. To give a few examples, basically what the model is capable of, right? So we went through a lot of those generated reports, so-called reports. So it's more like a whole slide caption, coming off this model with our pathologists. So at the top here, that's like the model outputs, and down there, so that's basically a description written by our pathologists to analyze basically where the model does well and where it does wrong. So in this part of the plot, basically we try to crop a region from the whole slide. So the model takes the whole slide as the input, right? But here, basically, we try to make sure the pathologist can see the patterns, right? We're not just seeing a thumbnail to make sure, you can make sense, what the model is talking about. So basically for this particular case, right, the model figured out, well, this slide has invasive lobular carcinoma in the left breast, right, ER and PR positive, right? It does basically get the detection of cancer right. It does get the subtype even pretty right. It hallucinated a little bit, let's say left breast, right? So the pathology image is pretty much impossible for you to just tell it from the image. So we know, well, there's still hallucination problem there. And for biomarker, let's say IHC biomarker prediction, it got ER and PR positive, but if you look at the ground truth here, so basically it got half of it right. So it's not perfect, right? So it got, the hallucination and plus some false predictions still. Another example, basically similar thing. So right subtyping, right detection. So it tries to say, well, the tumor is well differentiated where it's not, right? So it's like in the ground truth, it says moderately differentiated. So the grading was wrong. But overall, right? So that's something when we show the pathologists, basically blow their mind. So they didn't think that could work this well, but there's definitely a lot of room for the next version, which we're working on right now as well. Here's a quick visualization of the slide level embeddings. That we try to plot basically the clustering method, the results, of the slide level embeddings to try to see if they actually make sense, right? So we try to see it, in different data sets to see if you plot a cohort with different cancer subtypes, right, what they look like. So basically you can see there's a separation between different cancer subtypes or benign versus cancer tissues there. So that gives us the faith basically it's not learning something totally random. Yeah, so we try to also benchmark this, on different sizes of data sets, also different tasks. For example, cancer subtyping, also the pan-cancer detection task that we talked about before, and try to see basically how does it compare with, let's say, just training an aggregator, right? So without pre-training, with whatever language, right? We just train an aggregator, that's the baseline. Then we try to see, how does it work with zero-shot classification? How does it work, without fine-tuning the weights? You just take the slide embedding and then do linear probing on top of it, right? And how does it work if you just fine-tune it? I think eventually the message what we got from this little table is, so whatever you do there, so using this pre-trained aggregator actually always outperformed training from scratch. And then, so when you have a lot of data, right? So on the right side of the table, so that data set basically we have a lot of cancer detection ground truth in-house. then fine-tuning the select level foundation model actually make everything better. And on the left, basically, when there's smaller data, you try not to fine-tune the model. So that's kind of matching our expected conclusion, right? So that's another thing, we like to use this model for is to apply it on some of the biomarker predictions and try to constrain how much data you send to it, right? So let's say we have 1,000 cases for that biomarker, and we try to send like 10% and another 10% iteratively, right? And see basically with and without this select level foundation model initialization, basically what's the trajectory of the AUC on the test set, right? So the blue curves here, they're basically the trajectory for the scratch aggregator network, and the pink curve on the top is basically what you have with the pre-trained network, right? So the message here is also interesting, right? So that's basically for different biomarkers, right? It does have very different gaps, and for some of them, let's say CDH1, so you can actually achieve something like even better than 100% of the training data when only 10% of the training data sent into the select level foundation model, and right away beat what the best that you see before. So some of the other ones, let's say column SI, that we tried before, so that's the gap smaller, but most of them you see a difference there, so that also gives us a faith basically when we actually train, pre-train the select model when some of the genetic ground truth in, right? So does it actually make things even better, right? Do we see even bigger gaps here? So what's next, right? So I think, so far, with our team here, so what we're trying to do is to kind of move one way forward, let's say to make another version of PRISM model and try to make this adaptation a lot smaller in a way, right? So so we try to build a lot more capabilities right away in the same model and see how it works. So that's kind of the general thought here. And there are also, we encourage everyone to check out some other amazing work in the field. So basically for other select level foundation models, so the tau level foundation models, there are many encouraging works there, which also inspired us, right? That's the end of my presentation. So thank you so much, everyone. So a huge shout out to our colleagues at Page AI, also Microsoft and MSKCC. All right, thank you so much.
Presentation. Any questions from you?
Hi, thank you for a good talk. My name is Aryan. I'm from USFDA. If you could take us back to slide 18. I appreciate it. So you described a foundation model which is trained on millions of photoslide images. And you said it's going to replace each individual groups that you were dealing before across different tissue types. What I can see here is that your foundation model is used to extract better representative features, but you're still going to retrain or fine-tune a specific model for a specific target. Is that my understanding? Just a clarification, please.
Right, that's a very good question. Actually, it's just when do you fine-tune, right? So now, conceptually, I think there are, as you saw, so there are two types of foundation models we're talking about here. So for the tile level self-supervised learning foundation model, we almost never fine-tune. So that's kind of the goal. We don't want to fine-tune that model because it's so expensive. So far, we never needed to fine-tune it. So for the select level foundation models, as we saw before, so basically, when you don't have a lot of data, we also don't fine-tune it. So proof by numbers, right? So not fine-tune it, meaning just take the embedding for the whole slide and do linear prodding, and it actually outperformed the fine-tuning. And so when we have a lot of data, right, so then fine-tune that piece of the network only. But eventually, I think the goal and the mega trend also across the community is like, when the foundation model is like built with more data and also more capability, we kind of reduce the need for fine-tune. Yeah, so that's kind of the North Star, we're trying to head to at this moment. Not finally solved yet.
So, like all foundation models, they pretend they're explainable, but they're not explainable without autoregressive. So we know they give you better features, but sometimes you have a rare case, and they'll still say something that comes. Right? So do you have this problem, and how do you address it?
I think we're mostly addressing, let's say, the second part of the foundation model, right? So let's go probably here. Yeah, it does hallucinate. This is something, we're trying to solve with our colleagues right now, so the second version of it. So basically, number one is we try to not to predict everything all in once. Let's say just slide here, here's our model, and then speed up everything you saw. That's basically when we see the hallucination. So basically, we try to constrain the prediction in the second version there. So we try to say, well, don't tell me right away everything. You try to basically tell me what we ask for. And then we try to also benchmark. So the foundation model responds, according to that prompt, let's say. So then you have a better idea under each condition, what's the AUC for that foundation model's response. So they were kind of trying to go there. But in the short term, hallucination is there, so that you're right.
Do you use that in the for or area for foundation? What kind of foundation model do you use?
What kind of foundation model we use?
Is it like that would be for the latest, or you found the results you want to produce?
That's a really good question. So here we try to use a pre-trained language model here. So that's kind of a relatively smaller one. We use a BioGPT here. Yeah, so it's not GPT-4 level yet, but I think for next one, the ambition is we want to use something, large model levels of pre-trained as well. Yeah.
So, using self-supervised objective of the whole slide up, because it looks like I'm using language to supervise the whole slide already, but have you experimented at all using potentially a version of self-supervised?
We haven't, but there have been people who tried. I know Microsoft colleagues, they tried, right? I see some of them there. So basically, Gigapath, they tried it. So we haven't, basically. I think that's a good idea. So we haven't tried. So far, our path was like, try to use self-supervised learning at file level because we don't have ground truth there. So you can annotate, but, we cannot match something like, let's say, 3 million slides and every pixel we have annotations, right? So that's kind of the motivation. And for host line for training, basically we have ground truth. So that's the motivation why we try not to do self-supervised learning there. So for host line data, right, we have the diagnostic information there. We also have some of them, they do have even further tests like NGS tests and IHC tests, or some of them even have, let's say, the drug responses. So we try to use all this, right away, for the host line foundation model training. But whether, let's say, at another stage before that, would that help? I think we haven't tried, but we need to see the numbers. Let's say somebody try it and then not trying it, right? So then you see the numbers on a wide range of benchmarks. So we're also very curious to see. We're gonna see. Yeah, that's a really good question. Hey.
It's very great talk. Congratulations. My question is related to the data. Since we need a lot of data to train a foundation model, however, there's a lot of public data set like TCGA or like some other group use Twitter data or capture all of the image figure from the PubMed or publication to train the model. But my question is that all of that kind of data is kind of like not very clean. So you need to do a lot of like data cleaning or like QA. So my question is that what do you think the quality of the data to train a foundation model, and what's the balance between the number of the data and the quality of it?
I think short answer from me is that they both really matter, number one. And back to, let's say, the data source, right? So there are constraints. Let's say companies like us, we cannot use, let's say, PubMed data, right? We cannot use some of the data that we, let's say, got from Twitter, right? So that's there's a constraint. So we try to use our in-house data sets for that. But so far what we saw is, let's say, answer your question right away. Data curation versus, let's say, algorithms, also, let's say, the scale of the data set itself, right? So we have been really focusing on so far the scaling up data set size. So we haven't really reached even all the data sets that we actually have in-house. So that's kind of the first step. So we try to do that. And later, there's a potential step basically to see, well, that's kind of saturated, right? So we've sent, let's say, 6 million slides, and now we don't see any jumps in the benchmarks. I think that's kind of the thought, after that, let's come back to curate the data. Let's say there have been a lot of methods, let's say even from, let's say, the original DynamV2 team. So they try to propose something like hierarchical k-means cluster, right? So let's say you try to do a lot of k-means to find the representative tiles out of a very big data set and try to send only the representative tiles through foundation model training. I think that's a really good idea. We haven't tried it, so we don't know whether that's important here or not. Again, so we need to see the numbers. I think that's definitely on the roadmap. Yeah. Thank you so much for the question. Well, I'm very popular here.
So you talked about the report generation. I was curious, as you try to get this in the real world and are looking forward into building this text report generation into a product, and you're trying to deal with hallucinations and also possibly just like missing results of it's just not saying something that's important to say. How are you thinking about evaluating it or proving that it's correct, or given that there's all this flexibility in the output types and all that?
That's a really good question. I think we kind of reached that point for an earlier question as well. So that's basically number one. I don't think this current model is ready for product. So that's definitely not. So we have like actually interested customers already talking to us. That's, you know, when you guys are gonna let us try this as a product, we tell them it's not ready. You can see it's hallucinating and stuff, right? But it kind of gives us the face basically to know what's possible. And to answer that question, when that's going to be ready, right? So number one, I think such models, they need to be able to be prompted, number one. So that's basically the model need to know the question you're asking it for, and then you know basically what the answer is for. And then after that, you know, one thing really important is like you're going to be able to benchmark each specific task. You know, let's say cancer detection, you know, from this model, right? And if you just benchmark the cancer detection for a particular set of tissues, and you know the AUC coming out of those, you know, when it's in a constrained, promptable environment, right? Then you can claim basically that's a clinical product. So that's basically what I think is probably the two missing puzzles here. So one is basically we need to make it promptable, so you know what the model is really replying you for. And second of all, you need to know the response performance. So what's the accuracy, right? So for clinical grade AI, so normally there's a bar there, right? So let's say cancer detection, you need to reach certain sensitivity and certain specificity. Then we know this is not too much worse than human beings, and people can trust you. So that's basically two bars we're trying to reach, you know, with the next version. Yeah, but also we do welcome all the smart minds here to join force. You know, if you have an idea how to do that, please, you know, let us know. Yeah. Thank you. I think there was another one.
Last one. Other image information of the same patient. So I was wondering how the reports data of your data set like. Does it have the same problem of CPG data set, or it is strictly paired to the image?
I think we don't, that's a very good question. So the question is basically how well we can pair our image data to the reports data. Also, what's the noise ratio in it, right? So the reports that we have is totally clinical reports from the real clinics. So basically signed out by the pathologist, and we take it out as is. And we try to do a little bit pre-processing using large language model to filter out the irrelevant things out. But eventually that's, you know, basically what's happening out there. So is there mistakes? I think so. We hope there's not too big a mistake. Otherwise, every mistake, it means there's a cancer misdiagnosed. But, you know, pairing the data, that's something I think we have solved like a long time ago. So basically we can match the right report to the right patient. But then the question, as I said, you know, is there diagnostic errors out there? Then it means that, you know, the quantum of a single network, there must be some. But I believe, you know, it shouldn't be too bad. Yeah, that I also, you know, I also have the philosophy, we tried the best you can. That's kind of the best we can. So that's clinical reports. You don't need to do any extra effort to have them, and try to make the best use of them. Yeah. Thank you so much for the question. Okay, thank you.
A lot of interest. Thank you so much.
All right, thanks for the time. All right, thank you guys.
will be staying a little bit, Siqi. So there may be more questions if you are going to stay for a little bit.
Yeah, I'm gonna be here.
Okay, sounds great. Yeah, so you can still ask him during the coffee break. So upcoming, we have 4 more talks from accepted papers and also an invited talk. So Steve, you can, so next one is a talk from Clemson University. Would the speaker be in the room or via Zoom? Oh, okay, great, yeah.
Oh,
So you
perfect.
can come set up. Well, I'll help you set up and Steve can.
Yeah, so
Thank you so much.
please, my name is Steve Finkbeiner. I'm one of the co-organizers with May and others. And so I'm pleased to enter this section on accepted papers, and this first paper is going to be unsupervised microscopy video denoising. Are you married? Yes. All right, got the right speaker. Mary Diet Gigabo from Clemson University.
Are you shared on Zoom? Let me stop sharing and then you want to share. Are you logged in?
Yeah, and if there are any other accepted papers in this next session, please log into the Zoom that's part of the meeting. You need your 8-digit code to be able to do that, but we'll be presenting through the Zoom so
Yeah.
people at home can see.
Yeah, because there are like 30 participants in the Zoom room.
Yeah.
Actually, while Mary is setting up, Siqi, are you still in the room? Okay, not. There's a question in the Zoom room that I failed to bring up. I'm sorry about that. Oh, what happened? Oh, I see. People want the camera to the presenter. Okay, that's fine. One way is to, did you send me your slides? Yes. Oh, okay, you're there. Okay, good. Yeah. Otherwise, I can share it for you. Also, people want to see the presenter instead of the room, so I can change it. They want to see you. Okay. So that they see you from here. That's good. Yeah, it's coming out. It's coming. So it's good. Yeah.
Hey, hi everyone. Sorry about the short delay. My name is Mary, and thank you for having me for this talk. Today I'll be talking on unsupervised microscopic video denoising. This is a collaborative work with the Medical University of South Carolina. So why do we need denoising? Why is denoising microscopy imaging important? And one of the major reasons is because of how these images are captured. Microscopic images are captured mostly at fixed location, and this actually limits the field of view to a specific area. And this can, it necessitates longer light exposure, and this can also introduce what we know as persistent noise pattern. and which can actually obscure some of the biological signals that are needed for analysis. And also noises from microscopy images, they arise from different sources. We have electronic noise from the detector itself. We also have photon shot noise from low light, and some also environmental factors like vibration and then thermal fluctuation. Some microscopic videos also have issues with flickering, motion blur, and one major thing about microscopic images is the lack of ground truth. So which means that we can't use supervised method to denoise this. It has to be unsupervised method. So existing approaches uses blind spot techniques to denoise these microscopic images. And what this means is the exact frame of the video, or let's say the pixel that is being denoised, is omitted from the model input. And then the neighboring pixels are used, or neighboring frames are used to interpolate or estimate the denoised frame. One challenge is that these neighboring frames are extensively exploited. And this, because some of the noise are temporarily correlated, and what that means is if there are like temporary correlated noise across the frames, extensive reliance on neighboring frames can actually cause the algorithm to learn the noise in these frames. So it can lead to overfitting or learning even the noise pattern in the frame. And also some of these approaches for denoising, many video denoising approaches are actually used for Gaussian noise denoising. And like I mentioned earlier, for microscopic images, they're actually characterized by different noise types, photon and so many noise types. So hence the reason why we're working on this particular one, to be able to remove noise in microscopic images. And so our approach we're taking 7 frames as input to our model, and the goal is to denoise the middle frame, which is highlighted in red. And so first stage is it goes through what we call the feature generator. And in this stage, we use the depth-wise convolution. And the idea of depth-wise convolution is to be able to extract the spatial feature of each frame individually without having to extract along the channels of the frames. And once this is applied, we introduce a very simple approach here, which is what we call the deep temporal interpolation. And what this does is to mitigate the issues with over-reliance on neighboring frames. We have a linear filter here. And what this filter does is it applies a weight of 0 to the middle frame that we want to denoise, and also diminishing weight to all the frames that are closer to the neighboring frames. So we're still using the neighboring frames, but in this case, we reduce the effect of very close neighboring frames on our denoising effort. And once that is done, once we apply the linear filter into the feature map generated in the feature generated, we have what we call the weighted feature map. And this particular weighted feature map, as you can see, the middle frame is the value, the intensity value has been changed, and then it extends along the edge. So we have full inference from far away frames compared to frames that are closer to the neighboring frames. And then this, the output of the deep interpolation filter is concatenated and passed to the denoiser. And the reason why we concatenate this is so that we can also use for temporal consistency as well. It's important to also exploit some information, but not too much information such that it's going to lead to overfitting. And this is passed to our denoiser. Our denoiser is a very simple unit. It's a 5 layers unit architecture, and here we produce the denoised output of the central frame. And this is one of the data sets that was used. Here we used a simulated 2-photon caption imaging using the Naomi simulator. And the idea of using simulator is so as to be able to compare the results with the clean ground truths, just to validate some of the models. You can see that a lot of models, some of the models, like I mentioned earlier, actually overfit to the noise present in the frames. And compared to what we have here, our model is able to remove the noise while still retaining most of those signals that are very critical. And another data set we used here is one photon cation imaging. This image was actually locally collected at the Medical University of South Carolina, and you can see that the result is actually the noise is removed, and then even the important signals that are obscured in the noisy images are actually retained in the output. And here it's also to show another result on the fluorescent microscopy datasets. And just like the other ones, you can see that the result is relatively good. It's able to remove the noise, and we can see the signals clearly as well. Here we show the quantitative result of using the Naomi dataset, which has like a clean ground with disseminated datasets. We can see that the model actually performs very well in the PSNR, and the SSIF value of the model is very good. And then the other one is showing like example traces of a particular ROI region from the dataset. We can see how noisy the traces are in the noisy input, but compared to the noisy input, it's actually smoothing out and it removes any unnecessary spikes in the data set. And here is also to just show that even beyond microscopy images, this particular approach can also be used for any color images. This is a data set on this live YouTube data set. It's also able to remove noise at it's also able to remove different noise types and even higher intensity noise as well. Not only for Gaussian noise, it can also be applied to any other noise in RGB color images. To also further evaluate the contribution of our model, we also performed some ablation studies. And this is to show the effect of the temporal filter that we used in the model. You can see that the filter actually played a significant role in the noise in the models, the images. And also the other one shows that while we eventually chose the 7 frames, after performing several ablation studies, the 7 frames as input give us the optimal result. And so in conclusion, we presented a novel unsupervised noising method utilizing our deep temporal interpolation, which successfully removes noise, while still preserving crucial signal details in the data. And our approach also demonstrates strong generalization across different medical imaging data sets, and it can also be applied to any color images. In future, we aim to adapt. Currently, we use linear filter. In the future, we aim to dynamically adjust the filter so that the shape of the filter can be changed based on the observed motion in the data set. This is the team that made the work happen. Thank you for listening. You can visit our project page for codes and video. Thank you.
Any questions? Hey, come on up. Yes.
Great talk. Thank you. I was wondering if you tried other denoising architectures than a unit.
We tried not the complicated ones, because the major motivation for this work is how can we simplify denoising, because when you look at most denoising algorithms, they are very large. You see some using especially transformer models, they are really large. So how do we simplify that effect? That's the idea of using the small units, which is just 5 layers. Though we tried some convolution and so forth, unit is able to give us better results because of the skip connection that unit has. So it can retain those important, some signals in HP imaging can be very small. So you can retain those using the idea of the skip connection in unit.
Great, our next accepted paper is entitled Noise, Nuclear-Aware Osteoclast Instance Segmentation for Mouse to Human Domain Transfer. And it's going to be presented by Michael Wan from Northeastern University.
Yeah. Mary, you need to stop sharing. Oh, this thing. You need to share it also.
Yes. So.
Okay.
Okay.
Your presentation note is on the screen. Oh, that's.
I think that was for the last.
Last one. Okay.
Because it looks like it's not.
Yeah.
It's okay. Okay, cool. So, hey everyone, I'm Michael Wan. I'm a research scientist at Northeastern University. And thanks to the organizers for putting this workshop together. So I'm going to talk about osteoclast instance segmentation for, especially in the context of mouse to human domain transfer. This work is led by my master's students, including Sai Kumar Reddy, who's here and will be presenting the poster session, and also in collaboration with my biomedical collaborators at Maine Health and at Tufts University. So we're interested in osteoporosis and osteoclasts. So osteoclasts are specific kinds of multi-nucleated bone breaker cells. So they break down bone. And normally in a healthy person, bone is constantly being remodeled and mediated by osteoclasts, which destroy bone, and osteoblasts, which create it. But in osteoporosis, that balance is kind of affected, and it leads to low bone mass and deterioration of bone tissue, and that's a serious issue, and it leads to it causes high risk of fractures, and it affects hundreds of millions of people, and particularly older people, older women. So you can see how the high fracture risk would be a serious problem. And the way that people analyze, including my collaborators, analyze potential treatments for osteoporosis is to first apply the treatment to a sample of osteoclast cells on in vitro experiments and analyze the effects on osteoclasts in the regular microscopy. So here, this is sort of an illustration of an osteoclast. It's highlighted in the purple stain, and it has many nuclei, and that will sort of factor into our machine learning later. So currently, a lot of the work on this kind of research by bone and musculoskeletal researchers is done by hand, this image analysis. The task that they're trying to do is instance segmentation, osteoclast instance segmentation, so finding the locations and shapes of all of these specific cells. And they usually rely on a guided image processing procedure, which takes hours per cell, and even for a small translational study where they're just studying maybe 10 to 15 subjects, a treatment versus control, maybe a couple of replicates for their statistics, that can still take over 1,000 hours of analysis. And so the aim in my work is to alleviate this bottleneck in treatment research using computer vision. That's pretty straightforward, but one kind of feature of the problem, and I think this is common in a lot of translational research, is that there is a lot of data available for an animal model. So in this case, as is common, a mouse model, it's easy, mice are easy to work with and to control the genetics of and so on. So in this case, we have annotated data sets with 150 gigapixel images for mice, but maybe only 25 images for humans, and corresponding imbalance in the number of osteoclasts. And so we're interested in kind of this domain transfer. Maybe we have enough data to train properly on the mouse data, but not enough on the human data. So that's kind of a big, and there's also kind of a lot of variation in how the slides are presented. These are relatively small kind of in-house lab experiments. So this is a natural problem for machine learning, of course, but actually a lot of the work that has been done before has been relatively small scale. So kind of the problems that we're trying to fix are that many of these methods aren't fully automated, or they only consider detection or overall segmentation and not segmentation of the individual osteoclasts. A lot of them don't think about human cells. They only work with mouse data because that's what's available, or they work with specific fluorescent stains. And I think a lot of the work doesn't come from kind of the machine learning world. So a lot of the data sets and code is not made publicly available. So we're trying to do kind of like the first basic kind of deep learning end-to-end solution for this problem, especially in the human domain, where the translational applications are the most important. Okay, so this is just a recap of the task we're trying to solve: osteoclast instance segmentation, trying to find the locations and shapes of the cells, these specific multinucleated cells. And we're interested about the domain transfer problem, starting from the mouse domain and applying this to the human domain. And one of the techniques we found was effective in helping with this domain transfer is infusing this nuclei information about the nuclei, because osteoclasts are cells with multiple nuclei. So specifically infusing the locations of the nuclei in our model, similar to actually a paper that was discussed before lunch. So let me dig into that method, our kind of weak supervision method using nuclei information. So again, osteoclasts are multinucleated cells, and the idea is to guide our learning with this nuclei information. Now, there's so many nuclei, it's impossible to annotate these precisely. That would kind of defeat the purpose. So what we have, and also existing nuclei detection networks don't work that well for some reason in this context, because I think our nuclei look different from a lot of our stain looks different from the H&E stain and other stains that are common. There's a specific stain that people are using in osteoclast research. So we developed our own kind of small data set, kind of not expertly annotated. So like, unlike our osteoclast data, this is kind of informal, and we actually use it to develop a nuclei object detection and apply that and get kind of a larger nuclei data set across the images. So this gives us kind of like a weak data set of nuclei. The information is not, it comes from machine learning, it's not validated, and the annotations were originally not from cytological experts, right? So our goal is kind of to use this weak data in guiding our machine learning. And you can see the outcome of the predictions on the far right. They're approximately there, but they're not really paying that much attention to the precise size. And again, we don't have kind of the expert validation. Okay, so the way that we incorporate this kind of information weekly is to train in 2 stages. So in our first stage, so we're just using kind of a generic object detection kind of network. So in this case, YOLO V8. And we train our network, or pre-train our network on detection of both the osteoclasts, which is what we care about, and the nuclei, which is kind of the auxiliary information. This is not fully reliable, right? So then we do a second stage where we fine-tune on the task that we ultimately care about, which is the instance segmentation, so figuring out the shapes. And then we sort of ignore the nuclei at the second stage. So hopefully the model has learned a little bit about the nuclei information, but now it has a chance to kind of forget anything that is harmful or mistaken. So this is the paradigm that we found works. I'm probably other paradigms, other machine learning methods would work too. But and just to note, all this is training just on the mouse data, because again, we don't have very much of the human data to train with. And so to highlight the results, these are kind of instance segmentation results at different thresholds. But let me just focus in on kind of the standard medium threshold. This is on the top we have, so both of these are training on mouse data and applying it to the human domain where we want our algorithm to work. And the top model is kind of the default training without our nuclei awareness. And the bottom, the second one is with nuclei awareness. And you can see in precision recall and mean average precision, it actually boosts the performance quite a bit. Okay, so it's even a bit surprising. But I think probably because there's really not that much. We only have 2,000 human labels. We're not training at all on human labels. So I think using any kind of extra information, cytological, cellular information really helps. And then also comparing, if we do train with about half of the human data, comparing the baseline model and using our nuclei awareness, it does still boost the model performance, not as dramatically as you would, as expected, but still meaningfully. And this is kind of a visual kind of illustration of that. If you compare the models on the left, the two left models, one is without the nuclei and the noise model is with the nuclei information. So it picks up a lot more of the osteoclasts. And, so we put together a script for doing the inference on the whole slide. And I'll just mention something we're interested in pursuing next is trying to, now that we have the morphologies, trying to maybe use some unsupervised learning or some of the methods people have been talking about to analyze the morphologies and, tie them in with function and genomic information. So I think that's it. Thanks so much.
Any questions? Great. Well, thank you, Michael, very much.
Thank you.
Is our next presenter in the room or online? Oh, great. Thank you. Terrific.
Great, so I thought I'd introduce our next speaker. I think this is Jason. Yeah. All right. From Stony Brook University. And the title of this paper is Uncertainty Estimation for Tumor Prediction with Unlabeled Data.
Okay, hello everyone. This is Juyang Yuan from Stony Brook University. I would like to present our research paper called Uncertainty Estimation for Tumor Prediction with Unlabeled Data. There are digital pathologies has been developed a lot due to the advances of the deep learning technologies. However, there are several challenges in digital pathologies. First one is reliability and transparency. The second one is limited label data. Because labeling is the labor-intensive task, so it's sometimes error-prone results in overfitting, and it sometimes results in wrong decision about prediction. So utilizing unlabeled data is very important, but it's still challenges in utilizing unlabeled data. So how can we check the reliability of prediction? We use the uncertainty estimation. It is estimating how confident a model is about its prediction. It is identifying the possibility of errors in prediction. So what is the uncertainty? Let's see the below submax probability. When we see the below bar, if the softmax probability is near to 0.1 or near to 1.0, we can call this probability is certain. However, if the softmax probability is near to 0.5, if the probability is near to 0.5, we call this uncertain. So we focus on the patch-wise predictions uncertainty estimation. The patch-wise tumor prediction is extracting patches from the whole slide images and predict each patches from the trained model. We get the binary mask and probability heat map from the predicted patches. When we see the probability heat map here, there are so many sky blue spots. This is the uncertain predict. But when we see the ground truth, this isn't tumor area. So this baseline model has some inaccurate uncertainty estimation. So our goal is improving uncertainty estimation for tumor prediction with unlabeled data. Our solution is utilizing the semi-supervised setting that leverages limited labeled data alongside unlabeled data by calibrating uncertainty during training patches. So we use the surrogate functions to the loss for calibrating uncertainty. For this, we use both correctness ranking loss and consistency ranking loss in least square. So what is the surrogate functions? there is some limitations in the cross-entropy loss, I mean the baseline. Cross-entropy loss is comparing the softmax output with just true label. It is focusing on the classification accuracy, not for the uncertainty of the prediction. So it does not reflect how often of the model is during training. So by introducing the surrogate function, which is the alternate method to estimate uncertainty of the prediction during the train, it is providing the approximate measures of how often and how uncertain predictions are. So it makes online softmax output with these estimated uncertainties. For label data, we use the correctness ranking loss, including a surrogate function called correctness. It is calibrating the model's uncertainty by comparing the correctness of the different labeled data points. This one is the correctness. Correctness is how often the correct prediction for each data point during the whole epoch. So this is about how to calculate the correctness. We predicted every data point for across every epoch and calculate how often the correct prediction for each data. Let's see this first data set. This is predicted correctly 3 times during the entire 6 epoch. So the correctness is just 3 divided by the whole epoch is equal to 0.5. So correctness is basically focusing on just checking the correct prediction for entire epoch. However, how about the unlabeled data? For unlabeled data, we use the correct consistency ranking loss with the surrogate function called consistency. This is calibrating the model uncertainty by aligning the uncertainty with consistency of prediction. Here, consistency is the surrogate function, which is how many times the same prediction is made across consecutive epochs. Look at these figures. Labeled and unlabeled data is predicted with models in every epoch. We don't need to compare the two labels, and we don't need the two labels about the data point. We just compare how many times the same prediction. Let's see this one. First data is predicted samely every epoch. This is the this consistency is 1. When the predicted is changed at 1 time, it's just 4 times of the same prediction. So consistency is 4 divided by the epoch minus 1. So our solution is for the total loss. Total loss for our method for training is the cross entropy plus correctness ranking loss plus consistency ranking loss for training. After training the model, we extract the patches and predict every patches and make the probability heat map. These are our experimental setting. To compare our performance with other models, we use the baseline and MC dropout with the 50 samples and correctness ranking loss. We have the 2 types of the data set. First one is the SEAL, which is the breast tumor patches data set. We have 2 types of the SEAL data. One is the SEAL 10, and the other one is the SEAL 20. Both are having their own labeled data for training the consistency, like training the model. TCGA is the first slice of breast cancer image data set. This is very high resolution for our main task. This one is the experimental result. The top table is about the validation result of the SEAL data set for each method. At this point, our experiment result with correctness and consistency ranking loss has improved compared to other methods. When we see the down table, this is about the TCGA host line images, which is the patchwise tumor prediction results. When we see these results, the correctness and consistency, the combination of the correctness and consistency is the better performance in terms of the every matrix. F1 is a high value of the F1 is better performance. The other uncertainty matrix, low one is better performance. So according to the validation and test results, our trainer is better, has the better performance about F1 and uncertainty. This is one of the qualitative results for our method. When we see the red circle and magnified, these are the fat area, which is non-tumor area, but baseline has a lot of uncertain areas, which is sky blue or green. And MC dropout and correctness ranking loss has also uncertain area of this area, which is supposed to be non-tumor. However, correctness and consistency has a clear uncertain area of this part. The other qualitative measures, when we see this square having the stroma across the tissues, this is supposed to be uncertain because of the stroma across the tumors. However, the baseline and MC dropout and correctness ranking loss has some noise uncertain and uncertain areas. However, our correctness and consistency show the uncertain area of this stroma. According to this qualitative result, our correctness and consistency exam shows that better uncertainty estimations. So by utilizing the surrogate function to improve uncertainty, we can improve uncertainty estimation for tumor prediction with unlabeled data. And we can effectively use the both labeled and unlabeled data for model training. And we finally improve the reliability in tumor detection, which is crucial for pathologists. Thank you very much. And if you have any question, please contact me or just please come to our poster session, which is somewhere in 365 to 380. Thank you very much.
Any questions? Great, thank you. Is the next speaker in the room or online? Oh, great. Wow, we lucked out. 4 for 4. Yeah.
We will use my laptop.
Okay, super.
I know.
Yeah,
Finally
this is
in
like
person.
a high point after the pandemic.
I know, it's nice.
Great. Well, I'm pleased to introduce our next speaker. Is this gang down? Yep. Excellent. From University of Washington, local. And the title of the talk is Triage of 3D Pathology Data via 2.5D Multiple Instance Learning to Guide Pathology Assessments.
Not sharing this. Not sharing this. Let's see here. How am I going to share this? Here. Share. We have your PowerPoint open, right?
This one.
This one?
Yes. Go to the first page.
Yeah. Full screen?
Yeah, go to the first page. Yeah.
Oh, yeah, right.
Thank you.
Full screen?
Yeah. All right.
Okay.
You're on.
Yeah, yeah. Make sure do we have everything right. Just a second. Yes.
Okay.
Okay.
Presentation note is shown on the screen.
That's a previous one. And you are, yeah, all good. And are people seeing the speaker?
Okay, good afternoon, everyone. My name is Gan Gao from University of Washington. Today, my talk is triage of 3D pathology data via 2.5D multiple instance learning to guide pathologist assessments. So to start with, cancer diagnosis via conventional 2D pathology where the biopsy is sectioned into a few thin slices, mounted onto glass slides and evaluated under a microscope by the pathologist. This process has many limitations, mainly first limited sampling, because one thin slice is usually 4 micron in thickness. It represents less than 1% of the tissue of a 1 millimeter thick biopsy. And the second, human tissue morphologies are essentially in 3D. 2D visualization can be ambiguous. And third, this process is destructive to valuable human tissue specimens. Therefore, over the years, our lab has been developing non-destructive 3D pathology workflow, where we stain the specimens with a fluorescent analog of H&E, optically clear it to make it transparent, and image it with open-top lead sheet microscopy in 3D, and generate this 3D pathology data set. And here is an example of a prostate biopsy. So the advantages of a 3D pathology workflow include comprehensive sampling, because we're imaging 100% of the specimen. And we can achieve 3D visualization of the tissue morphologies. And also this process is non-destructive to the tissue. Therefore, based on that, we are hypothesizing that comprehensive 3D imaging of spatially heterogeneous specimens offers the potential to improve diagnosis. Let me elaborate further on this point. So here is a 3D data set of prostate core needle biopsy. If we zoom into this small region and review the volume and the stack of 2D slices, and some depths, 100 micron and 200 micron, we can see high-risk morphologies pointed by the green arrow here associated with lethal prostate cancer. Oh, come back. All right. So in other depths, and 50 micron and 150 micron, those structures are not the same, and the tissue appears low risk. This further reinforces our point that arbitrary selection of 2D sections may fail to capture diagnostically important tissue morphologies. So therefore, to encourage early clinical adoption of 3D pathology, we are here using a human-in-the-loop strategy where we have pathologists to interpret 2D slices from 3D datasets, leveraging their expertise. However, 2D pathology images has already been large. As you can imagine, in the 3D dataset, there could be hundreds or even, what's happening? There could be hundreds or even thousands of 2D images. So it would be not practical for pathologists to review all slices manually. This process would be too time-consuming. Therefore, in this study, we are proposing AI triage pathology, where we have the AI algorithm sift through all slices within a 3D volume and identify the highest risk slices for manual review. And this process would be time efficient. In this study, we are targeting prostate cancer diagnosis problem. To train the model, we have a panel of pathologists to annotate selected 2D slices from 3D datasets from different patients. And we train the model. Once the model is trained, we would let it perform slice-by-slice inference on the 3D dataset, and we can generate a risk profile. And based on that, we can select the highest risk slices for pathologists review. As for the model architecture design, the most straightforward way is to use 2D multiple instance learning that has already been popular in computational pathology. In this process, the images are split into patches, encoded into patch features, and aggregated with attention-based network, where the patches are weighted by the attention score, indicating its importance to the final diagnosis, and we can aggregate the slice level feature for prediction. However, as a triage algorithm for 3D pathology data, this kind of 2D analysis is not ideal because 2D visualization of inherently 3D human tissue morphologies can be ambiguous. And this method is not able to harness contextual information along depth axis of the 3D pathology dataset. Therefore, in our study, we are basically exploring 2.5D multiple instance learning, where we aggregate contextual information from neighboring slices to produce a context-aware risk score for each slice. We have tried different strategies. So basically, the first here is naive pooling. Similar to 2D method, we convert the patches into patch features. But for the slice of interest, we want to make a prediction. We also encode its neighboring slices and get their patch features. And we have the attention module to aggregate all the patches from slice of interest and neighboring slices and generate a context-aware slice of interest feature for prediction. So the second strategy here we use called average pooling. Similar to the naive pooling strategy, we are encoding both slice of interest and neighboring slice patch features. But what is different that here we have two steps of aggregating contextual information. So the first step is attention-based intra-slice pooling, where we have the attention module to only aggregate patches within each slice and generate slice level features correspondingly. And the second stage, we aggregate neighboring slice level features into the slice of interest feature and generate the context-aware slice of interest feature. And in this method, we use a simple averaging. And the third method here, we use weighted averaging. So same as average pooling, we have two steps to aggregate neighboring slides, neighboring patches and slice of interest patches and generate slice level features. But what is different is that we assign different weights to different slice level features. And the rationale behind it is that for some slices, it may have more diagnostically important tissue morphologies. So we want to emphasize them by assigning a higher weight and make them contribute more to the context. And advantages of all the 2.5D analysis methods is that we could leverage contextual information along depth axis of the 3D dataset to improve predictions. So here comes the results. We benchmark our results on the images from 115 biopsies from 54 patients using the one-out cross-validation. The baseline here is 2D method, not considering any contextual information. We report AUC and F2. F2 here is similar to F1, but we emphasize more on recall because as the screening algorithm to detect higher grade prostate cancer in 3D data, we don't want it to miss any disease. With naive pooling, we see a big improvement in AUC, indicating the importance of contextual information. The F2 is on par with 2D method. It probably because the attention module is tending to too many patches, making the positive patches containing forces of cancer hard to be identified. But with two-step method where the attention module aggregate only patches within each slide, we see a clear improvement in both AUC and F2. And finally, with weighted averaging, the AUC is slightly better than average pooling, but F2 is better than average pooling by a clear margin, which shows the importance of weighting diagnostically important slices. And finally, we can conclude that integration of contextual information along depth axis enhances models' discriminative capabilities and identification higher grade prostate cancer. We deploy the trained model on the example 3D pathology data set to do slice by slice inference. We generate this kind of risk profile. And we ask pathologists review select levels from this data set. We see in the blue level, the diagnosis is low-grade prostate cancer, and purple is intermediate grade, and red here is high-grade prostate cancer, which shows the pathology diagnosis is broadly agrees with our predictions. Finally, the take-home message for my presentation is that we present a 2.5D multiple instance lineage triage pipeline to identify highest risk slices within 3D pathology dataset for improved diagnosis. Future work include generalized to other clinical applications and large-scale clinical validation, comparing standard care 2D histology versus rare triage 3D pathology. And I'd like to thank all authors contributing to this work, especially Dr. Andrew Song, the co-first author, and Dr. Jonathan Liu and Dr. Faisal Mahmood for their guidance. And we have a poster session. Welcome to check it out and talk with us. Thank you.
Thank you very much. Any questions? All right, if not, we'll move on.
Thanks.
We'll move on. Thank you very much.
We have Charlotte.
Is she online?
Yeah, Charlotte online. Charlotte, are you in the room, in the Zoom room?
Yes.
Yeah, sorry we're running so late, but you still definitely have your time. We'll have a bit of a buffering poster session.
Okay.
Can you share your slide? Let's stop sharing in the room. Can you share your slide, and then we can get started. Now we can have the camera looking at the audience, since she will be sharing her. And then other questions. I don't know his question. Okay, that's not.
Do you see the slides? And do you hear me?
We can do both.
Yes.
Okay, perfect.
All right.
Yeah.
So I'm very pleased to introduce our next speaker. She's an invited speaker, Dr. Charlotte Bunn. She's an assistant professor of computer science and life sciences at the EPFL School of Computer and Communication Sciences and School of Life Sciences. She previously was a postdoc at Genentech at Stanford University with Aviv Regev and Jura Leskovec. And the title of her entitled talk is Predicting Patient Treatment Outcomes Using Diffusion Generative Models. Thanks, Charlotte, for joining us.
Thanks a lot for having me. Yes, so I'm currently transitioning. I finished my PhD last year. I'm currently transitioning from a short postdoc into the tenure-track assistant professorship. So if anything of what I tell you today is interesting to you, then please reach out. We are just launching our lab. So we all know, I guess in particular people in this room, how complex biology actually is, and in particular that every one of us is really a unique and complex biological mosaic. And complexity here does not only arise through scale by the sheer amount of genes and cells that are interacting, but in particular that everything is very much a dynamic interplay of those biological processes happening on so many different scales. So in particular, the human body has 3.7 times 10 to the power of 13 different cells. And in those, every cell has around 20,000 genes. And every cell has, of course, the same set of RNA, but is then very, very different, in particular through very different rules that are then steering RNA production, protein production, and so on and so forth. And so we are what we have in the end is many, many different cell types, cell states that all execute very different functions. And one way of somewhat characterizing cells and cellular phenotypes is through, for example, a gene expression pattern, but there's many different phenotypic outputs we can measure and record. And what we see is that somewhat fine disturbances in this regulated system are often the root cause of disease. And this is, of course, a very crude approximation or generalization I'm making here, but somewhat we would like to understand what are the perturbations and disturbances that somewhat steer the system that is so well functioning into a system that, for example, leads to unrestricted cell growth and cancer and so on and so forth, and transforms a healthy cell state into a disease phenotype. And the running example I'm going to use throughout this talk is in particular cancer biology. And this has to do with clinical cohort study or results from a clinical cohort study we are currently running using the methods I'm going to introduce in this talk. And this is on melanoma. It's an observational clinical cohort study on melanoma patients. So what we usually have is a patient that comes into a clinic with a very particular, very unique disease phenotype. We are able to somewhat understand the molecular properties of the tumor, and now would like to understand what is the best treatment to choose such that the patient responds most effectively or like very efficiently to the treatment. So we would like to understand the treatment response of a patient and learn this using machine learning, because then the moment a new patient comes into the clinic, we can take knowledge we've gathered and now decide or make like a meaningful prediction of what could be the best drug for this new molecular profile we are dealing with. And so this is, of course, the root idea, the kind of the root of the personalized medicine, really looking at how can we provide targeted therapies tailored to a molecular profile of a patient. Molecular profile of a patient, what does it mean? I mean, we've seen a revolution in biotechnology. We are not only able to understand somewhat the tissue level and spatial architecture of a tumor, but we can really zoom in, and the data I'm going to work with mostly are imaging data or omics data that capture molecular profiles of individual cells. So genomic transcriptomics, and in particular proteomics, using very different types of multiplex imaging. What we are ending up with, though, is basically very high throughput data from thousands and thousands of cells. We are able to measure thousands and thousands of features. Just to give you an idea of the scale of the data the field has gathered and why this is going to be relevant in particular towards the end of my talk, is just the human cell atlas alone has more than 10 to the power of 8 profiled cells, and other data consortia have like 36, or depending on which pharmaceutical company you work in, you will have millions and millions of cellular profiles, very distinct tissues, diseases, and so on and so forth. The question is, though, what type of neural architectures do we need to design that really excel on this type of data? Because these data is very different from what we've seen. And in the last years, we, of course, everybody is aware, have seen a revolution and like really convincing success in NLP, computer vision, mainly due to the development of new architectural principles, specialized network architecture such as transformers or vision transformers, and very specific type of loss functions that somewhat capture the structure of the data, like token prediction, masking when it comes to imaging, and so on and so forth. The core question is, what are those principles that are present in biological data that allow us to, without annotation and labels, because this is something we have really sparsely provided within biology, what architectural principle we can find that really capture the structure and essence of those biological systems, these cellular phenotypes that tell us something about how a patient is going to respond to a treatment. And biology is much more complex than anything we've solved so far. And this is mainly due to many reasons. I'm going to highlight just a few that I find key. Everything we measure is very much indirect and obscured and noisy, right? So if I want to understand a cellular phenotype by the gene activity pattern, it's not that I'm measuring gene activity, but I'm always measuring a different biological entity that somewhat correlates or should correlate directly with what I'm interested in measuring. If I take several measurements, I always need to deal with experimental noise and batch effects, and they can be very, very strong, in particular when I have small patient cohorts, and I always have small patient cohorts. Biological data is very multimodal. There's many different types of measurements I can conduct, many different entities I can measure, and we somewhat need to integrate this. And there's, of course, more and more methods that allow us to have really paired samples. For example, Xenium that gives us H&E stains, as well as multiplex imaging, as well as spatial transcriptomics that are somewhat aligned. But still, the question is how to really integrate information across those layers. And then, as I mentioned already before, and this will be the core component of this talk, is biological systems are inherently dynamic and driven by countless interactions. And we somewhat need to capture those dynamic systems, even though they happen on so many different scales, like from femtoseconds to sometimes like hours or even an entire life. So problem when looking at these dynamical systems is that these measurement technologies are mostly destructive. Every time I measure a cell, we need to somehow fixate it, we need to open it, we need to somewhat destroy it in order to look inside and understand the molecular phenotype. But if I want to reconstruct a dynamic process over time, one core method is that I usually am able to trace my objects. For example, if I'm on the stock market, right, I see the same somewhat stock and how it developed over time. This is something in biology we cannot do. We cannot conduct somewhat live imaging. We can, but very sparsely. Most of the methods that are high throughput, they are destructive. And so this core principle of machine learning, of always having a paired somewhat what I'm giving and what I want to predict, for example, how the cell looks perturbed and how the cell looks unperturbed. This pairing I'm not even provided with. So from the root on, we need to think somewhat different. And thus, we are somewhat only provided with non-time resolved measurements of temporal processes. And this is a very particular challenge, and this is something we are going to address in this talk. So here, this is when I say we have unaligned data or snapshot data that somewhat describe still a dynamic process, but not in a time-resolved manner. So the question is, or the core principle in order to be able to understand how a patient is going to respond, so something we will explore at the very end of my talk, is the question on how can we actually in silico model cell behavior? How do we build machine learning models that are able to somewhat understand how a cellular phenotype measured through some omic technology, for example, or microscopy, the data we are going to use within this talk. Here is cell now lying in this very high dimensional, potentially high dimensional, basically feature space. How is the cell going to change from a disease state into a healthy state or vice versa? And we are not only interested in how they are somewhat, how the disease phenotype looks like, no, we are really interested in what are the molecular patterns and the molecular features that are going to change. And why this response pattern is so important, we will explore this in results I'm going to show you at the end of the talk that really provide us with insights from a clinical perspective, new clinical associations that are relevant to then understand what are the best drugs to choose, what are the type of drugs we should develop. And similarly, right, I would like to understand how would a cell then from a disease state look or respond to a particular drug I'm providing it. And again, we want to understand the response pattern. What are the features or what are the somewhat properties of the cellular phenotype that are going to change? We want to do this on the level of single cells because cellular populations are very, very heterogeneous. And this is particularly the case for cancer, right? So we know that cells respond very different to a drug and every patient has a very different set of subpopulations. Some might be resistant and others might not. And we want to really understand what are the subpopulations that are sensitive, which others do I need to basically target with a potential combination therapy. And last but not least, we need to somewhat learn from unaligned data, what I just mentioned on the previous slide. How do we do all this? I mean, biology can now benefit from somewhat core inventions of the machine learning community of the last years. And this is in particular generative modeling, where we see the evolution of a distribution over time. We have a very simple distribution on the one hand, a simple Gaussian, potentially also a condition. And by now we have built AI models that have the ability to really generate high quality samples, right? So I can give it a condition, a pope with a puffy jacket, and the results are very, very remarkable. So how can we make use of this? One of the classes we are going to discuss and the methods we've built are based on these diffusion models because they have several properties that might be very relevant or are very useful when looking at biological systems. Because they are based on this core idea of a system of stochastic differential equations. So on the one hand, the core principle, and I'm sure many of you in the room are aware of it, is that I have just a very simple SDE with a linear and non-learnable F. And I'm going to, like, over time, basically corrupt my image such that I end up with random noise. So if we go through this, it's basically we continuously add noise until I end at something that is completely Gaussian, basically resembles a Gaussian. Now I can invert this process. I can look at the reverse process, or the reverse stochastic process, that now would get me from noise back to this high quality sample that follows the data distribution. And so here, of course, the reverse SDE or backward SDE has this score function, but because I'm dealing here with a very nice setting, I have an easy to sample distribution, I have a non-learnable F and a tractable objective, I can parameterize the score function with a neural network and very efficiently learn to parameterize this. And I have a generative model that allows me to turn one distribution into another. We are, of course, not very interested in its simple image generation. We are interested in understanding how cellular phenotypes emerge. We also don't want to just generate new cellular phenotypes. We in particular care about how do unperturbed cellular phenotypes turn into their disease phenotype or respond to a drug, right? Now we have a slight issue because here on the right, we don't see a simple Gaussian anymore. So all the nice properties of score-based models are somewhat gone. We don't have an easy to sample distribution. A linear and non-learnable F is not going to describe our complex cellular dynamics over time. And I don't have a tractable objective. But there are some solutions, and I'm going to basically run over the entire, or like kind of skip the entire derivation. But I somewhat need to find a non-learnable, non-linear drift that describes both the dynamics from unperturbed to perturbed and perturbed back to unperturbed. I give you the solution, which is going to resemble something or is based on a theoretic concept known as Schrödinger's bridge, which is based on the famous physicist Erwin Schrödinger. We can plug this in, and I'm going to slightly basically plug this into the forward as well as the backward process, simplify this expression. What I'm provided with is a pair of two neural networks, a forward drift or a forward policy that gets me from unperturbed to perturbed, and a backward drift that gets me from perturbed to unperturbed. So we still have this somewhat framework that resembles diffusion models, just slightly more complicated because now I actually need to parameterize two neural networks. And this is quite challenging. It works, and many people have actually applied this directly to learn those image generation tasks, interpolate between distributions, or actually apply this to cellular dynamics. Among others, this has been also done in our previous work. But now I would like to provide you with one more somewhat derivation on how we can slightly transform this problem into something that is, again, easier to parameterize, where we don't need to basically simultaneously train an alternate optimization fashion two neural networks, the forward drift and the backward drift. And here we are again using insights obtained from the biotechnological community because there's constantly innovations happening on that end as well. So of course, everybody's well aware that it's a huge limitation that those omic technologies or also microscopic images are going to destroy cells by fixating or by opening them, right? And so there have been a couple of methods, one coming actually from EPFL and ETH, which has developed this live-seq methodology that only extracts a bit of cytoplasm so that the cell can still be recorded over time, but we can do this multiple times from the same cell. Similarly, and this is a method the data we are going to work with, we can tag cells with a genetic barcode. And this genetic barcode is a DNA barcode that is transferred to the next generation of cells. So the moment the cell divides, it's going to be split. So even though I'm using high throughput measurements that are going to destroy cells, if I find the same barcode again in a downstream population, at least I know clonally it must basically originate from the same, somehow from a similar cell, clonally related cell. And I call this partial alignments because these methods are either very low throughput, so they give us a partial alignment, at least a subset of the data we have an alignment for, which we can use to train this stochastic process, or they are very much affected by stochastic cell division and sampling. But still, we should make use of them. And so now the goal, again, as I said before, is to reconstruct the stochastic process that describes these cellular dynamics. And we do so by using Schrödinger bridges or these diffusion Schrödinger bridges and basically get to an objective that more or less resembles the score-based generative modeling objectives or flow matching objectives. You can give it many different type of names. So here's the same problem, just in a very different way of writing it down. But we are looking for a stochastic process that gets us from our starting population to our target population, from our unperturbed to our perturbed state, given some knowledge of a reference process. And we are going to use two ideas, or two sets of tools. On the one hand, something known as Dupes H transform, and then just classical Schrödinger bridge theory, in order to use these alignment or aligned data in order to derive a basically more efficient optimization framework for solving these problems. So we know already, right, that the solution of the Schrödinger bridge can be expressed as a system of SDEs. I showed you on the slide by basically making the connection of like score-based generative models to these diffusion Schrödinger bridges. Now not anymore with F, but we just have an unknown drift B that describes our cellular dynamics. And because now we have an SDE that is conditioned to start at a certain point, and we know where it ends, right? Because we have data that at least we know this is a clonally similar cell, we can expand this drift by Dupes H transform that takes care of this conditioning and have a new expression of our SDE that we need to parameterize, basically now parameterizing both terms. At the same time, though, we know the reference process, if it's conditioned to start at x0 and end at x1, we actually derive at an SDE that has a closed form expression for this drift. And now in order to learn the drift that describes our cellular dynamics, I just need to regress it to this drift that I can compute in somewhat for every point in time. And this is now a regression objective that very much resembles these like score matching objectives that have been very successfully employed in previous works. And now I'm ending up with a system that allows me to describe my cellular dynamics continuously in time, and really understand how, for example, a very undifferentiated population of cells results or transforms into a population of cells that contains very heterogeneous set of cell types. Basically, this is a differentiation process in hematopoiesis, and we are able to recover these emerging cell types over time. Then, of course, in particular, when we look at cancer, we have processes that are heavily governed by cell death and birth, meaning by proliferation events, and then the moment the drug is actually effective, of course, by heavy levels of induced apoptosis, cell death. And the question is, how can we model this using these diffusion frameworks? And so what we did in a recent work is that we derived the time reversal of those SDEs, this forward-backward SDE framework, that allows us to integrate somewhat processes of cell death and cell birth. And here now, cell death is modeled by just expanding our SDE with some form of killing rate, like a probability of a particle to die. And the core way of implementing something like this is to create a coffin state, like a cemetery, where basically the cell will move after it has been basically simulated to die. And then the core theoretical contribution is this insight that time reversal of a diffusion with killing states corresponds to a diffusion with birth. So similarly, basically, we can create a reservoir where we can take cells, they follow some form of shadow path, and then are basically reintroduced into the system. We did this now with the cell lines that are treated with cancer drugs at very distinct time points. And we can see that we are able to basically present those, or like recover those cellular dynamics. And here you see that initially, basically, there's a lot of like birth events, so that the effect of the drug is actually delayed. And only at a later point in time, we see actually heavy effects, or like a lot of like death events. A problem, of course, is that we can never validate that this is actually a true moment of apoptosis. We can only see if the predictions we make basically recover the mass or the amount of cells, this number of cells over time. And then we can just check for many different drugs, are these patterns somewhat conserved? So how does this help us to have those models that transform an unperturbed into a perturbed distribution? So here I'm going to show you actually results of a slightly different method that is theoretically connected. But here we are working with a cohort study that is running in Switzerland since many years, where we have patient samples in control and treated state. And the idea is to learn now a generalized perturbation model, and then the moment we have a new patient coming into the clinic, or like a patient we held out from the training, to then predict how those cells would respond to a particular treatment, and then make decisions on if this is a particularly good treatment to choose. What is the rate of survival? Can we say anything about the chances and the effectiveness of this treatment? So this is data that are recorded within this tumor profiler consortium. So we have here 37 melanoma patients, and they have been basically, these patient-derived cells have been screened against 34 drugs heavily used in the clinic. The data we are using are based on this technology known as 4I, or multiplex imaging, so technology developed by Gabriele Gut and Lucas Pelkmans. And so this is, of course, a project in collaboration with both of them, as well as with the Reg Lab at ETH. And so now we have roughly 50 features that are extracted, both morphology features as well as protein intensity features from different proteins. And we look at the average signal inside and outside of the nucleus. So the problem is that this clinical study is very, very diverse, right? So we have 37 patients. They all show very, very different type of clinical meta-information. So it is a particularly challenging problem. So they have very different number of treatments in the past, subtypes, driver mutations, and they, of course, are also, these meta-information are also very relevant when making a prediction about their responses and their success in the success of the treatment. So the method I presented in this talk, they fall more into these diffusion models, but these models are somewhat theoretically connected to a series of methods that I developed during my PhD, starting from those methods that just do a simple shift in how does the perturbed, unperturbed population transforms into the perturbed population. So these are also generative models. They are not just based, they're just not based on diffusion, but these two methods are now going to be the core of method of this clinical cohort, as this is running for much longer. But they are all theoretically very nicely connected through the principle of optimal transport. So these are all somewhat OTE type of methods that continuously, so Schrödinger bridge is actually equivalent to an entropy regularized optimal transport just from a dynamic point of view. And for those interested in understanding how all those methods are connected and what are the benefits one over the other, we gave a tutorial last year at ICML about optimal transport for dynamic systems, covering both the core components of optimal transport, but also how it's basically the workhorse, or like the methodology behind diffusion models, or like some form of diffusion models, diffusion Schrödinger bridges, and so on and so forth. And for those who care much more about having this always in perspective with the biological problem, there will be a Nature Reviews Methods primer out soon that covers all those connections, as well as the somewhat core problems it has been applied to in single cell biology as well as spatial biology. So the title of this review will be Optimal Transport in Single Cell and Spatial Biology. So now what is the setup for this study and really understanding how patients are going to respond to a treatment is that we have patients before treatment given a particular, and given a drug, we want to really learn some of the map, the model that transforms this unperturbed into a perturbed population. And we do so by parameterizing this, and then at test time use this parameterized model and now predict how all cells individually, right, respond to a drug. So as the exact same setup as throughout this entire talk. Now what I do then is that for every cell feature value, right, I'm able to predict the perturbed features, right?
Charlotte,
how they would look like. Yes.
Hold on a little bit. Yeah, try to speak again.
Test.
Okay, now it works. Just
Perfect.
this slide. Everything was good.
Just this slide. Okay, so basically, right, the setup I just introduced is now we want to basically, for an unseen patient at test time, predict how every individual cell is responding to the treatment. And this has been exactly the same. So exactly the same setup as what we saw throughout the talk, just trying to iterate on this once more. And what we do then is really predict for every individual cell how every feature basically changes upon perturbation. And now what we can see, and so here I'm just walking through this, first is in light blue, you see for just 2 features here and for one particular patient for a particular drug, we have those panels basically for every patient, for every drug, for every feature. We can see the distribution of those cell states over in the control and the unperturbed population. And we see that there's, for example, a decrease. And we want to be able to understand how this light blue curve changes into this dark blue curve. Same here. How does the light blue curve changes or transforms into this dark blue curve? And of course, this simultaneously over all features. And what we can see is that the methods, these basically distribution-based methods that transform one distribution into another, really well capture that part of the distribution is not going to be responding, but the other one really shows a heavy shift, right? So this entire part of this distribution actually shows quite a shift to the left. And of course, meta information matters. So we can condition those on the information we are provided with, such as number of treatments, as well as driver mutations. And then we are actually able to capture that some of the patients that have been that would usually not respond, that if they carry, for example, this driver mutation and the number of treatments, that they actually show response, right? So here sellout is actually staying on the control population, while it should really simulate a shift to the left for this particular p-ERK signal. And last but not least, sometimes, right, given a particular condition that doesn't make like a perfect translation into the cell profiles we measure, is actually not showing any type of response to a drug, even though the methods that would be trained in this unconditioned sense, like unconditioned based on the clinical meta information or the clinical information we are provided with, would actually simulate or predict, forecast a response to this. So this is very nice because we have now a very good understanding of how do patients on the level of individual cells and for different sets, like a set of different biological markers respond to a particular treatment. So we can use this now to make downstream analysis on, does this tell us something about estimation of overall progression to a treatment, like or progression of a treatment, like how are the survival rates, and what are particular clinical associations that we should be aware of that we can then use in order to design future treatments or more effective future treatments. As I mentioned before, it's not only important to understand how cells are going to respond. What we now can extract by basically, we have now a pairing between an unperturbed and a perturbed state of a cell. We can subtract those and really understand the response patterns of every individual cell for which we made a prediction. And now we have data where we actually have survival. So we know those who have basically died very, very quickly, those who were alive much longer, and those who are still alive potentially. And what is really remarkable is to see that the information is in the response patterns. The information on the survival is really in the response pattern of the single cells. How are those different subsets of cells are going to respond to a treatment? It's very much a good way of forecasting how successful a treatment will be. Similarly, right, we can make a basically an analysis of basically this response of the response of the treated cell as well as the treated states. So here we just look at the observation of the treated set of cells. Here we really look at the response pattern of individual cell and put this against the marker, the medical clinical information we measure, and we really see subpopulations in this patient cohort that, based on a particular meta information, just show an overall different response, on overall different response pattern than those which don't carry, for example, this particular driver mutation here, BRAF. And so something like this, these type of patterns are very, very hard to see by just forecasting how cells are going to respond to a treatment. Based on this, we can do a hypergeometric test to understand certain clinical associations of a treatment to a particular clinical information which we have, like driver information, location, biopsy, and so on and so forth. And we can see that the predictive response gives us much more of those type of associations that we can look into and then analyze, and are ultimately very important for the selection of certain treatments. Just to highlight one of them, for example, here, this connection of Paclitac cell and the NRAS driver mutation. And this is something very well known in the literature because Paclitac cell enhances microtubular stability. causing cell arrest. So we know that those patients which carry an NRAS driver mutation actually show a certain activation of the ERK-MAPK pathway and then show a very different response to pancreatic cell than those who do not carry this. And this is just one example. And so this is ongoing work that will be soon out. So what I was mentioning at the very beginning is that we, of course, collect much more data. And what I showed you just on the recent slides is that we have, so far, we've always tried to do this type of analysis on patient cohorts that are very, very small, because all patient cohorts are very, very small. So here we were dealing with 37 patients, of course, like thousands and thousands of cells, but we have so much more data acquired that should give us a much better understanding of cell states and their response to certain treatments. And so this is currently ongoing work of trying to understand, can we learn those foundation models based on all cells or all phenotypes that have ever been recorded, and somewhat find a representation of a cell state in this like virtual cell embedding. And so this is very much a paradigm shift from learning basically or training a model on this particular data set I'm giving with to trying to understand what I'm doing or what my aim is in the context of models that have been trained on all data available. And so now we have somewhat a distribution over cell states, and this would allow many, many different things. On the one hand, of course, align many different data modalities or many different type of tissues from different species, but then ultimately enable us to discover novel cell states, understand cells in space, not only in time. And then those type of methods I have been introduced, methods I have introduced in this talk, methods to understand really continuous time dynamics or responses of cells to extrinsic perturbations. And then down the line, and this is also going to be the focus of my lab, is how can we use these type of technologies then to derive in silico experimentation, really like the engineering of novel cell states by simulating basically in silico how a cell state could look like, getting it back into the lab, testing it out, feeding it back in. So really a closed loop between the lab and something I can design in silico. And then, given that those patient cohorts are so small, how can we look at those patients in basically with respect to all data previously recorded so that we can make those studies I showed you just before on the slide? How can we basically look at those studies in the context or make use of previous data and previous insights we've gained to make decisions on patient treatments in context of those knowledges. This will be very important for future clinical decision support systems, and this is all future work, so stay tuned for what is coming next. If this was of interest for you, so why now, why in Switzerland, why the lab we are starting or we are launching is that on the one hand, we of course are still in this growth phase of massive amount of data. So here we have really data doubling every 6 months, and very nice consortia that are organizing such data in whole organism cell atlases. On the one hand, there's still a huge desire for methods that can deal with those data. We are at the very starting point. We are not even able to really properly align certain modalities in principled manners or really deal with this type of massive amount of data. So there's still a very important need for model development and model design. Switzerland just has invested incredibly in like a large supercomputer with 10,000 GPUs. So for an academic setting, this is quite unique. And there are Swiss-wide consortia running that are really gathering those patient data across many different hospitals, which is really exciting. So with this, I would like to thank my PhD advisors. So this is just half a year ago that I basically graduated, and I'm deeply knowledgeable for the work I've done with both Andreas at ETH and Marco at Apple. Currently, I have a fun time working with Aviv and Yuri at Genentech and Stanford, and I have had the pleasure to visit Anne and Shantanu at the Broad during my PhD. The work presented here in this is both with, of course, Gabriele and Lucas at University Hospital Zurich, as well as the University of Zurich, as well as with the lab of Gunnar Rätsch and Stefan and Kyong at ETH. Students I've been working with, as well as Yaping, a postdoc in Andreas' lab, and Valentin now at DeepMind. And so if this is interesting to you, visit our newly launched lab page, as well as follow me on Twitter for updates and news. And in case this is interesting for you as a future endeavor, reach out to us. And with this, I'm thank you for the attention, and maybe there's time for questions. I'm not so sure what the setup is. Thanks a lot.
Thank you very much, Charlotte, for a wonderful talk. Are there questions in the room or on chat? See anything in the chat? Yes. Maybe you can, I'm not sure, do we have a microphone or?
The mic.
Does that work? Yeah. Do you mind? Please. Maybe you can say your name and your affiliation.
Hi, Charlotte. Thanks for the great talk. Really nice. I'm Ruby from Oxford, PhD. I was just wondering if you could just basically go over the part where the diffusion model and how from the diffusion model you get to the response to treatment.
Yes. So basically what you predict in the end here, and so maybe this slide shows it the best, is you basically have, again, your cells in the, like this is now a PCA projection of them, so just in 2D, but so this is every cell. Here, this is actually RNA sequencing data in a very high dimensional cell space. And now what you basically predict is for every individual cell, so you can almost imagine this being a single image. We are basically predicting how this single image or how this feature vector is basically changing over time. So the diffusion method, having here a drift, basically the drift is an RD to RD function. It's an RD to RD function that tells us how how this feature vector is changing over time. So from X0 to X1. And so then this is basically the perturbed cell profile, which we record with the same measurement as the initial. Basically, you really predict a transformation of your image over time, or here, this is not an image, right? This can be an image of a like microscope, but this can also be just an extracted feature vector for an individual cell. Very much depends which type of measurement you're working with. But it's basically, you're just learning a very big RD to RD function, like just a map on how the cell profiles changes upon perturbation. Yeah. And then you can learn one model, of course, that is basically conditioning on many different type of drugs, or you learn basically distinct models for a particular drug. So this depends on how complicated your model wants to be.
Okay, great. Thanks very much.
Anything else? Yeah, Dimitri.
So, Charlotte, very nice talk. I've been doing a lot of physics-based modeling myself in the past, but what I want to ask you is, some cells under the influence of drugs will die, some will do better, some will not better. So your way of modeling, does it capture abnormalities that you find naturally in cells? Like I've done
yes. Yeah, so there will always be outliers, and if these outliers are very scarce, they are going to basically, it's going to be very hard to capture. So in the end, this is always trained in a mini batch setting. So we sample a batch of cells, that somewhat represent the distributional state of the tumor, and based on this, we try to forecast for this set of cells how they will respond, and then the loss is computed by sampling actually a treated population, like a sample of a treated population, and compare them with a distributional metric or like some form of loss. So overall, if there's like one cell that has really an individualistic mind, it's going to be very hard to capture. There are, though, some of the methods we developed that allow for a certain level of unbalancedness. So basically, some settings that are a bit more generous to those type of outliers, but the methods here I introduced, they are not, right? Yeah.
Yeah, there's an additional question I have, which is related to this, is that if sometimes the cells mutate, so basically mutate because they become resistant to the drug. So while in your initial experiments you see that the drug works, after a while, I mean, I've done some experiments of course with biologists, where I saw they looked like they were dying, and after 2 weeks they sprang back and they were even more potent than they used to be before.
Yeah, this is actually something we see, right? We see sometimes initially they're actually dying and then they start to proliferate. I mean,
Right.
so having said this, our method is able to recover this. And also it's going to probably say like which of the subpopulations, like I didn't show you, but let me just end this. I think you should see. Basically, we still are able to somewhat understand, you know, what are the subpopulations that are really resistant or that have very different type of correlation to a certain survival rate. We are never going to be able to say, okay, so this particular cell, we can only, because we never have a ground truth alignment, we can always tell you, okay, so this is a particular subset. And we also see this, so we see, we see basically sometimes that there's first actually cell death and then proliferation. But if things continue, you know, beyond the measurement horizon, like there's no guarantee that those methods basically are going to extrapolate into the future. So the hope will be that if we've seen, you know, many different type of data, how all those somehow data we ever recorded are going to respond, you know, and show what type of response patterns they show, that this might enable us to do those generalization. All the methods we currently are dealing with, they are very poor in terms of generalization. So this is why we are actually thinking of these very large models trained on all type of data. So generalization performance also to a new drug, right, to a new cell state right now is still pretty poor.
Well, thank you very much.
Yeah. Any other questions?
I had a quick one, Charlotte. I was curious. So it's a really beautiful work and really clear about sort of the application of diffusion modeling to sort of predicting treatments for patients based on the data that you have. It looked like most of the examples that you have were patients who were given monotherapies, and so I could imagine how you might be able to then predict, you know, what a new patient might get. I'm curious, have you ever, or do you think the system would work to predict new treatments? So, for example, combination therapies or something like that. That maybe you could even before, you know, urge people to do a clinical trial for combinations.
So here actually the data actually contains a couple of combination therapies, but we basically just treat this as a new drug combo. So we don't really train, we don't learn the combination. I have another work which is this. It's just in a NEUROS paper. And the idea was that, okay, instead of learning like individual maps, right, can't we just condition it? Here in this example here, I showed you, okay, let's condition our map on the clinical meta information we have. One of the experiment was also, can we condition basically the map we learn on the type of drug we are provided with?
Yeah.
Unfortunately, most of those data are not public. So I tried to do this for genetic intervention. So basically genetic knockouts, where we have a couple of like knockouts that are done in combination. And for this, if you learn a very good embedding somewhat on this, on how this drug effect is, then you can do predict something to unseen combination. I think I actually have probably, yeah, here. So this is, for example, basically you have certain genetic perturbations and you're trying to basically embed them and then condition your map on how that tells you there is perturbation to unperturbed to perturbed transformation based on this condition. But there is just really the moment you are not working in a pharmaceutical company, I'm not sure we'll ever have really access to a lot of those. There's a couple for bulk data, but the data situation is not great. So I think mostly work here has been using genetic perturbations and not drug perturbations.
Yeah, well, that would still be really helpful for understanding polygenic.
But it's super hard. So basically it's incredibly hard. So you can see we are better than other methods, but all methods are somewhat a bit bad. Yeah. So, but this is again, same system, right? Like if we actually acquire all data and we use all data, how much better can we do? That's the big question of the next couple of years.
Great. Well, no other questions. Thank you for a wonderful seminar. It was really informative and clear, and good luck on your new lab.
Thanks a lot. And thanks for the invitation, and enjoy the rest of the workshop. Bye-bye.
Yeah, thank you so much, Charlotte. That was a wonderful.
Bye-bye. Thanks a lot.
So, yeah, to wrap up the session, we would run awfully late, but I think the talks are really interesting. Everyone is still very interested in staying. So I think I feel bad toward the poster session, maybe, but we can't run too much over because there's additional talks, people are still waiting. So we resume at 4, if that's okay. And I think for the poster, usually we can only use the boards during the poster session time, but I think by this hour, probably can just leave the poster until the end of the day, if that's okay. Yeah, so let's resume the talk in the room at 4, and then I think posters, until they scream, I think we can just leave that on and people can continue to engage. Yeah, thank you so much.
Yeah, so okay, so we are starting now with the first talk of this afternoon session. And it's gonna be about gene level representation learning via interventional style transfer and optical pool screening. And it's a collaboration between University of Washington DC and Berkeley. And Mahtab Bigredi is going to do the talk.
Hey, good afternoon, everyone. I'm Mahtab, a second-year PhD student at University of Washington. Today, I'm excited to present gene-level representation learning via interventional style transfer in optical pool screening, which was done during my internship at Genentech. So let's start with phenotypic screens. In phenotypic screening, we introduce perturbations to the cells, and with imaging the perturbed state of the cells, we record the effect of the perturbation. And in the next step, we perform the phenotypic profiling to embed the phenotypes to the common latent space to allow us to relate the perturbations to one another, study gene function, discover gene relationships, and search for candidate targets eliciting known phenotypes. And in this project, we are focusing on phenotypic profiling. The dataset that we use in our project is from a study published in 2022. It has cultured human HeLa cells. It contains CRISPR knockout of about 5,000 genes, 4 guides per gene, 250 non-targeting guides that we consider as unperturbed. And there are 4 channels for each single cell image, 3 for morphology, and 1 DNA damage. And one more thing to mention about this data set is that it is pooled. So now let's explore the concept of pooled screens. In array screens, there are many wells and plates, and each of the cells in a single well receive a single perturbation. But in pool screening, in each of the wells, cells receive a pool of perturbations. Array screens allow both genetic and compound perturbations. However, pool screens only allow barcoded genetic perturbation. And there are always trade-offs. So array screening is more expensive, but pool screening requires more processing, and it's harder to demultiplex the data. So now, one of the main and most important challenges in phenotypic profiling is learning embeddings that are not influenced by confounders and background noise, such as batch effect, illumination. And generative adversarial networks have shown promise in addressing the impact of technical confounders on learned representation through the use of style transfer. But what is style transfer? Style transfer is a technique that takes in an input image, content or reference image, and a second image, which is our style image, or it could be a representation or code called a style code, and it generates an output image that has the content of the reference image or the first image, but it has the style of the second image. Here in our work, we view genetic phenotypes as style codes and content image as single cell image with all its background information and confounders. So now we introduce our work GRAPE, GANs as robust adversarial perturbation encoders. So the way it works is that we pass single cell images through the model with our target gene embedding, let's say gene I, and then we will get our output or prediction, which is the image, the single cell image that has the background of the input cell, but it has the genetic phenotypes of the gene that we pass through our model. And here we are looking at target phenotype. This is a different cell from a different batch, but it has the same phenotypes from this genetic perturbation. Now let's move to the architecture. So in the architecture here, we see that we pass single cell images through the model, through our generator, which is an encoder-decoder based model. And we also pass our target gene embedding through the model using other in layers. So here, this embedding layer, this is the most important part of our model. This is what we use after training and we use for further evaluation. So here, when we want to generate an image, a single cell image that has phenotypes of gene I, then here we will take embeddings of gene I and pass it through the other in layers. So here, other in layers, what they do is that they match the statistics of feature maps of the decoder with the input that we gave to them, which here is our genetic embeddings. So here in our model, we're using adversarial loss. And like other GAN type models, we have a discriminator. But the difference here is that our discriminator is multiple head. Here it means that for each of the genetic perturbations, so if we generate an image for gene perturbation I, then we will look at the head I and do the real versus fake classification. It is different from traditional GAN models that they have a single fake versus real classification. And it's better than doing a separate classification for perturbations because perturbations are very subtle and then it will make it harder. So here are some visualizations of the generative performance. As mentioned before, perturbation phenotypes are subtle and not very easy to for even experts to see with naked eyes. So here we are looking at real images, like real cells that they're perturbed with these two genetic perturbations. And in the middle row, we are looking at images that were generated from our model. As I said, it is very hard to notice it with naked eyes. So to gain more confidence, we did style transfer. We featureized generated images using Cell Profiler and trained random forests to classify unperturbed and perturbed cells and plotted the distribution of the 5 most important features from random forests. And as you can see here, the distribution of the generated images, which were shown in green, are very close to the real and experimentally perturbed cells than unperturbed input. And for more quantitative evaluation, we look at biological ground truth, quorum, which is a database of protein complexes. And the notion here is that if genes function together as a part of the same protein complex, knocking each of those genes should yield a very similar phenotypes. So here we built a gene interaction network based on the quorum labels and saw how our embeddings correspond to the gene interaction network. From the 5,000 genes that we had in our data set, we selected only 106 genes from 14 non-overlapping and functionally diverse protein complexes and trained a model with these subdata. And we used 2 types of metrics to evaluate our model. One of them is mean average precision retrieval, and the other type is clustering-based methods. That we have 3 metrics: purity, adjusted rand index, and normalized mutual information. That each of them capture a different aspect of clusterings. So after getting our embeddings from our model that I showed in the previous slide, after getting the genetic embeddings, here then we do the k-means clustering. And now we have our ground truth labels using quorum, so we can do the evaluations. And here in the bottom figure, what you're looking at are numbers for k-means clustering for different Ks, for different baselines, and also our model grade, which is shown in purple here. And we can see that for all Ks, for k-means, our model is outperforming. But we know that 14 is the ground truth number of clusters. And even here in the table, we can see that our model is outperforming all other baselines like engineered techniques, IMPA, which is a recent paper, Gene2Vec, and random baseline for all clustering metrics. And it's doing on par with engineered techniques on mean average precision. We have also done a lot of ablation studies, like switching on and off some of the losses in the StarGAN V2 model, which our model was built upon that. And we have also added attention layer on top of our features to see that if it improves the embeddings on evaluation metrics. We have also changed the position of the embedding layer, like before the mapping network, after the mapping network, which is a number of fully connected layers. And we saw that in this setting that we use our embeddings before the mapping network, without any attention layer, using exponential moving average, we will get the best numbers, as you can see here in the table. So thanks for listening. If you have any questions, please feel free.
We have time for a few more questions. All right, thanks a lot. If you have, you can always email the speaker, or you can talk to them.
Thank you so much for this.
Okay, so the next paper is about super resolution of biomedical volumes with two-dimensional supervision. And it's going to be presented by Cheng Jiang, right?
Yep.
From University of Michigan.
Sharing.
So 10 minutes, 4:25.
Yeah, it's coming.
Good afternoon, everyone. My name is Cheng, and I'm very excited to talk to you about our project super resolution of microscopy volumes with two-dimensional supervision. So I'll start with talking about volumetric microscopy with stimulated Raman histology, or SRH, and then I'll talk about our method, MSDSR, and I'll wrap up with some experiments and results. So let's dive right into volumetric microscopy. So here we're working with stimulated Raman histology. It's a rapid optical label-free method that allows us to do intraoperative imaging, and we can acquire a image like this within minutes in the operating room. And as we can see here, with the SRH or with any biomedical microscopy, we have a two-dimensional cross-section. And there's a lot of inherent ambiguity with these two-dimensional specimens when we're trying to use them to characterize three-dimensional biomedical specimens, right? So an ideal system to analyze these biomedical specimens will be three-dimensional. So right now we have very limited technology that we can do to acquire these three-dimensional data. So here we're hoping to demonstrate that SRH is a viable candidate for three-dimensional imaging. So at the University of Michigan, we have been collecting some of the three-dimensional data, and one of those volumes might look like this. So what's hidden in plain sight here is a complex system of blood vessels that we can only see with three-dimensional imaging. Right? As you can see, there's a lot of system here that you just can't see in 2D. So indeed, volumetric microscopy can improve diagnostic yield and accuracy. But there are a lot of challenges that come with biomedical volumes, right? There's a lot of barriers to integrate them into clinical medicine and biomedical research. Mainly, there is long imaging time that's associated with each of these specimens, right? The depth resolution is still not optimal. It's worse than we can get in the X and Y axes. And there is insufficient volumetric data that's just publicly available. So if we want to start with some kind of large-scale research with machine learning, this data is just simply not available. So the key computer vision question that we want to tackle here is, can we super resolve these microscopy volumes with two-dimensional data alone, right? Because if we can do this, we can speed up imaging time while also increasing the resolution of the data that we collect. And if we can do this with only two-dimensional data, we don't really need to start with a large three-dimensional data set to get started. So to tackle this problem, our solution is mass slice diffusion for super resolution, or MSDSR, right? Our method is based on observation that all spatial dimensions are equivalent with biomedical imaging. So a specimen doesn't really matter which axis that you look at the specimen from, is a representation of the same underlying tumor biology, right? So there's the lack of orientation that really allow us to train the model with any two-dimensional plane, such as XY, and we can use that to generalize onto different planes, such as XZ and YZ, right? So we can approximate this conditional probability of high-resolution volumes by modeling each slice independently. So here is our approach. Our approach has two parts. The first part is a training of a conditional diffusion model, and the second part is to aggregate these volumes together. So let's talk about training the diffusion model. So as we are familiar with diffusion model, there's really a forward diffusion process where we gradually add noise to an image, and there's a reverse diffusion process where we have the model to gradually denoise the image step by step. In a super resolution task, however, we can condition the training of these models with a low resolution image or the observation that we get, right? So what we can do here is to do this through a row-wise mask to interlace our observation with our noise as the condition. And at training time, we actually don't have to do this evenly, right? We can randomly select rows to insert these these images, the rows, right? So our model actually looks like this. So in the reverse diffusion process, we have these row-wise masked noise images that we have through the reverse diffusion process. So once we train our model, we can then use our diffusion model to do the super resolution of our volumes, right? So in the step procedure kind of look like this. We have a low resolution volume in the Z direction, and we slice the volume in X or Y direction, right? We can slice them in either way. And what we can do is we can then super resolve each slice, right? And how we do that is we can evenly interlace the observations with the noise. And from there, we can concatenate the volume together, and we can then have this high resolution volume. Since we can slice our low resolution volume in X or Y direction, we can actually slice this both ways and have two predictions for each low resolution volume. And from there, what we just do is an average process to combine both combine both predictions into our super resolved Z-stack volume. Right. So moving on to some results. The first set of evaluation that we did is a paired 2D evaluation, just to make sure that our diffusion model actually works, right? So what we can see is we use FID to measure the perceptual quality of our generated images, and for FID lower is better. And as we can see, our model MSDSR outperforms all the baselines that we have tested. And visually, we can also see that here, each row corresponds to a different sampling ratio during super resolution. And what we can see here is that MSDSR produce high quality images even with very large, with very small sampling ratio with a lots of rows that's being skipped. The next set of evaluations that we did is a two-dimensional super resolution. So diving into a little background into our data set. So we've been collecting this Z-section data set at the University of Michigan. And our data set consists of 1 micron per pixel on the Z section, on the Z dimension, and 0.5 micron per pixel for the X and Y dimension. So how do we evaluate our model if we don't have the ground truth isotropic volumes? Right, so here we present slide FID, which is a novel metric that we are using. Because to tackle this problem, if we have a high resolution volume, then the high quality volume should have high quality slices across all three dimensions. So because biomedical microscopy is orientation agnostic, so we can then use two-dimensional, high-quality ground truth two-dimensional images to evaluate our slices, right? So we can evaluate these across the XY, YZ, and XZ plane. And so here are the metrics. Again, lower is better, and we can see that MSDSR outperforms all the baselines that we have. We can also visually inspect the volumes that we have. Let's zoom into some of the volumes. And as you can see, there is a very significant stretching artifact on bilinear interpolation. And the UNet methods are all very blurry as expected. And the MSDSR method produced high quality images across all the dimensions. We can also apply MSDSR to much deeper volumes and much larger slices. And as we can see that these super resolved images look really great. Looking forward, we have a lot of exciting next steps planned for MSDSR. First, we want to improve the 3D consistency of the model to produce a more coherent volume and reduce the concatenation artifacts that we can see. We also want to enhance our data collection to collect with higher Z resolution and higher depth. And finally, we do want to evaluate our data in our MSDSR on downstream clinical tasks such as patient survival or patient response or treatment response. So in conclusion, MSDSR super resolves two-dimensional volumes with conditional diffusion and only uses two-dimensional super resolution. And SliceFID is a new volumetric unpaired perceptual quality that allow us to evaluate our model. So for more details, please visit our website at mlens.org/MSDSR. And I would like to thank all of our co-authors and our sponsors who make this research possible. And I'll take any questions that you have. Thank you.
Yeah, I must have missed it, but what's the advantage of using the row approach to the denoising?
So the row approach allows us to condition our denoising model so that it stays consistent as the observations that the low resolution observations that we have. Yep.
So are you training the diffusion model as a 2D model still?
Exactly. So the diffusion, that's one of the advantage of our model is that the diffusion is only done on a 2D model. So we can use any existing two-dimensional data set to train this diffusion model. As we know, the diffusion models are pretty data hungry.
Right. So the reason why not to train a volumetric like 3D model is basically the data efficiency.
Because the collection of these data is really, really expensive.
Another question, so since for all the generative models, right, do you worry about hallucination?
So far, I haven't really seen any hallucination with our data. If we condition the model with a lot of observations, so with like 2x super resolution, then there isn't much room for the model to really hallucinate because every other row is being supervised. With more larger skips, like if we skip A rows, then there will definitely be instances where the model will try to hallucinate because there are just multiple correct answers, right? Because there are multiple possible solutions in between two observations. So it's definitely possible for the model to hallucinate in those cases.
Yeah. So how do you evaluate quantitatively, basically, how well this works?
Yeah, so for the three-dimensional volumes, we're using FID to evaluate them. And the assumption here is that a three-dimensional volume, and if you slice them in XZ, YZ, or XY, you will have the same high resolution slices, right? So we can then do this unpaired evaluation with a set of ground truth high resolution images using FID.
Thank you so much.
Any other questions?
Can I ask a follow-up on that? You say, I mean, your ground truth is low resolution in Z direction, right? And
So
you're basically trying to super resolve it in Z direction by factor of 2 and keeping XY the same.
Yes.
And then what do you test against for the Z direction?
So FID is an unpaired metric. So we can slice the volume in X and Y. So each slice is a Z-dimensional view of the volume, right? So FID is a measurement of the distribution of the data. So if we compare that to a ground truth high resolution, then
Oh, so you're essentially measuring the statistics of the image and saying it's consistent with a high dimensional.
with
High resolution.
Yes.
The statistics are.
Yeah.
And is that dependent on the statistics you choose?
FID is a pretty standard metric that people use in the computer vision field. It uses learned deep embeddings to measure the distributions. Yep. Yep.
Which downstream tasks can you imagine with your method?
Yeah, so the first one that I can think of is cell segmentation, right? Because a lot of 2D images, if you're going to segment cells on them, sometimes you just don't know if you're slicing a cell at the wrong Z index, right? So maybe you have a tiny cell there, or maybe you are crossing the cell at the correct Z, then you have a really large cell nucleus there, or maybe you're slicing it at the wrong Z, then maybe it just looks like tiny, or maybe it may even look like a bubble, right? So to evaluating this is where Z section can really benefit our like the cell segmentation task. But we're also interested to just broadly to do histologic classification or other survival prediction and other type of tasks as well.
So,
okay, thank you.
Yeah, move to the next speaker. So the next speaker is going to talk about weakly supervised self-assistive learning, which involves morphological profiling of single cell images. And Hemming Yao from
Shows presentation. Slideshow. Oh, go to the first slide, please.
Okay, hello everyone. Today I'm going to talk about our work on developing weakly supervised dyno for morphological profiling of single cell images. Our work focusing on data from OPS. OPS is a powerful tool that combines high content images with genetic engineering to investigate gene function. Here I give a quick overview, and by the way, the data set is the same as Matap's presentation. In the OPS data, usually in one experiment, a library of guide-RNAs targeting multiple genes are introduced to a pool of cells. Then each cell is usually receive one guide-RNA, meaning in that cell there's one gene is perturbed. After that, a phenotype imaging is performed to characterize the cellular response to the genetic perturbation. Here I give an example of 4-channel high-content images. And we can see for each channel, it describes the morphology of the nuclei and other subcellular organelles. In this data, we also have a different modality of the images that is used to identify the perturbation introduced to each cell. In this data set, in this study, we used a public OPS data set, including a CRISPR knockout of 5,000 essential genes. In this data set, it contains 8 plates, and each plate has 6 wells. Usually for each well, it contains 1 million of cells, and the library of guide-RNA is introduced to. Should I continue?
Okay. I don't even know what, but sometimes something will fix it.
Magic. Hello? Okay. You still?
Okay.
You come back.
Yeah, thank you.
Yeah, well, yeah, for each well, the library of guide-RNAs are introduced. So every well, they are biological replicates. In this study, we use wells from 6 plates for model development and wells from 2 plates for model evaluation.
Here I give a very general overview of the workflow. After we have phenotyping imaging, we will generate single cell crops and then extract the morphological features for every single cell crops. The idea here is later we want to aggregate the morphological features of the cells with the same perturbation to generate the representation for each gene. After that, based on all the morphology profiles from the genes, we can calculate the pairwise cosine similarity to build a similarity map and infer gene function. So our assumption here is if perturbing two genes respectively leads to similar morphological changes of the cells, then we may assume these two genes have similar function. There are multiple existing morphological profiling methods on high-content images. Here I list 5 categories. I won't go into too much details. Here, for the first type of the method, it's engineered features. And the cell profiler is a very famous, well-known method in the last 10 to 20 years. The weakness here is they are very sensitive to cell segmentation. From existing literature, usually people find the self-supervised learning are very powerful feature extractor for this task, especially for Dino and MAE. But also people find out it is like for those methods, it may be very sensible to confounding factors. So Recurrent did a fantastic job on building MAE for high-content images. Here we focus on Dino model and try to make Dino more robust to confounding factors. And also, maybe worth to mention here, the confounding factors are usually experimental artifacts across different experimental mini-batch. So we also call it batch effect. Cross-batch sampling is an existing strategy from a paper shown here. It is used to enforce the embeddings consistency across different experimental batches. So usually in a regular Dino framework, the same image with different data augmentation are fed into teacher and student network, and then across entropy loss is used to train the model. With cross-batch sampling, instead of using the same image, we will use the image with the same perturbation, but from different experimental batches. So using this way, we can make the model more robust to batch effect. We applied this method to our OPS data. However, the training failed, and it's mainly due to the heterogeneity of the single cell data. Because for two single cells, even though they are with the same genetic perturbation, but they may in different cell cycle or cell states. So forcing the model learn consistent representation for those two single cell images will make the model collapse to a trivial solution. To solve this problem, we proposed SciDino by introducing site-level representation. So here, instead of sampling one single image from different experimental batches, we sample a set of the cells. And our assumption here is if we sample a set of cells, then the distribution of the cells in different cell cycles may be similar across, between these two sets of the cells. Then this set of the cells are fed into the Vision transformer to generate a set of the feature vector. Then in the projector, we have an aggregation layer to aggregate those set of the feature vector into a unified feature vector. And then we can compile these two unified feature vector from two experimental batches. From our study, a simple average works pretty well in the aggregation layer. After we train the Vision transformer, we will generate the embedding of the single cell images as the single cell profiles. After that, we will aggregate the cells with the same perturbation and also in the same batch into batch-level gene profiles. After that, we will did some batch alignment and also aggregate the batch-level gene profiles across different batches into consensus gene profile. We did some evaluation on the embeddings. The first evaluation is to check out the batch effect contained in the embeddings. We built a k-nearest neighbor classifier on batch-level gene profiles. Is to predict the batch ID of the target nodes using the batch ID from the neighbor nodes. So here, a lower value means the embedding has less batch effect. Similarly, we also did a reproducibility analysis is to build a k-nearest neighbor on perturbation ID. Here, a higher value means profiles of the same perturbation, but from different batches are clustered together, meaning those embeddings have a higher reproducibility and also they are more consistent. As I previously mentioned, our final goal is to infer gene function. So here, a more important evaluation matrix is to say how much known biology can be recapitulated in the similarity map we built on consensus gene profiles. To perform this evaluation, we need to first build a ground truth gene graph using existing data set. Here we use the quorum, which is a data set including gene sets for each protein complex. So every genes in the same protein complex, they are connected to build this ground truth gene graph. Then with this gene graph, we can model this task as an age prediction task. So we will first look at our similarity map and then choose the top 5 most similar pairs, and then to see how many ages in the ground truth gene graph can be recalled. So a higher value means our embeddings contain more biology. We compared side-dino with regular dino and also engineered features. Very impressively, we found out our side-dino has the best performance in terms of the reproducibility and the biological recall, and also it has the least batch effect. Here, we tried 2 variants of the side-dino. The side-dino sgRNA means we sample images with the same guide RNA but from different experimental batches, while side-dino gene target means we sampled images with the same gene target, but maybe they are from different guide RNAs. On the bottom figure, I showed precision recall curves for our biological analysis. It further shows the superior performance for side-dino when it is compared to dino and engineered features. This is a closer look at the biological analysis. In the first column, I show the adjacency matrix of our ground truth gene graph from the curated column. And here, every diagonal block means a protein complex. So all the genes within, forming this protein complex are connected. We could say for the engineered features or dino features, the genes relations within some of the protein complex are totally missed, but our side-dino embeddings can capture those relations. It further means the side-dino embeddings record more known biology relations, and also it gives us more confidence on any novel relations discovered by side-dino embeddings. It can help us generate more reliable insights for future drug discovery. We also did some ablation study, like the same image here. We will try 3 different strategies. The same image when n equal to 1, it is the regular dino. And the same image n equal to 4, meaning we use regular dino. We use regular dino with set level presentation, but we are not sampling cells from different batches. Within batch means we use cell level representation, but we sample cells from the same batch. And then the cross batch is what we proposed in this study. From our ablation study, we can say both cell level representation and cross batch sampling are critical to achieving success. And thanks for the attention. If you are interested in this work, please check out our paper. We also released our code, and also feel free to chat with me during coffee break. Thank you.
From the audience?
Any questions?
Yeah, can you go back one and explain? So the bottom is just choosing different number of n for the number of samples you stack up cross batch, right?
Yes.
Or within each batch. Okay. Can you explain the same cell n equals 4? Same cell n equals 1 is dino.
Yes. Same cell when n equal to 4 means we sample 4 single cells with the same perturbation. And we use this same 4 images for both teacher and student. We didn't do cross-batch sampling.
Okay, and within batch?
Yeah, within batch A, we sample different set of cells, but all those cells are from the same batch.
Got it.
Then the outcome is we can say for this strategy, the batch size is very high because the model just trying to extract batch related information.
Any other questions? Okay.
I'm sorry, one more.
Go ahead.
Have you tried this on other, like, so in a medical application, you have different labs with different sample prep, and so you have the same batch effect. Have you tried this in other contexts, or is this your test case basically?
You mean like other cases such as?
Do you have other test cases? I'm thinking it would work for other test cases, and I'm wondering if you happen to have tried any other ones yet.
We have tried this method on JUMP, but JUMP is a bigger set including images from multiple institutions. So they are actually have a higher batch effect. From our result, using cross-batch sampling, it actually works much better than using regular binary.
Thank you very much. So the next talk is Grant Camo, learning interpret of single cell morphological profile from 3D cell painting images and rivet Gopalakrishnan from MIT.
Sorry, could the previous presenter stop sharing so I can't share this one?
Oh, yeah.
Oh, you're fine, you're fine. Thank you. Okay, cool. Thanks everyone. Hi, I'm Vivek. I'm a PhD student at MIT. This work has nothing to do with my thesis work. I did this while having the opportunity to intern at a really awesome imaging and microfluidics startup in Cambridge called Zeller Biosystems. So if any of this is interesting, definitely check out Zeller. The opportunity to do this work was all through them. Yeah. Okay, cool. So yeah, today I'm going to talk about grad-camo or grad-chem overlap, which is a method for assessing how biologically relevant your learned morphological profiles are from whatever deep learning architecture you choose to use. And in particular, this is a cool way for seeing if your morphological profiles are related at all to the cells of interest, or if your model is perhaps learning to cheat in some unexpected ways. So this is specifically pertaining to 3D imaging. The reason why we care about Z-stacks is because they're a much more relevant model of in vivo biology, especially if you hook them up to a microfluidic system and can passage drugs through them over time. But the workflow is basically the same as anyone who works on 2D imaging. You start with a big Z-stack and you want to learn some morphological profile of these cells and use that to elucidate some downstream biological tasks. But of course, when we talk about morphological profiles, we really just mean feature vectors here in some d-dimensional space. And there's some algorithm that takes whatever images we give it and gives us these numerical representations of the cells that we have. And from these numerical representations, we seek to answer all of these downstream biological questions. To break it down into just a bit more detail, what we particularly care about in this application is single cell morphological profiles. So from your Z-stack, you can get single cell segmentations very easy using any segmentation network like CellPose, for instance. And then using 3D crops centered on a specific cell of interest, you can train whatever model you want to look at downstream morphological profiles. In this talk, I'll talk both about weakly supervised models, or supervised feature extractors, as well as unsupervised feature extractors for 3D images. So to illustrate how we got to grad-camo, I'll walk through an experiment that I did over the summer during this internship, where we were trying to do this feature extraction task, and we ended up with some really surprising results that motivated us to look at this in the first place. But just to start from the beginning, single cell segmentation with CellPose was incredibly easy for doing 3D segmentations. I just want to give them a quick shout out because I feel like it's very rare that you actually download someone else's model off the internet and just run it on your data and it actually works with minimal hyperparameter tuning. So it's a very, very impressive tool and I really appreciated having it open sourced. So just to show it working on 3D data, which is kind of a more niche application of CellPose, I'm running through Z-stacks of the images that we had. So this is just iterating over all the various slices in the stack. What you can see on the left is the input image that we fed into CellPose. And then the other three images are the outputs or the segmentation masks that we get. The red circle, or the red square, sorry, at the top left is just blown up in the bottom row, so you can see this in more detail. And so from these really accurate 3D segmentations, we can now go ahead and extract 3D crops of all the various cells that are in our data set. So this data set is a big set of liver cells, and they're treated with basically 6 different concentrations of a drug. And so we get all of these various 3D crops centered on cells of interest throughout the data set. And here are the other 3 concentrations that we have. And so we end up with this really great array of single cell crops. In total, there are about 50,000 cells in this data set. There are 2 wells for each concentration of interest, and within each well, we image 4 times, so 4 samples per well. And then we ended up with about 50,000 cells, and the setup is basically what you'd expect. So this is a direct screenshot from a paper called Deep Profiler, where you feed all of your cell painting images into whatever deep learning you want, and you try to regress the treatment label that you gave to each cell. The only difference here is instead of feeding in 2D cell painting images, now we take these 3D Z-stacks and feed them into a 3D deep learning architecture and try to regress the label. And so here, I'm going to show a quick example of what the profiles that you extract from each of these cells look like. So this is specifically on the testing images. The way that we did this is we trained on cells exclusively from one well and then tested on cells from another well. So there's no mixing between wells. And so what this animation shows is the profiles that you learn after just a few iterations of training this model. So this model takes about like 1.5 hours to train, and then by the end of it, you have perfect classification accuracy on whatever labels you're trying to get. So in gray are all of the control cells, and so we use a batch normalization effect to just treat the variation in the control cells as noise and flatten all the other cells onto that plane. And we can see very great separation. The clusters look wonderful, and so at this point I thought, okay, cool, internship project is done. But one of the things that I was very curious just to look at is looking at some of these like explainability metrics, if you could figure out how the model arrived at these conclusions in the first place. So by far the simplest way that you can do these interpretability analysis is with a technique called GradCam, which I'm sure most people in the room are familiar with. In case you're not, basically what you do is you just look at whatever label the model regressed, you use the gradients with respect to that label to weight your activations, and then what you end up with are these very nice saliency maps that tell you basically where the model was looking. So here, red means the model was paying a lot of attention to this region of interest, blue is very little attention at all. And I think when I saw these images at first, it was like a very interesting revelation. For some of them, the model behaves exactly as you expect. There's a single cell crop focused on some image in the center, and all of the model's attention is placed directly on that cell. In the middle, you can see kind of like a different sort of phenomenon. Here, the model is more interested in a neighboring cell, which still might capture something biologically relevant. Perhaps for some phenotype, the interactions of cells is important to pay attention to. But for the final row, there is just clearly something that we do not want happening with our morphological profiles. Whatever is in the background, some imperceptible noise, is what the model uses to achieve this perfect classification accuracy on this task. And so you can just, here's some more examples that show the same effect. And it's just really surprising when you visualize these, how many of the GradCam maps are focused exclusively on the background, not even on neighboring cells. And so this inspired a interpretability metric, which I call GradCam overlap or GradCamo. So as I showed earlier from CellPose, you can just effortlessly get these segmentation masks. And so one of the ways that you can quantify this GradCam map is just ask what proportion of the GradCam map is centered in the segmentation mask of the cell. So basically how much of the red stuff is within the white segmentation mask in the bottom row. And the more this overlap is, the closer the score is to 1, which means whatever morphological profile you learned is very much centered on the cell of interest. And the closer that this score is to 0, the more it's saying that it is outside of this segmentation mask. And so here is just a quick visualization of if you break down GradCamo and compute it over all the cells in your testing data set, what you end up with. So I think there's some really interesting findings here. So in the figure over on your right, there is this plot of the GradCam scores for every single cell in the testing data set. And on the x-axis are all of the different concentrations that one might have treated the cells with. And you can see that for a lot of these cell concentrations, the model's GradCam scores are very low, like below 0.2 or like below 20%. So for the vast majority of cells in these groups, the model was using information that did not come from the central cell of interest in order to predict what the concentration of drug that the cell was treated with. I do also want to highlight that grad camo can sometimes give you a result that you may not want. So just to highlight that, I think the middle illustration here is particularly interesting. So this is a cell with a really strange morphology. It's not round, it's like got this sickle shape. And where grad cam is really showing that the model is paying a lot of attention is to basically the missing center of the cell. So this is like a cool way of looking at like there actually is something different about this cell, but it doesn't perfectly overlap with the segmentation mask. So in some case like this, grad camo may not be giving you the interpretability metric that you want, but for basically every other cell that's visualized here, they're just round cells, normally looking. And for the vast majority of them, when you use these supervised learning methods to extract morphological profiles, they're not overlapping with the central cell of interest. So I know a lot of people are probably thinking, well, we've heard a bunch about like the self-supervised stuff. Weakly supervised is very last decade, we've moved on. So I think that is very much a fair criticism. When you look at the attention maps that self-supervised methods give, they are much more concentrated on the cells of interest. So this is my favorite paper that's done the self-supervised thing. This is called single cell dyno. Oh, sorry. So in this case, what they're looking at is if you can figure out the cell state using just these dyno features. And for 2D images, they work absolutely great. So what I tried to do as well is to create a 3D version of dyno and get it working on the 3D images that we have. And so you can do the exact same framework, just get, okay, sorry, what?
You're on.
Oh, okay, cool. Thank you. So you can take the 3D props you have, apply basically all of the same augmentations you do in Dyno, and now just train a 3D vision transformer. Unfortunately, and I think for some very interesting reasons, this does not work very well at all on the 3D dataset that we tried. So whereas the previous model I showed you in supervised fashion trains in about an hour and a half, this trained for like 2 days and learned nothing. So I think there's a couple of reasons for this. The first is that a lot of the papers that use Dyno and stuff like this are starting from pre-trained models, either pre-trained on ImageNet or pre-trained on the massive amounts of cell painting data that exists in 2D. And so when you have this massive amount of pre-training, you can actually go ahead and leverage that when you're training on whatever data set you have. For 3D, we have nowhere near that amount of data for doing synthetic pre-training. The data set I showed you here has only 25,000 images in the test set, which is nowhere near enough to train these self-supervised models. So it's one knock against the self-supervised approach. You just need a lot of data to scale this. And so for 3D paradigms, this might be preclusive for looking at these things. But the upshot of all of this is that basically grad camo gives you a way of auditing whatever deep learning model you're using to extract your features. It applies for both 2D and 3D cell painting images. It works for self-supervised and supervised learning models. The idea is that it just gives you a way to interrogate if the features that you extract for each cell actually correspond to that cell in the first place. And I think the most interesting takeaway is that for supervised models, it clearly demonstrates that sometimes these models can cheat when you run them or just train them exactly as you would think to train them naively. So it gives you a good way, I think, also to evaluate all of the different architectures that one might try. There's an infinite search space over models that you could try for extracting features, but grad camo might give you a way for saying if one model is better than another at extracting biologically relevant features. So yeah, I'm happy to take any questions. Thank you very much. Yes.
2 questions. First, is this published yet? Really interesting talk.
Oh yeah, it's published here.
Okay.
Yeah. yeah. There's a paper with the proceedings, and it's also on archive. And there's a GitHub repo with more results.
And then second question, this might be a naive question, but did you benchmark this against any very low-powered algorithms like the SIFT algorithm for feature extraction, or did you go straight to these more complex neural networks?
I just went straight for the supervised feature extractors. The reason being for cell profiler in particular, or any of these things, it's just for cell profiler, it's quite hard to actually get good 3D features out, I've found. And so jump straight to using deep networks to extract features for 3D.
Great.
Yes.
Hi, so first of all, as I was wondering, in the beginning you showed the LED specs, and it looked quite a bit then when going to the LED specs, you could see different focus points.
Yep.
And I was wondering is like, how much is it actually like looking at 3D architecture and how much is it you get a slice that's in focus for the cell of interest?
That's a great question. I think it is mostly, the attention of the model is mostly centered in the center of the Z-stack where you get the most like in-plane resolution. If you actually look at the GradCam maps, a lot of the attention is very much in like the very center of your crop. So yeah, where you have these blurry images, there is less attention paid. Yes.
First, a quick technical question and a comment. So when you calculate your metric and the overlap, are you taking the mass of a continuous image, or are you taking a continuous cam map, or are you taking like a threshold and then doing straight overlap?
Oh, so this is like so the cam map that's colored up here at the top is just like per pixel there is a cam score basically.
Right.
And so like element-wise multiplication with the binary map, and then sum that up, and then divide by the total mass in the cam map.
So it's a mass of a continuous.
Yes,
map.
exactly. Yep.
Okay, great.
Mhm.
And Philip Isola is
Mhm.
at MIT. He's here and he trains models with noise.
Yes.
And so presumably you could train 3D models with noise because you don't need images. You just need noise generators.
Are you talking about the model that achieves like state-of-the-art on ImageNet, but trained on like the outputs of Midjourney?
No, the outputs of noise generators.
Just pure noise. Okay, I don't know this work.
Well, he's
but-
here, so then he's at MIT, so you should talk.
Yeah.
Isola, Isola.
Okay, I'll go to his office hours sometime too. Okay.
Okay.
Cool. Thank you very much.
We will finally move on to the last invited.
Thank you all for staying. Let's see. Dr. Peng, are you there in the Zoom room? See if our invited speaker is still there, or maybe he gave up on us. We are so awfully late, but the talks are interesting, so I hope people find that useful. Dr. Peng, are you online? Oh, what happened? Let's see. Why am I seeing this? Let's see if our invited speaker is online. Maybe he indeed gave up on us. I know he's traveling. Dr. Han Chuan Peng, are you in the Zoom room? We are more than half an hour late. He's traveling, so maybe it didn't work out with his travel. We have to see. Oh, that'd be too bad because it's actually a very interesting talk on 3D. Are you coming? Okay. How long? 6? Okay, I'll wait here. Yeah. Okay, thank you. Seems like our invited speaker is not currently in the Zoom room, so let's move on with the program. And maybe he has to travel, cannot wait for us anymore. So I think continuing our tradition of supporting people who do work that may not be ready for submission but want to get feedback from the community. So let's have our work in progress presentation. Adriana, are you in the Zoom room? You're here, great. Yeah, let's get started. You are going to project from this and then use my laptop for the audio. Yeah, let's connect to this one. USB-C.
I don't know if I'll be able to output these laptops.
I see. So you can project.
So I think it's coming up.
That's okay. Yeah, and then share your slides in the Zoom room and put it in. So it will drop. Yes, it's okay. Yeah, and here is showing you. You just need to. Now we go to slideshow.
Okay, and it shows up. Oh no, it shows up as my. I don't know how to get it to not show up like that. Sure.
Slideshow, see if that helps.
We're seeing the slides.
Oh, you're seeing the slides. Oh,
Zoom
in
room,
Zoom it
somehow
shows
see
the notes. I don't know, maybe that's okay.
Showing results.
It's showing that just
Oh, I see.
Somehow.
Okay. Oh, here we go. All right. No, it's fine. No. All right. Hello. Okay, so my name is Ariana Nawabi, and I'm a computational scientist at the UT Southwestern Medical Center's Whole Brain Microscopy core facility. And today I'm going to be presenting some work I did with Matthew Kenwood, who's a PhD student at the UT San Antonio Health Sciences Center, and Denise Ramirez, who's the director of the core facility at UT Southwestern. And specifically, I'm going to be talking about removing brainstem sections from serial to photon tomography images of the mouse spinal cord. So I'm going to talk a bit about what we do with our images and why we want to remove the brainstem, as well as some of the challenges we had to overcome to be able to do this. So one of the advantages of volumetric imaging is that it allows researchers to get a more comprehensive understanding of their samples by preserving depth-wise anatomical relationships in the tissue. The other cool thing that we can do with volumetric images is we can simultaneously quantify signals in hundreds of regions of interest by mapping our volumes to three-dimensional pre-annotated common coordinate frameworks, which we refer to as atlases. Now, when people are doing this kind of analysis and quantification in their spinal cords in particular, they typically only want to analyze signals that come from actual spinal cord tissue and not from surrounding tissue such as the brainstem. However, the brainstem and the spinal cord share a close anatomical boundary, and it's one that can be difficult to delineate on a macroscopic level. And for some of the labs that we work with, they want to study the very topmost levels of the cervical spinal cord, so it's absolutely critical that all of those topmost sections are in the image volume. So to ensure that this happens, we routinely include brainstem sections in the dissection volume, so that way we can make the delineation between the brainstem and the spinal cord on a microscopic level. However, if these brainstem sections remain in the image volume, then it can lead to downstream errors in the quantification. So it's important for us to remove them before we proceed with further steps. Typically, this would be done by someone manually saying, okay, which sections are brainstem, which sections are spinal cord, and then we would take those out of the image volume. But this takes the time and effort of a trained expert, which isn't ideal because they could do other things with their time. So we want to automate this removal. And basically these days, the default way to automate a classification task would be with a neural network trained classifier. However, for our specific use case, there are some challenges with this. So to start off, our data set is highly imbalanced. And this is because typically there are only a couple of brainstem sections included with each spinal cord dissection, although the exact number can also vary widely from sample to sample. The other issue is, since we're a core facility, we receive samples from a wide variety of different client labs, which might have different staining patterns or different preparation methods. And so this can lead to a wide variety in overall appearance and global illumination patterns. And since each of these samples comes from a separate animal, we have a limited number of samples to combat batch effects. So we tried a lot of things to deal with this. And what ended up happening was our classifiers kept either overfitting to specific samples in the data set or just classifying everything as spinal cord. And neither of those situations were particularly useful for us. So what actually ended up working with us to remedy this issue was a particular data augmentation strategy. And what we do is we interpolate between images in two different ways. And we do this within each class in the data set. So the first kind of interpolation that we do is what we refer to as intra-sample interpolation. And we do this to combat one of the limitations of our imaging method, which is that there is a 75 micron gap between slices. So with the aim of capturing more of the possible anatomy between serial sections, we generate new images interpolated between physical planes in the image line. I kind of just want to add here quickly, there was someone who was talking about doing something similar to this earlier, and someone asked what like downstream applications of like a 3D generative model would be, and this might be one of them. So it might be a better way of doing this, but for now we're just doing basic interpolation. Then the other kind of interpolation that we do is what we refer to as inter-sample interpolation. And in this case, we interpolate between images from two different samples. And we do this to combat the model from overfitting to the specific distributions and specific samples by generating images that are effectively new combinations of those characteristic sample distributions. And then what we can then do is we can combine these two methods by first doing intra-sample interpolation on all of our data, and then doing inter-sample interpolation on all of those samples, and we can basically create a rather large augmented data set. And what we see when we visualize the data is that we've reduced a lot of the sparsity that was previously present in the data set, and we do this with the aim of preventing the model from overfitting to specific clusters in the data. And so when we actually do this on our classifier, what we see is an overall improvement in our classification. So in this plot, specifically, I first reweighted the data to deal with our class imbalance, and I've done this by increasing the loss penalty on the minority brainstem class. We see that after doing this and introducing our sample augmentation method, we're able to get increased performance on our data set. The other interesting thing that we found that we could do is instead of reweighting the data with the loss function, we can actually reweight the data by upsampling our minority brainstem class by including more interpolations of it in the data set. And we see that this also offers an additional boost in our classifier's performance. So this is a work in progress, and we still have some future work that we want to do. We want to do some more hyperparameter optimization on the specific data set for the specific application. So that's going to include like adjusting the weighting ratio, as well as we want to see what the effect of increasing the number of interpolations is. And we kind of assume that we'll see a plateauing effect, but we haven't actually tried it out. So we want to also test that out. We have research groups that we work with that are specifically interested in the cervical spinal cord and not any of the downstream regions. So they would like to also remove thoracic cord sections that might end up contaminating their image volume. So we're working with someone, or we're actually working with Matthew right now to start annotating those and introduce that as well. And we have light sheet volumetric images. So we are right now in the process of acquiring those images. So we're hoping to see if this also extends well to light sheet. And yeah, thank you for taking the time to listen to me, and I'm happy to answer any questions.
First, it's wonderful to see a concrete targeted application. That's great. A couple things. First of all, you have this long spinal cord, tiny brain stem, and is there a reason not to ignore like 80% of the negative data because it's far away from the brainstem and so it looks different, or is it still valuable training data?
Yeah, that's a good question. And there's kind of two answers to that. One of them is that we, like in our preliminary experiments, we did see that including it as well, and then reweighting was like better for the performance than just like ignoring it. The other reason is we want to extend this to the thoracic cord as well, and that data is going to be valuable to be able to distinguish between thoracic and cervical cord sections. So we wanted to first like trial this out on the brainstem and then say, okay, we can also extend this to the thoracic cord because that will also be a minority of sections, but something where it's very important that we have all of the cervical cords like morphological information.
And the second question is when you show the point clouds kind of as a result of having all your augmentations, I mean, first it works better, which is like the ultimate proof, right, that it's good, but it filled in, there was kind of the C shape, and then you filled in to make a convex, it filled in to make a convex structure. Is there reason to believe that convex structure is a more accurate representation of reality, or is that?
So
were those added points biologically real in some sense? Does that make sense as a question?
So I wouldn't say that we know for sure that those are necessarily biologically relevant. I can say it's like in one example, if I go back to the plots, like this section right here is images from, we have two twin systems. So this one and this one are images from one of our twin systems, and then this distribution is images from another of the twin systems. So it kind of like interpolates between the two characteristic noise patterns that we see between those two systems. So it might be more robust to like different types of noise by having, I guess, a denser manifold in those areas. And so that was kind of the primary goal of this, was we don't know what we're going to see in the future. So we want to have the data set that we're training on be as dense as possible so that any gaps that might be possible to be in the model will most likely somehow be encompassed. So I wouldn't say that they're like necessarily biologically relevant gaps, but rather the idea is that if we just fill in as much information as possible, then the actual distribution that the model is learning will be smoother and therefore more likely to be able to accommodate types of data that we haven't seen before.
Total sense. Yeah, wonderful work. Thank you.
Other questions or suggestions from the audience? Thank you so much.
Thank you.
Yeah.
Let's see where we are. And I think let's move on to, so we've been doing this, I think, for at least 5 years or longer. There was a paper publishing a data set, and afterwards they've been organizing a challenge every year and report out the challenge result. And let's see if I'm projecting. And so Athava, you are online, right? Are you ready to share your screen? Yeah.
Yes,
Thank you.
I'll just start sharing my screen.
Yeah, so you are going to report out the cell tracking and mitosis detection dataset challenge. Please take it away and let me share the room with you. Yeah, you can start.
Thank you, Dr. Chen. Hello, everyone. My name is Atharva Peshkar, and today I'll be announcing the cell tracking and mitosis detection dataset challenge results for this year. So we'll begin by discussing the motivation behind the challenge, followed by an overview of the dataset and evaluation metrics, and finally I'll announce the challenge results. We often see biologists analyze these types of live cell videos to examine cell behavior and interactions. Two critical tasks or algorithms that help them with this investigation are tracking cells across the video and characterizing cell life cycles, such as mitosis, where a cell divides into two new cells. These tasks are crucial in various studies, including tissue formation and repair, wound healing, cancer treatment, drug development and analysis, immune responses, and many more. And while there has been a significant development in the state of the art for cell tracking algorithms, we know that for algorithms to be successful these days, a large and diverse dataset is crucial. We see evidence of this within the broad scope of mainstream multi-object tracking, where objects such as humans and vehicles are tracked. A few examples include MOT, Kitty, and UIDIT track datasets. In comparison, we have 3 popular datasets for cell tracking as shown here. However, there's still work to be done. For example, in terms of imaging modality, across the 3 datasets combined, there is a good representation of phase contrast and fluorescence microscopy videos, but only one, that is CTC, comes with an evaluation server. Furthermore, these datasets lack sequences obtained using differential interference contrast microscopy, or the DIC modality. And finally, all these datasets provide segmentation masks, which are more stringent and expensive for expansion compared to detection boxes. And so, to fill in this gap, we introduced our CTMC dataset in this workshop. Here's a preview of the dataset. It contains 14 different cell lines, and as you can see, it contains a diverse set of cells with varying shapes and sizes. This dataset was collected from the Nikon Microscopy website, and all the data was obtained using the DIC modality. The cell lines have been collected from various animals, such as humans, rat, mouse, rabbit, etc. And in terms of applications, it covers a variety of them, including wound healing, cancer research, vaccine development, etc. The dataset also presents a variety in cell types, including epithelial fibroblasts, myoblasts, and endothelial. Here are some further details. So our dataset contains 86 sequences or videos, 2,900 cells, 14 cell lines, and over 150,000 frames. Compared to other datasets, we can see that CTMC provides a larger number of sequences and frames, as well as a more diverse range of cell lines. Additionally, it complements the existing datasets by offering DIC sequences, which are absent in other datasets. To annotate this dataset with two tasks, that is tracking and mitosis information, we utilized our in-house annotation tool based on Batik, which is a popular video annotation tool from UC Irvine. 2 graduate students were tasked with annotating all the 86 videos, spending over 164 hours in the annotation process. And the final result after interpolation contained about 2 million detection boxes. We hosted our challenge on the MOT benchmark platform, which has been hosting similar challenges for various natural video datasets for over 5 years now. And here's how the dataset was split. So we have a total of 86 sequences with 47 sequences in the training set and 39 sequences in the test set. It's important to note that all the 14 cell lines were distributed between both the training and the test splits. This year, we followed the same winning criteria as the MOT challenge. That is, we used a metric called multiple object tracking accuracy, or MOTA, which in general evaluates the coverage of all the objects across the video. Specifically, it considers the number of false positives, false negatives, and the number of times object identities were incorrectly switched. MOTA score ranges from -infinity to 100, and the score can be negative when the number of errors made by the tracker exceeds the number of all the objects present in the scene. Moving on to the challenge results. This year's challenge received just one submission from Team Mimotor Cell, achieving an average MOTA score of 41.8%. However, the submission did not outperform the winning submission from last year by Team PP Bot Sort, which achieved an average MOTA score of 51.6%. So for this year, Team PP Bot Sort retains the top position on the leaderboard. They've shared their GitHub page if you're interested in learning more about their method, which they have tested both across mainstream and cell tracking datasets. Here's how the leaderboard looked for the top 3 entries, along with this year's submission, all of which beat the baseline performance. We also observed the performance based on a different metric called IDF1, which focuses on object identity across the video. Once again, all these trackers, the top 3 entries in this year's submission, beat the baseline. Digging a little deeper, we wanted to analyze the difficulty level of the sequences based on the performance of the top 3 trackers on the leaderboard. And here we can see that about 50% of the sequences were still challenging for the trackers. That is, they achieved a MOTA score less than 50%. And here's an example of 2 challenging videos from the top performing team. When the cell begins to change shapes often, that's when it gets challenging for the trackers. And these trackers fail to maintain the identity of the cell throughout the video. This suggests that tracking deformable object still remains an open and challenging problem. And that brings us to the end of our discussion. Please feel free to reach out to us if you're interested in learning more or collaborating. And you can find our dataset on our project page listed here and continue participating on the challenge website. I'd like to thank you all for your attention. and the team for their participation. We would also like to thank our sponsor, Silicon Valley Community Foundation, and Chan Zuckerberg Initiative for funding this work. Thank you.
Yeah, thank you so much for the presentation, for sharing with us every year and also doing the challenge every year. It's a lot of work. Yeah, thank you for doing that. So we are miraculously actually ahead of time. So I think it's been a long day. Let's wrap up here. Thanks to the audience for attending and staying till the bitter end. And thanks to all our speakers and presenters and authors for contributing. And also thanks to our organizers who really made sure every year we have an excellent and really rich program. Next year will be our 10th instance. Hopefully we get accepted. And we look forward to see you there again. Maybe we should try to do something special for this special 10th year anniversary. Yeah. Thank you all. We hope you enjoyed it and hope you enjoy the rest of the conference. We'll see you next year. Now it's time to enjoy Seattle, enjoy dinner.